and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button.
Well, good morning, everyone. Good morning, good afternoon, or good night, depending on where you are in the globe. And welcome to day one of our Generic Drugs Forum, GDF, we love to call it, 2026, right here at our home in White Oak campus. So I see a good number of unfamiliar faces. Who in the room as an attendee, this is the first time on White Oak campus? Oh, raise your hands, raise your hands. All right, welcome. Hope you like it. So again, it's good to see so many of you in person with us and online today. And we certainly appreciate your commitment in attending this event from wherever you are. We recognize that there are a lot of challenges in different time zones, etc. So for those in the room, the on-site Wi-Fi is there, FDA guests. And you do not need a password for it. We want you to get online in this room, so no password. And then we'll talk about the SBIA events that come in a bit. My name is Brenda Stoddard. I'm with the CEDAR Small Business and Industry Assistance Program, SBIA, better known. And we like to use that acronym in all of our materials as well. So when you see it, think, CEDAR, SBIA. Which operates within the Division of Drug Information and which resides in FDA's Center for Drug Evaluation and Research. If you had a chance to look at the agendas, we have them outside flanking the exhibit, tabletop exhibit there. We have a truly tightly packed agenda for the 2 days. So I'm going to be as brief as possible, which is difficult for me because I like to talk. But it's a great pleasure, along with my colleagues from the Offices of Generic Drugs and Pharmaceutical Quality, to be with you here over the next 2 days. And we entreat you to make the most of this experience, interact with the subject matters for those of you who are here on campus, and for those of you online, submit your questions. We'll get to as many questions as time allows. Learn all that you can and network in person and online as well when the chat room is up. We are also offering live YouTube streaming. So if you want to, just text your colleagues or email your colleagues and invite them to attend as well. Okay, so as of this morning, we had over 3,300 registrants representing 94 countries. And an interesting factoid is that 43% of these registrants have self-identified in the registration process as small businesses. And of those, 25% report having 1 to 9 employees. So that's good for us. We're targeting, we're reaching the right audience, even though our audience is universal. This here is a snapshot of our geomap for today's event. And the map is always quite invigorating to us because, you can't really zoom in on your screen, but you can see the concentration of the registrants. So it's a very powerful visual as to the impact and extent of our outreach. And then here you have our the QR codes for various social media avenues. It will also be displayed during the break. So at that point in time, you can feel free to click on it and see scan it and see what happens. Okay, so let's do some details and logistics that pertain to everyone, online and in room. So this is SBIAevents.com. It's the one-stop shop where you'll get answers to your technical questions if you are experiencing any. For downloading the files, namely the slides, accessing the survey link to obtain the certificate of attendance, and so much more. For online attendees, this is the same page that you used this morning to access the event. So please bookmark the page as you'll need it over these 2 days. If you have any technical questions about this conference, check the webpage first. We usually have some questions there that you may be able to apply. On our one-stop shop page, there are session evaluation links and also a final evaluation/survey, which will be activated this afternoon. And the survey link will also be sent out each day post-event conference. So look out for that email, please. Upon completion of this survey, an attendee can then download a certificate of attendance. Let's see what the next thing. Yeah, certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick is that the certificate must be downloaded immediately. Don't try to do the survey and then go back for the certificate because you're not going to get it. And note that both the evaluation and the certificate are only available for 2 weeks post-event, which means that the deadline date is May 7th. Now, recordings of the individual presentations of all over 43 speakers and panelists will be posted to SBIA's webpage once available. But in the interim, the YouTube links will be posted on the one-stop shop page. And for online attendees, submit your questions using the on-screen Q&A pod. Okay, now let's go to details and logistics for in-person. We already addressed the FDA guest, no password needed. We have 2 mics in the center. Line up of the 2 mics for your questions when the time comes. What we'll do then, and I'll explain it again later, is we'll take 1 question from each person in-house, and then we'll go to the question online. So hopefully everybody gets a chance to ask the question. I do want to emphasize that the questions, we can only answer questions pertinent to the material that is presented over these 2 days. Anything outside that realm, we probably will not be able to answer, and we definitely cannot answer questions to any specific applications. So please keep that in mind. Please silence your phone and use the internet sparingly, as it may negatively affect our online delivery. For those of you who pre-ordered lunch, please take advantage of the morning break to pay for those lunches at the kiosk. In that way, lunchtime, you'll have the full extent of your lunchtime, so you just go pick up your box there. Talk about upcoming events, SBIA upcoming events. We have 2 scheduled for May. May the 5th is an over-the-counter monograph user fee amendments, and that's a webinar for 1 hour. And then May 19th and 20th, we have our regulatory education for industry, READY for short. We have acronyms for everything. Annual conference. And this year is an emphasis on innovative regulatory strategies to advance medical products. And for the plenary, we will have participation by all 3 centers because we're offering 3 tracks: drugs, devices, and biologics. So it's a really very good. All our conferences are good. What can I say? This is the one that's offering 3 tracks. And for the plenary, we'll have representation from all 3 centers, featuring all 3 center deputy directors. And at this point, I'd like to have the slide up here to recognize those individuals who made this event possible. And as everyone knows, there are many names in the background, but couldn't fit on the slides. So thank you all very much, many of you who are in the room. My co-host for the next 2 days is Dr. Corey Adair. She's seated there at the SBIA table. And we have Will Tetlow from the SBIA team on technicals, AV, yeah. So just a few housekeeping notes. Bathrooms all the way down this corridor. Just follow the main corridor, past registration, past the kiosk, follow the main corridor, and it'll take you to the dead end, and the bathrooms are there. And a blatant plug for our SBIA Learn repository, where we currently post over 300 webcasts curated from previous SBIA conferences and webinars. So for this audience, you can also go back and get slides and presentations from previous GDFs. And at this time, I'm sure you're ready for us. How am I doing on time? 53 seconds. I'm sure you're ready for us to launch fully into today's agenda. And as a reminder, all of our speaker bios are available on the One Stop Shop website. But for now, I am honored to introduce our two keynote speakers. Our first keynote speaker is Dr. Darby Kozak, the Deputy Director for the Office of Generic Drugs, where he serves as a senior agency advisor in the development and implementation of FDA policies and long-range objectives for generic drug scientific programs and activities, including the development of a strategic plan for the generic drug program. And then our second keynote speaker will be Dr. Susan Rosencrance, who is an executive leader at the FDA's Center for Drug Evaluation and Research, with over 20 years of expertise in pharmaceutical regulatory assessment. Dr. Rosencrance currently serves as Deputy Director for Science in the Office of Pharmaceutical Quality, where she provides executive leadership for 5 sub-offices focused on the CMC portion of application review. Please help me welcome Dr. Darby Kozak. Thank you.
Thanks, Brenda. I'm honored to kick off this generic drug forum today and the opportunities to facilitate the American patients' access to these needed medications. As Brenda mentioned, I'm Darby Kozak, the Deputy Director of the Office of Generic Drugs. So today I'm going to cover 4 major aspects of the generic drug program and initiatives to improve patient access. I'll highlight some approval and submission metrics along with notable first generics from last fiscal year, as well as discuss important programs to improve efficiency and timing for generic medicine development and approval. I'll wrap up with a few ongoing initiatives to improve transparency and confidence in generic drugs. So I do want to start off with highlighting some encouraging outcomes. In particular, in fiscal year 2025, the generic drug program continued to have strong performance with the approval or tentative approval of over 939 ANDAs. Of those, 92 were first generics, 125 were complex generics, and this trend is actually continuing into fiscal year 2026. Over the last 6 months, we've already approved or tentatively approved 483 ANDAs, with 36 of those being for first generics. So the generic drug program continues to evolve since its conception in the GDF establishment in 2012. Notably, the volume of complete response letters has decreased slightly year over year, while information requests and prior approval supplements have increased as FDA and industry continue to work to reduce the number of review cycles and maintain the marketing of already established ANDAs. Even more notable is that the total number of generic drug actions FDA has taken has increased over the last 5 years, from approximately 9,500 in fiscal year 2020 to over 11,000 in the fiscal year 2025. So among the 939 ANDAs approved in FY25, there are several notable first generics that exemplify the impact of the work that we do together. In August last year, we approved the first liraglutide GLP-1 indicated for chronic weight management. GLP-1s and other peptide drug products continue to be a growing and critical market of considerable interest. In fact, earlier this month, we saw the tentative approval of the first generic semaglutide ANDA, and we're expecting to see the submission and approval of many more peptide drugs over the next few years. And another major milestone, we approved 3 generic iron sucrose injection products in 2025. Approval of these complex colloidal iron products came after considerable GDUFA research, as well as generic drug industry development. These generics provide greater patient access to rapid, effective treatment for iron deficiency in anemia. Regardless, each ANDA approval addresses a significant public health need while helping to ensure patient access to more affordable medications. The sustained volume of the generic drug-related submissions reflects the continued demand for generic drug development and marketing. We routinely receive approximately 18,000 submissions a year, over half of those being ANDA supplements that continue to help ensure medication labeling has up-to-date information or that manufacturing process includes the latest quality controls. One of our most significant achievements has been the dramatic reduction in refuse to receive actions. In fiscal year 2017, OGD refused to receive 142 ANDAs because they were not substantially complete. That was almost 11% of all of the submissions in 2017. Last year, only 3% of the ANDAs were issued or refused to receive. We understand the negative impact of an RTR can have on a generic application in the generic drug program, so we continue to provide guidance and presentations on common issues that can lead to an RTR. Probably the most common RTR issue can be readily prevented simply by acknowledging and providing justification when an application would deviate from general FDA guidance recommendations. Including this information supports that the application is being substantially complete and ready for filing. Then that justification will be evaluated during the ANDA assessment. So as Brenda also mentioned, one of the things here is, as a reminder, we always want to continue that FDA continues to develop helpful presentations like those being presented here at the Generic Drug Forum, and there's a repository of previous year's Generic Drug Forum topics, as well as other webinars and CRCG events that is a wealth of information for you to use. Last year, OGD launched a website as a similar one-stop shop for collating all of these resources for generic drug development and entitled and put into specific counters so that way you can follow a generic drug development program with specifics for style. I encourage your organizations to make use of this website as well as the other websites and resources and materials available. They're designed to help avoid common submission issues and create high-quality ANDA submissions. This benefits not only your company, our agency, but more importantly, patients waiting for these critical medications. So last year, we also announced an increased transparency pilot program for ANDAs with missed goal dates. This is initially focused on applications 60 days past their goal date, but we've evolved this initiative, and we now utilize this enhanced communication approach whenever complicated issues arise that will likely cause an ANDA to miss its GDF goal date. If an ANDA will likely miss its goal date, you'll receive notification on which disciplines may be affected, and you'll continue to receive regular touch points with your regulatory project manager on the status of the ANDA. Internally and externally, we've also built in clear escalation pathways to ensure that no ANDA application falls through the cracks, and we can have continued sort of action on all applications. So one thing I'm really pleased to report is this has been a major incentive and focus of the last year, is that of last quarter, OGD only had 2 ANDAs that were more than 60 days past their GDUFA goal date, and we aim to make that 0 very soon. The number one issue we saw last year was actually due to data integrity investigation of the studies conducted at a CRO. This not only caused ANDAs that use that CRO to miss their goal dates as FDA conducted its investigation, but in instances where the type of studies raised data integrity concerns, it also required applicants to repeat those studies. And this is a real core reminder for industry of the importance of vetting and selecting a reputable CRO when conducting those sorts of studies. So I'm also really excited, I think this is something we've been waiting for quite some time, by the recent Q1, Q2 legislation that was enacted in February this year, as it enables FDA to identify to applicants a specific inactive ingredient and amounts in a generic drug formulation that don't match its reference listed drug. Now, a reminder, this applies to all products required to demonstrate formulation sameness, such as those intended for parental, ophthalmic, or otic use, but also applies to those formulations where sameness is a part of a bioequivalence approach. So generic applicants, this means you'll receive more specific formulation feedback from FDA that should reduce the regulatory guesswork and facilitate more timely generic drug development. You should actually already be receiving and seeing the impact of this as FDA has implemented this detailed Q1, Q2 feedback to all pending and submitted controls and ANDAs since the day that legislation passed. So one other thing I want to really speak candidly about, and this is one of the continuing challenges we face together, and that still continues to be the public perception around generic drugs. Healthcare professionals and patients continue to express concerns about generic drug quality, and particularly around foreign manufactured drugs. This perception gap is something we must address collectively. FDA continues to ensure high-quality standards in the approval and monitoring of generic drugs sold in the United States and continues to develop communications to improve public trust through greater transparency about our regulatory process and oversight. But we also need generic drug industry to be a partner in this too. The generic drug industry must continue to maintain rigorous quality standards, ensuring manufacturing consistency, and communicate early and transparently about potential issues with applications and marketing products, so that way we can build and maintain public trust. Before I close, I do want to mention an ongoing recruitment efforts. OGD is actively hiring pharmaceutical and public healthcare professionals. If you know a talented individual passionate about ensuring high quality, affordable medications reach American patients, I encourage you and them to consider careers within the Office of Generic Drugs. If you're a U.S. citizen, permanent resident, and are interested in joining OGD, you can submit your CV and cover letter to ogdjobs@fda.hhs.gov. This announcement will go throughout the rest of the day, so if you miss it here, be aware that you can see it a couple more times. We're highly encouraging folks to join our team. So in conclusion, I'm looking forward to the next 2 days of excellent presentations and panel sessions where we'll gain insight into generic drug development and assessment processes. I hope that you will take advantage of this unique engagement opportunity and remind you that these and other similar resources are available on FDA's website for future reference. As we look ahead, I'm optimistic about the generic drug program and our shared mission to ensure every American has access to the medications they need. Thank you.
Good morning, everyone. Welcome to the Generic Drug Forum, and it's a pleasure to have this opportunity to speak with you today. You may have seen this slide before. It's a standard slide that we have in OPQ that we like to show in public presentations. Over the past year, there have been a lot of changes at FDA, as you know. But one thing that remains constant is that everyone deserves confidence in their next dose of medicine. And pharmaceutical quality remains as important as ever for ensuring availability, safety, and efficacy of every dose. To begin my presentation, I'd like to acknowledge OPQ and OGD's continued dedication to excellence and sustained prioritization of patients. Working together, we have maintained a strong assessment performance in GDUFA. We achieved over 90% on-time completion for original ANDA submissions and amendments, as well as GDUFA-controlled correspondence. To sustain this performance, within OGD, we focused on two key strategies: expanding capacity and leveraging innovation. We began cross-training our pharmaceutical scientists to create workforce flexibility in assigning applications, and we are using temporary details to fill critical positions on an interim basis until we are able to hire permanently. And innovation has always been a cornerstone in OPQ, and we leverage it whenever we have an opportunity. We use advanced systems such as Knowledge-Aided Assessment and Structured Application, or CASA, to manage product and process knowledge in a comprehensive digital manner. And within the past year, the usage of CASA expanded to over 60 different dosage forms, encompassing 95% of all generic submissions. Also, AI tools such as ELSA have helped our review staff quickly identify, organize, and collect information. For example, we developed AI prompt engineering techniques to support complex science and risk-based evaluations such as nitrosamine assessments. These techniques don't replace scientific judgment. Rather, it makes a reviewer's challenging job easier and more efficient by pulling all the needed information together for them. Another innovation and transformative initiative is FDA's PreCheck. The PreCheck program reflects FDA's commitment to strengthen Americans' pharmaceutical supply chain and to accelerate access to critical medicines for patients. It's a comprehensive program designed to fast-track the establishment of new manufacturing facilities within the U.S. And a core feature is proactive engagement before facilities become operational. So rather than waiting until a facility is built and ready to submit applications, we're offering to work with manufacturers from the early design phases through the application assessment. We don't want to just inspect these facilities after they're built. We want to help ensure they're built correctly from the start. And as indicated in this slide, the pre-check program officially launched on February 1st, 2 months ago, and the submission deadline for initial applications was March 1st. Beginning July 1st, FDA plans to start engaging with those participants that were selected. This timeline reflects our commitment to moving this program forward quickly while also ensuring thoughtful implementation based on the stakeholder feedback we received. And of course, drug safety always remains top priority. And regarding patient safety, I'll talk briefly now about our efforts in collaborating with manufacturers to implement mitigation strategies to limit nitrosamine impurities in our medicines. As you know, nitrosamines are impurities found at low levels in water, food, and certain medicines. FDA and manufacturers share a common goal. We are committed to addressing nitrosamine impurities while ensuring continued patient access to needed medicines. This requires commitment from manufacturers to follow FDA's nitrosamine guidance recommendations, evaluate the risk of nitrosamines in their products, conduct confirmatory testing, and implement changes to mitigate these impurities when they're detected. In August of 2025, sponsors were asked to submit progress reports on their mitigation strategies for nitrosamines, and FDA is currently reviewing the information that has been submitted. If you want to engage the FDA on this issue, we encourage using established pathways such as meetings and controlled correspondence for application-related discussions. And today's generic drug forum, along with the upcoming GDF public workshop in June, are opportunities where we can exchange ideas and discuss nitrosamine-related challenges. It's imperative that we, regulators and industry, continue to engage to ensure safe, affordable drugs are available for all of us. Another good example of collaborative engagement is CDER's Quality Management Maturity Program, which fosters a strong quality culture mindset among drug manufacturers. We have seen from other industries such as electronics and automobiles that investment in robust quality management systems is a key driver for business success. It's a core component of modern manufacturing, and research confirms it reduces losses due to product rejection or rework. Last July, OPQ published a Quality Management Maturity, or QMM, white paper, which talks about investments in quality management initiatives within the pharmaceutical industry and how these investments have the potential to yield returns for both companies and public health. In OPQ, we have developed a prototype assessment protocol for evaluating a drug manufacturer's level of quality management maturity. In 2024, completion of the first cohort of 9 QMM site assessments demonstrated that this protocol could in fact distinguish differences in maturity. In 2025, we began the assessment of the second cohort of 9 establishments, and that's scheduled to be completed this month. And to date, OPQ has completed QMM assessments at 17 different manufacturing establishments. 2 months ago, in February, we began soliciting more volunteers for a third cohort via a Federal Register notice. We've received strong positive feedback on the QMN program, and we're eager to increase voluntary participation, especially from those regions or establishments we haven't heard from yet. So the QMN program helps us assess and strengthen manufacturing quality systems. But another critical component of ensuring drug quality is research. Generic drug development, especially for complex products, requires robust scientific understanding of equivalence. CDER's GDF research program provides the tools that both FDA and industry need to evaluate generic drug equivalency effectively. What makes CDER's research unique is our in-house laboratory capability. We don't just review applications, we also conduct regulatory scientific research. This allows us to proactively identify scientific gaps and address them before developing product-specific guidance, and it ensures our recommendations are grounded in real-world data. Our research is integrated across the drug product's life cycle, from product-specific guidance development through pre-ANDA meetings, ANDA assessment, and then post-approval. Whether it's a straightforward solid oral or a complex, long-acting injectable, our research provides the scientific foundation for determining equivalence, and we share our findings broadly through peer-reviewed publications, scientific conferences, and our guidances. It can be hard to measure the value of our scientific research in totality, but there are some undeniable examples that show its worth. Let's take cyclosporin ophthalmic emulsion for patients with dry eye, which had an annual market value of more than $2 billion. We invested $2.3 million in research to understand the product's complexities and developed appropriate approaches for determining equivalence. The result was approved generics that reduced cost and generated savings, providing an indirect return on our public health investment. Our research portfolio extends to some of the most challenging areas in generic drug development. It includes new approaches for peptides and oligonucleotides, testing for inhalation products, bioequivalence for topical and transdermal medicines, in vitro, in vivo correlations for long-acting injectables and implants, and analytical methods related to nitrosamines. This research-driven approach is another way we're accelerating access to safe, effective, and high-quality generic medicines for patients. So let me close with a self-evident truth. Generic drugs are essential for accessible health care. It's reported that generics represent 90% of U.S. prescriptions, but only 13% of cost. And in the past decade, this has had a savings of over $3 trillion for Americans. We value dialogue and collaboration, and public engagement such as today's forum strengthens our processes and helps us better serve patients. Together with industry, we can maintain confidence in generic products, keep patients the top priority, and ensure every American has access to high-quality medicines. Thank you very much for your attention.
Thank you. Thank you. Okay,
I can go off this way.
anywhere you want. Okay, so let's get started with the other speakers. What I'll do for the sake of time, I'll introduce all the speakers for the next session, and then we'll go in rotation, and then I'll introduce the speakers to welcome them back to the stage and our extra panelists as it occurs. So for the next session, we're going to have first speaking on control correspondences is Lieutenant Commander Marcia Fields from the Office of Regulatory Operations in OGD. Then we will have common bioequivalence. Well, you guys can read it. Then we'll have Dr. Zen Zhang, who's a master pharmacologist in the Office of Bioequivalence at OGD, followed by Commander Vin Feng from the Office of Regulatory Operations as well, followed by Dr. Eric Toom from the Office of Quality Surveillance within the Office of Pharmaceutical Quality. So let's welcome our first speaker, Commander Dr. Marcia Fields. Thank you.
Good morning and welcome once again to the Generic Drugs Forum. We're hoping you're feeling welcome by now. We've said it a few times. We hope this presentation will be useful to you in your generic drug development. The presentation is focused on control correspondence, but you may hear me refer to control correspondence as controls, submissions, or plainly correspondence. The goal is to equip you with information on ways you may avoid having your submissions declined for review. Outlined here are the objectives. I'll provide a definition for each type, review what it is expected in those submissions, give examples of the kinds of submissions that result in not being accepted for substantive review, as well as offer some references that you may consult beyond this presentation. For valid controls, the agency has committed to responding to at least 90% of level 1 submissions within 60 calendar days and at least 90% of level 2 submissions within 120 calendar days from the FDA receive date. So you may be wondering, what is level 1? Level 1 controls are defined here on this slide. And I'd like to highlight that you may send correspondence pre-ANDA submission as well as post-ANDA approval. There are some instances where after an ANDA has been approved, you might have questions related to post-approval changes that are not specific to your ANDA. These questions may also be submitted if they're not addressed in the already provided CDER guidance. Level 2 is defined here. And it includes instances for submissions if, for example, clinical content needs to be evaluated, if you're requesting a covered product authorization to complete testing and development, if you propose a bioequivalence approach other than what is in the published product-specific guidance, and in cases where other offices need to be consulted or other centers need to be consulted. When you submit your control correspondence, please date your documents. The date of your document should be within 7 days of the FDA receive date. So for example, if you had a submission and your date for that correspondence was this past Friday on the 17th of March, and then you submitted it today on the 22nd, it would be acceptable because it falls within the 7 days. The submission should have the company letterhead, and it should have complete contact information. We want to be able to communicate with you. The submission should state that your drug development is related to an ANDA submission, whether it's a future ANDA submission or if it's an ANDA that's currently in review. If the control is related to an existing ANDA, pre-assigned or approved, please indicate that in both the portal and in your documentation. The control should contain complete reference listed drug information, both in your documents and selected appropriately in the portal. In your submission, please refer to the product as it is listed in the Orange Book. If it is not listed in the Orange Book, it should be related to an approved suitability petition. Otherwise, it would be declined for not referring to a strength that's in the Orange Book or a strength that has been approved by that suitability petition. We have arrived at our first challenge question. Is it true or false that a controlled correspondence should be dated and submitted within 14 days of the date of the correspondence? It's false. It should be dated and submitted within 7 days, 1 week. For inquiries related to maximum daily dose of the active ingredient, please propose a specific value and share how you calculated your proposal. Please also include your supporting documentation. For the maximum daily dose of an inactive ingredient, the Office of Generic Drugs will review a maximum of 3 proposals at one time per applicant. That may be 1 inactive ingredient with 3 proposed levels, or 3 different inactive ingredients, each having 1 proposed value, or a combination of 1 inactive ingredient with 2 proposed values and another inactive ingredient with 1 proposed value. Please do not include supporting toxicology or pharmacology data, nor provide the full proposed generic formulation. These are common reasons that we decline qualitative and quantitative, or Q1, Q2, submissions. Tables should have a complete and consistent information. So, information such as hydration forms, units of measurement, they should be the same for each proposed formulation. Different strengths and different fill volumes should be submitted separately. If the Office of Generic Drugs has informed you that your formulation would not likely result in a refuse to receive ANDA submission, additional formulations generally are not reviewed. Some product-specific guidances recommend a specific bioequivalence approach if the formula meets criteria for sameness. These inquiries should not be requested as Q1, Q2 reviews. Rather, asking if the formula is suitable for the approach outlined in the guidance. For post-approval inquiries, you still should select the reference-listed drug that was the basis of submission in the drug product information field. If you have an assigned ANDA, please select yes to the question that your control is related to your previously submitted ANDA or pre-assigned ANDA. You will then be prompted to enter the associated number. The reference-listed drug and related ANDA numbers should be referenced both in your uploaded documents and selected in the portal. If your inquiry has previous related correspondences, please include them. For example, you may have had an attempt or a few rounds for Q1, Q2 formulations. Please include the previous controls that are related to the same strength that received a substantive response. Include that submission, the original documentation, any information requests, if that was applicable, your response to those information requests, and the final agency response. We're not interested in multiple copies, nor unrelated controls, or controls that were not accepted for review. Lastly, we decline to review a submission if you include those that were not accepted for review. So please just don't include those. Second challenge question: what should your controlled correspondence include? Any information requests sent by the agency and responses to information requests? Multiple copies of previous related controlled correspondences that are not related to your current inquiry? or references or copies, multiple copies of control correspondence that have not been responded to. You guessed it, your information requests, your responses to the information requests, please include those with your inquiries when they're related. For post-approval inquiries, you still should select the reference listed drug that was the basis of submission in the drug product information field. Sorry, I went back wrongly. Please upload a copy of your complete response letter. For post-complete response letter control correspondence, upload the letter. Sorry about that, everyone. State what the deficiency is. What is your control addressing in that complete response letter? If your letter was issued prior to GDF 3, you may submit inquiries related to nitrosamines. Usually controls with pending ANDAs are not valid. However, if you're asking to add a new strength or a new fill volume to your ANDA, this will be considered valid. A request for a covered product authorization to complete testing and development or questions related to nitrosamines would be valid controls if your ANDA is pending. Foreign applicants must utilize a U.S. agent. U.S. applicants may also use a designated agent. In both cases, a letter of authorization is required for this relationship. The document is valid for 1 year, and it should include full contact information for both entities. Please include a copy of this authorization with your control correspondence. When requesting a covered product authorization for testing and development, please clearly state if the samples will be used in humans or they will not be used for human use. The submissions for covered product authorization in humans should also include study protocols and informed consent documents. For device evaluations, please send the samples of the innovator and the proposed device. The control is not complete without receipt of the samples. The address where to send the samples is listed on the slide. To help prevent issues with the samples being delivered, we suggest that they be sent without a required signature and that they are not sent under temperature-controlled conditions. Submit the control only after you have received confirmation of delivery. Please reference the delivery confirmation in the control. Only one prototype of the device is reviewed at a time. Challenge question number 3. What should the letter of authorization include? Should it be on a corporate letterhead? Should it be dated within 1 year of the submission date? Contain full contact information, company name, title, address, phone number, and email? All of the above. Yes, all of the above. If you have concerns regarding your proposed active pharmaceutical ingredient, the form of it or the level of it, you may submit a control correspondence requesting the evaluation of the API. It is not part of a Q1, Q2 review. These inquiries for API may be submitted only after the agency confirms your inactive ingredients if your product is required to be Q1, Q2. Applicants may submit for a clarification of the agency's final response to a control correspondence. It is not a new separate control. It's rather a request of the original event ID. In your submission, include a copy of the original control, the information requests, your responses to the information requests, the letter of authorization, if applicable, and the final agency response. You have 7 calendar days after the agency's final response is uploaded into the portal to request a clarification. Be careful with waiver requests. Valid controls are those where the applicant requests waiver eligibility, for example, for BCS Class 3 products. A summary of things to keep in mind when entering information in the portal. If a product has a reference listed drug, select it. The way the company is listed in your documents should be the same way it's listed in the portal. This applies to both the applicant and the U.S. agent. Also, the point of contact should not be the same for both the applicant and the U.S. agent. Select Yes in the portal when asked if you are a U.S. agent so that you may complete the fields for the applicant information. Our final challenge question is a true or false. In a clarification of ambiguity request, you may ask FDA to review the new or additional information, as well as ask follow-up questions of the agency's response. That's false. So the agency will clarify the response, but it will be based on what you originally submitted in your original submission. So they will address how they could better communicate what they wanted to communicate to you based upon your original submission, not with consideration of new information or clarification from your point of view. Here are a few references. The guidances are listed here. Generic drug user fee authorization, our commitment letter that identifies what we've committed to do as far as deadlines are concerned. The guidance for covered product authorization. We have our websites listed here, and then there's also a previously recorded presentation regarding control correspondence that just highlights the changes that were implemented with GDF 3. I now welcome Dr. Zhang from the Office of Bioequivalence.
Good morning, everyone. I will be presenting common bioequivalence study design deficiencies and how to avoid them. After my presentation, you should be able to understand the study designs and the relevant guidances and identify common deficiencies related to study design and analysis. And you will also be able to identify unacceptable post-hoc modifications in BE studies and apply strategies to prevent common deficiencies and improve study success. There are many types of bioequivalence studies. So there are two main categories. One is in vivo BE studies. The other one is in vitro BE studies. In vivo BE studies commonly include in vivo PK-BE study, PD-BE study, and comparative clinical endpoint study. In vitro BE studies include in vitro release testing, in vitro permeation test, particle size distribution test, single actuation content test, and many, many others. In vivo PK-BE study is the most common BE studies. Approximately 50% of ANDAs submitted over the past 12 years include at least one PK-BE studies. When you design your study, you can refer to this guidance. These guidances. The first guidance is FDA PK-BE guidance. It provides a high-level framework on most the critical aspects of PK-BE study design. The second guidance is ICH M13A. It was recently published. This guidance is focusing on immediate release solid oral dosage forms, but the scientific principles outlined in this guidance can also apply to other dosage forms or route route of administration. The third guidance is ICH-M 1310, which recommend how to conduct, how to develop your bioanalytical method and how to validate it. The fourth guidance is FDA BE statistical guidance. It includes the high-level detailed recommendation for different types of in vivo BE study, how to statistical analyze in vivo PKBE study designs. It also includes other statistical methods for other types of BE study designs. In addition to these general guidances, you can also refer to product-specific guidance for your specific product. So currently, there are around 2,400 PSGs published. Among these PSGs, 61% of PSGs have in vivo PKB study recommendations. This is a list of main components of a BE study protocols, such as study objectives, study design types, study population, dosing conditions, washout period, blood sampling schedule, bioanalytical method, PK analysis, statistical analysis, safety monitoring, and others. So in my talk, I will be focusing on 5 of them: study design type, washout period, blood sampling schedule, PK analysis, and statistical analysis. This is the first example. It is related to end-on study. The guidance recommend for BE studies end-on subjects after the pre-specified number of subjects have been reached are generally not encouraged, except in an adaptive study design, with a pre-specified adaptation to add subjects and statistical methods to control the type 1 error rate and the nominal level. So one example shown here, applicants completed statistical analysis and they found the study failed. And then they add a few more subjects and to make the study pass. And this is called a post hoc modification of the study design, and the statistical analysis is generally not acceptable. How to avoid this deficiency? So first option is use a pre-specified adaptive design when the uncertainty is very high. And another option is, so for non-adaptive designs, any sample size revision must be implemented before statistical analysis begins. The second one is related to sample size. The guidance recommends the total number of subjects in a study should be sufficient to provide adequate statistical power for a BE demonstration in the proposed study design. Inadequate sample size is one of the most common causes of failed BE studies and often results in major deficiencies. Applicants have to repeat their study. So how to avoid this inadequate sample size? So first one is you can try to develop a test product closely comparable to the reference list, the reference standard. So in that way, a TR ratio will be close to 1. So you will get the sample size should be usually should be sufficient to for a successful BE study. The second one is estimate a reasonable TR ratio and variability. So for example, you cannot really use, assume your TR ratio is 1. So in that case, you most likely you will get inadequate sample size. So just use a reasonable TR ratio. The most commonly used TR ratio, estimated TR ratio is 0.9 or 0.95. The third one is account for a reasonable dropout rate in the sample size estimate. So in some cases, you may come across a high dropout rate than you originally expected. So in that case, you may start an investigation and see what causes this high dropout rate, and modify your protocol as early as possible, and also before the statistical analysis. The fourth one is to conduct a pilot study when appropriate. So in this pilot study, you should be able to get a good estimate of TR ratio and also the variability. So it will help you to design your pivotal study. The last one is to use a pre-specified adaptive design when facing significant uncertainties. The next case is related to washout, inadequate washout, which lead to significant carryover concentrations. The guidance recommends for crossover B studies, use a sufficiently long washout period, such as at least 5 elimination T-halves, and also so subjects will be excluded based on the 5% Cmax recommendation, meaning if the pre-dose concentration is greater than its respective Cmax, 5% of its respective Cmax, then subject will be excluded from the BE analysis, so which will reduce the study power. One example is that inadequate washout resulted in a failed study because of exclusion of many subjects based on the 5% Cmax recommendation. So how to avoid this deficiency? So first, consider a parallel study design if the drug product has a really long T-half. The second one is do not base washout duration solely on the mean T-half. So you should consider the variability of T-half, especially when the T-half is very long and the variability is high. You should give some buffer when you estimate this washout duration. Approaches in the protocol with scientific justifications. Also, this washout duration is already long enough. Fraction of pre-dose concentration or population PK is related to first point Cmax. The guidance recommends that for products with rapid absorption, collection of blood samples at an early time point between 5 and 15 minutes after dosing, followed by additional sample collections, such as 2 to 5 samples in the first hour after dosing, is usually sufficient to assess peak drug concentrations. However, if failing to meet this criteria may lead to exclusion of some subjects from the BE analysis. One example is that a large proportion of subjects have had Cmax at the first post-dose sampling time points of 30 minutes. So it indicates that the peak concentration was not adequately captured and the study failed. So how to avoid? So first, thoroughly understand the PK properties of the drug product, especially when this drug product is expected to have a rapid absorption. So if that's the case, include early time points between 5 and 15 minutes. And then if uncertainty is high, conduct a pilot study to better understand the PK properties of the drug product. Next one is related to PK analysis. The guidance recommends Cmax, AUCT, and AUCI for single-dose PKB study, and Cmax and AUC-TOL for steady-state BE studies. And there are also additional PK parameters to support bioequivalence, including partial AUC and Tmax. There are two case examples. First one is in a fasting PKB study, subjects with missing samples right next to Cmax were included in the B analysis. The inclusion of these subjects was not acceptable because the missing samples right next to the Cmax, meaning the Cmax estimate is not accurate. So we don't know whether Cmax is Cmax in that measured Cmax or Cmax actually from the missing samples. So usually that will lead to the exclusion of that subject from the B analysis. The second one case is in a steady-state PKB study, subjects who completed the study were excluded from the B analysis because AUC-TOL could not be estimated for these subjects due to software limitation. The exclusion of these subjects was not acceptable because software limitation is not a scientific justification. How to avoid? So first, pre-specify scientifically sound methods for calculating PK parameters in the protocol. So for example, when you calculate AUCT, there are different methods such as linear up, linear down, or linear up, log down method. It's better to pre-specify which method you use to calculate this AUCT. Secondly, pre-specify inclusion and exclusion criteria related to PK analysis. But this inclusion and exclusion criteria should be scientifically sound. So you cannot just so if it's not scientifically sound, even you include pre-specified inclusion or exclusion criteria, we may not agree with this criteria during the review. Lastly, minimize deviations in blood sampling, particularly at critical time points. Next one is related to statistics, covariance in parallel study statistical model. The guidance recommends the applicant may consider pre-specified inclusion of important demographic factors and baseline prognostic covariance in the statistical model for parallel study designs. One case example is that the study failed using the pre-specified statistical model without covariance, and then the applicant add covariance to their statistical model post hoc to obtain a passing result. And this is a typical post hoc statistical analysis, which is commonly not acceptable. So how to avoid? First, pre-specify covariance in the statistical model for parallel study designs. And if statistical model revision is necessary, revise the statistical model before actually conducting the statistical analysis. Lastly, is related to group analysis. The guidance recommends the statistical model should take into account the multiple group nature of the BE study, such as by using a model including terms for group, sequence by group, subject within sequence by group, period within group, and formulation. One common deficiency we see is that applicants didn't include group-related terms in the statistical model, and when they use their own model, the study passed. But when we during the BE review, so when we include appropriate group terms in the statistical model, study failed. So applicant has to repeat their study. So how to avoid? So if possible and if feasible, try to avoid using multiple groups. So just use one group if possible. And if multiple groups are necessary and unavoidable, then you include group-related terms in your in the final statistical model. So summary, so in my presentation, I reviewed key regulatory recommendations for PKBE studies and highlighted common deficiencies in PKBE study design and analysis, emphasized that many failed studies resulted from inadequate planning, and stressed the importance of pre-specified scientifically and statistically sound protocols. And we highly encourage proactive strategies to prevent deficiencies and improve BE study success rate. Last but not least, I would like to thank my colleagues Utpo, Bing, and Yuhua for their constructive comments to my presentation slides. Thank you. Yeah.
All right, good morning, everyone. So today I'll be discussing filing major deficiencies. The objectives for today is understand OGD's mission and DFR's purpose in this regulatory framework. Want to provide you with available FDA resources, recognizing filing major deficiencies, describing any stumbling blocks in addition to that or that could result in a major deficiency, and essentially providing justifications to deviate from any requirements and or recommendations. So OGD's mission is to ensure high quality, affordable generic drugs are available to the American public. And part of that regulatory framework is a filing review, which determines whether an ANDA is substantially complete to permit a substantive review. In the division filing review, our objective is to file substantially complete ANDAs, and that's ideally without issuing an information request. But we do so in a way that maintains and applies uniform review standards. So here are a couple of resources. Right off the bat, the first 3 bullets are very specific guidance related to RTRs. In addition to that, there are guidances regarding what information is needed for an ANDA submission, such as the content format for ANDAs. There's also a filing review map, and that includes a filing checklist for your use and review when you submit an ANDA submission. There are other resources that we use as part of that's mentioned in the RTR guidance, such as the stability Q&A. We have the stability testing of drug substances and products. There's also a content format of composition statements to help with your composition drug product that's submitted in Module 3.2P1. And then there's also a map I've included in there. It describes what we do and how we communicate certain deficiencies during the filing review. And lastly, product-specific guidances. And there's also one other guidance that I didn't mention, I forgot to mention. It's waiver requests in regards to pH adjusters related to ophthalmics, otics, and injectable products, which I'll elaborate later on. So before we dive into the module and the ECTD backbone, I want to make sure that you're aware that these following slides will have three components. The first is a major, which is a deficiency that would result in a refuse to receive. The next is a stumbling block, which actually impacts our ability to file an ANDA. And because of that stumbling block, it could actually result in a major deficiency, or in certain circumstances, it might go into information request in which the filing team needs further information. They might need clarification, action, or confirmation that data or information is in the original ANDA submission. And I want to add emphasis there that it's in the original ANDA submission. So as I go through all the slides, we'll be going back and forth between these three major key components: major, stumbling blocks, and information request. So let's dive right in. So Module 1. So Form FDA 356H is a required form. It should be signed and completed. If this form is omitted or unsigned, then it is a major deficiency. So the stumbling block related to 356H and signatures is a lack of appropriate signatures. Specifically, when you're a foreign applicant, you should have a designated U.S. agent, and that U.S. agent should sign or countersign the 356H. Another major deficiency would be having no legal basis of submission. So as generic drug manufacturers, you must be the same as the reference listed drug. However, if it's different from the reference listed drug and there's no approved suitability petition, then you don't have a legal basis of submission, and therefore it's a major deficiency. Now, one of the stumbling blocks, let's say there is a change that has been approved that to be different from the reference listed drug. Right, that's through the suitability petition. So it should be approved before submitting your ANDA. And when you're relying on approved suitability petition, you should have a docket number and a copy of the FDA's approval letter. The thing with suitability petitions is that there are certain circumstances where we would issue a minor deficiency where you have to administratively correct your basis of submission, because that approval letter will identify a reference listed drug and it should align with your ANDA submission. Sometimes that information isn't provided at all, so we actually have to issue an information request to ask for the suitability petition, the docket number, and the copy of the approval letter. But also you should note that there are instances where filing may ask you to withdraw that application if there is an NDA that's already approved for the same change as approved in the suitability petition. So just keep that in mind when we talk about Module 1. So moving towards the Module 2, the in vitro data component. So typically, this is the dissolution data for solid oral dosage forms. The major deficiency in this space is missing comparative both test and RLD dissolution data, and in the recommended media for both test and RLD and for each strength. And in particular, for ophthalmic solutions, there's missing comparative physiochemical data. Those two items are major deficiencies. if it's not submitted in the original submission. Now, the stumbling block is that comparator data, a dissolution, generally is provided in Table 5, and for ophthalmics is Table 17 for the physiochemical data. We find sometimes that the comparative data for each of these strengths is placed in another section, or may not be in the original submission because it could be anywhere. It doesn't really have a true ECTD home, but typically it's provided in Module 2.7 or Module 5. But the information request that we would be issuing is asking to point out the location of the data in the original submission. So as you can tell, there's a common theme here that it should be in the original submission for us to consider substantially complete. Moving towards to in vivo data. When we talk about major deficiencies, and I'm kind of going to move around a little bit, when we're talking about just providing a failed BE study only and not demonstrating bioequivalence to the brand, that is a major deficiency. In Table 10 in Module 2.7 in the BE summary table, there's missing, if the missing long-term storage stability is missing, that is a major deficiency, or if the long-term storage stability storage is less than the sample storage days. So kind of pivoting back to the following, to the PSG recommendations. So when you submit an ANDA, we use the PSG that's recommended at the time of ANDA submission. So not following a PSG recommendation, and if justification is not provided to demonstrate that you're choosing an alternative approach, for instance, it would be a major deficiency. Now, the stumbling block with this particular issue about product-specific guidances is that it really depends on your ANDA submission and the timing of it. So when you submit an ANDA, and if there's a new or revised product-specific guidance that has been updated before your ANDA submission, you want to take a look at that to make sure that it has not changed, and that if you need to provide justification to deviate from the approach, then you should do so. Our colleagues in ORS actually has a great website on future revisions and projected revisions, and they're categorized by different types of revisions. So you could look at that and kind of project and foresee how that could impact your ANDA submission. The other stumbling block is about cross-referencing BE studies. So if you're cross-referencing a BE study in another application, which you can, just be mindful that, again, it plays into when that ANDA is submitted, because when that ANDA is submitted, the filing review is based on the standards at that time, at the time of submission. And just want to point out that these are generally not considered a minor deficiency, so we usually do not issue information requests for this. As we move on to the drug substance in the quality section of your ANDA application, if your ANDA relies on a type 2 DMF, which is active pharmaceutical ingredient, a drug master file, that DMF undergoes a completeness assessment. If that completeness assessment is incomplete, then it is considered a major deficiency. Now, a stumbling block isn't necessarily results. This particular stumbling block is really for how to avoid having this major deficiency. And that's essentially making sure that the type 2 DMF is on the public available for a reference list to make sure that you can submit the ANDA and this would not rise to a level of a major deficiency. And again, just like the previous slide, this is not considered a minor deficiency. Okay. So let's talk about justification of specifications. So this particular, as if you could recall, the third bullet on the FDA resources that I provided earlier, there's a specific RTI guidance just for justification of specifications. So this encompasses both the drug substance and the drug product. So when we're looking at 32S45 and 32P56. If you're proposing limits that exceed the QT and or IT thresholds without justification, that's considered a major deficiency. One of the stumbling blocks that we often see for unspecified impurities is that when the proposed limits exceed the QT and IT threshold and the justification is based on a USP monograph. So in that RTR guidance, it explains that you should adopt more stringent thresholds, right? If there's no justification, that could rise to a level. If there's no justification other than USP monograph, that could rise to a level, rise to a major deficiency. And again, these aren't considered minor deficiencies unless the tables are not in the recommended format, right? Or if a discrepancy that warrants clarification. So in addition to the RTR guidance, there's actually recommended tables for these impurities. Okay, let's move towards the drug product. And when we talk about the drug product, this is the 32P1, your composition, right? So if you're using an inactive ingredient that exceeds any of the inactive ingredient database and there's no justification, that would be a major deficiency. In addition to that, another major would be having a formulation that's required to be Q1, Q2 same in pursuant to the regulation 314.94. If your formulation is not Q1 or Q2 the same with regards to non-exception excipients per those regulations, that would be a major deficiency. Now, a stumbling block with this particular issue that I want to point out is for ophthalmic drugs. So although you could have Q1, Q2 deviations for exception excipients for ophthalmic products per 314.94A9 Roman 4, the RLD should be accompanied by an in vivo BE study. That recommendation is mentioned in the RTR guidance. So if you are choosing to pursue an ophthalmic product that has deviations that meets the regulations, right, you just make sure that it also meets the recommendations in the RTR guidance. And that kind of lines up with the third bullet on the stumbling block, right? When we're talking about Q1, Q2 sameness requirements, and maybe there's recommendations in PSGs. So ophthalmic products may have PSGs are readily available to demonstrate bioequivalence. That could, one of the approaches could be Q1, Q2 sameness. The other stumbling block is about differences in pH adjusters. This is the guidance that I forgot to include in my previous slide. But if you have a difference in pH adjuster, that's a non-exception excipient for injectable, ophthalmics, and otics, right? Those are required to be Q1, Q2 same, and the regulations do not mention pH adjusters as exception excipients. So if you have a difference in pH adjuster from the brand or the RLD, just be sure to refer to that guidance, and it explains what waiver requests that should be submitted under 314.99B waiver, plus the supporting documentation under 314.90. And please note that that waiver request is not a BE waiver, right? So that's waiving the requirements for Q1, Q2 sameness as it relates to pH adjusters if there's a difference. And again, these are not considered a minor deficiency. Okay. In module 32P33, this is where the master production records are. These are where the blank manufacturing records. If these are not provided, that's considered a major deficiency. And this particular stumbling block isn't necessarily a stumbling block that would rise to an RTR, but it's just something that should be, that we wanted to educate everybody on when it comes to common blends used in exhibit batches, but maybe it's not intended for commercial, right? Sometimes it's not clearly evident in the application and we have to figure out what's happening in your application. And again, this is not generally considered a minor deficiency. In module 32P35, this is where the aseptically filled products for those products, they're missing validation of bacterial retention for the sterilizing 0.2 micron filter grades. Right. If that information, if that data is not provided in the original submission, that's a major deficiency. The stumbling block with this is that this particular study or data is buried within other reports or might be placed in an incorrect ECDD section. So similar to dissolution, the in vitro data that I mentioned in module 2.7 is that we will issue a minor deficiency to ask you to point out the location of that study in the original submission. And 32P8 is where a lot of the major deficiencies occur from filings perspective. The major deficiencies listed here are not using 2 API lots for each strength. Perhaps you're missing stability data entirely, and that could be a result of one of the stumbling blocks here, which is duplicating stability data, because it's a lot of files. You could have duplicated stability data in which now we're looking for something that's actually not there. Maybe there's less than 3 time points or less than 180 days. And specifically for liquid drug products, if you're missing, there should be 2 orientations, both worst case and non-worst case orientations. And that's the recommendation. If you're missing either one of those or both, then that's a major deficiency. The stumbling block with this particular issue is that there's unclear it can be unclear sometimes in the data, or there could be typographical errors that prevent us from trying to determine whether an ANDA is substantially complete. Or it could be missing stability initiation dates and or poll dates, in which case now we can't evaluate whether you actually have 180 days of stability data. These aren't generally considered minor deficiencies unless information or clarification needed, such as typographical errors or if you're missing stability initiation dates. As we move forward towards the end of the ECTD module 3.2R, this is where executed production records should be provided for all manufactured batches. Similar to stability data, if there are duplicate executed production records, now what we're looking for isn't there, it's considered a major deficiency. One thing that I want to point out is particularly for solid oral dosage forms, there's this recommendation to package greater than 100,000 units in your marketed container closure system for each strength, right? So the stumbling block with this is that there could be clear intentions of your marketing container closure, but there's no supporting stability data to have that qualified as your 100,000 threshold. There could be missing container closure system information in module 3.2p7, and or the labeling in module 1.14.1 is missing entirely. Now, a lot of the things that I talked about is the major deficiencies, but what happens when we talk about the minor deficiencies in general, right? So these are, if there's 10 or more minor deficiencies, that in itself is a major deficiency. And typically the stumbling block with this is not following the filing map or checklist, and now there's things that are generally considered minor deficiencies. If there's 10 or more, then now it's a major deficiency. Or sometimes you might have an issue with your technical issues with submitting your ECTD submission, which results in files that are missing, or files that can't be found, or corrupt files that can't be opened. And again, this is not, information request is not applicable unless there's 9 minors or less. Okay, so here's how to avoid. Justifications is really important, right, on multiple aspects. So the first is, if you, now that you're aware of all the major deficiencies and of the stumbling blocks associated with those, if you want to deviate from any recommendations, such as an alternative approach, then you should provide that justification in the ANDA submission. It should demonstrate that it satisfies the requirements of the applicable statutes and regulations. And it should be submitted either in the cover letter or in the specific module or section to draw the reviewer's attention. And I wanted to add emphasis here that it needs to be provided in the ANDA as originally submitted to be substantially complete. And what we do ask is that we're asking you to help us help you, right? When we talk about justifications and wanting it to be and how it should be in the original ANDA submission, from what I've presented here, filing a review is more than just, is the document there or not there, right? That's only the first step. So if it is there, then we have to look at that data, that information, to see if it meets the filing recommendations. But what it isn't to piecemeal certain information together, analyze the data, and then draw conclusions on behalf of applicants to say that they meet a particular recommendation, right? So that's an important point that I wanted to emphasize here. So the key takeaways, applicants can submit a justification to use the alternative approach to avoid RTRs if it satisfies the requirements of the applicable statutes and regulations. Be proactive and provide the justification upfront. Again, it has to be in the original ANDA submission in order for us to consider it substantially complete. And if an information request is sent, pay attention to each deficiency, adequately address each, and submit a response by the prescribed timeframe. And remember, our objective is to file substantially complete ANDAs to permit a substantive review. And we want to do that, and we're doing that in a manner that is completing in a way that's a uniform review standard. And with that, I'll hand over to the next speaker. Thank you.
Good morning, everyone. It's an honor and a pleasure this morning to share with you key updates on CDES quality management maturity program. So here's a quick overview of what we would cover today. First, we'll begin with the foundation question. What is CDE's quality management maturity program? And then next, we'll address why the program was developed. And then we moved into the lessons learned from the first cohort of the KMM program. And then finally, we will close with what the next steps are and what it means for industry. So what is the CDE's KMM program? CDE developed this program to move industry beyond simple compliance, because compliance alone is no longer sufficient to ensure a resilient and a reliable pharmaceutical supply chain. So at its core, the KMM program encourages drug manufacturers to implement quality management practices that go beyond the minimum regulatory expectations. And to that end, the program measures and scores manufacturing establishments across 5 key practice areas. Now, in terms of the practice areas, let's break them down. For practice area 1, the area focuses on how senior management evaluates company-wide commitment to quality. And also, we also assess whether management aligns quality and business objectives with strategic plans. We also assess whether management models the desired quality culture and effectively communicates with stakeholders. Practice area 2 focuses on business continuity and assesses reliable supply chain, including how the establishment anticipates disruptions and responds effectively to maintain consistent product availability. Practice area 3 addresses quality systems or advanced PQS and focuses on optimized practices that enhance quality systems across the product lifecycle beyond basic regulatory compliance. The practice area also looks at how establishments use quality risk management and process performance and product quality monitoring system to prevent supply chain disruptions. Then there is practice area 4, which is technical excellence, which addresses how the establishment develops and implements advanced technologies, processes, and methods that are fit for purpose. Technical excellence also covers the implementation of novel solutions to optimize operations, and also the establishment's commitment to technological advancement and innovation. And last but not the least, the KMM program evaluates employee empowerment and engagement, which looks at how the establishments cultivate environments where employees can take ownership and become proactive partners in achieving quality and business objectives. I think I must have slipped the goals here, so I'll go back slightly to the goals, why we actually developed this program. Right. So we developed the program to basically foster a strong quality culture mindset, because we realized that conforming to compliance is no longer it's no longer going to help us avoid disruptive supply chains. And also one of the goals that we wanted to really hone on for this particular program was to recognize establishments with advanced quality management practices, and also to acknowledge those firms that are actually going the extra mile to do more in terms of improving their quality management systems. The program also aimed to sort of work with establishments to identify areas where the firms had gaps in terms of quality management, so that we could work with them and then identify areas of opportunity with them and then provide suggestions for them where they could improve in those areas. And then finally, but not the least, the last goal was to minimize product risk to product availability to assure that we have reliable supply chains. So I'll skip forward a little bit. So this slide brings into focus everything in terms of what quality management maturity means. It's really about integration. Mature manufacturing establishments do not treat quality, operations, and business as silos. Rather, they integrate them so that quality is no longer a checkpoint at the end, but it becomes part of how the establishment runs every day. The key takeaway from this slide is that in mature manufacturing organizations, there is a deliberate integration of business operations, manufacturing execution, and quality management practices with technological advancements to optimize product quality, to enhance supply chain reliability, and also to drive continual improvement. So the natural question becomes, why did the FDA create the KMM program in the first place? The answer starts with a very real and persistent problem: drug shortages. In 2019, a multi-agency report was published on drug shortages, the root causes and potential solutions. And one of the underlying root causes, which was identified in the report, was the failure of the market to recognize and reward manufacturers for mature quality systems. And the goal was to look at how these manufacturers who actually focus on mature quality system, how we could actually help them to sort of continue in that stride. The report also noted that the pharmaceutical supply chain has become increasingly complex and also fragmented, and that disruptions on one end could create ripples that are transmitted and magnified across the globe. And without a doubt, the COVID-19 public health emergency brought this reality into clear focus and shed light on various supply chain vulnerabilities, which were not only limited to shortages of active pharmaceutical ingredients, excipients or intermediates, but could also include things like primary packaging components and disruptions to electricity and energy supply, which is needed to keep equipment and facilities running. The impact of these shortages further supported the need for approaches to characterize quality and drive continual improvements to boost supply chain resiliency. Subsequently, the development of a rating system to incentivize drug manufacturers to invest in quality management was proposed as one of the enduring solutions to drug shortages. It was also noted that better management of the risks in the supply chain would benefit all stakeholders, from pharmaceutical manufacturers to wholesale distributors and pharmacies. And ultimately, consumers and patients will benefit from better access to quality medicines when they need them. So this slide here provides an illustration of why the KMM program is also important. Here, the chart, as you can see, shows the various reasons for new drug shortages. You'll notice that manufacturing and quality issues represent a substantial portion of drug shortage causes. Again, this underscores the importance of mature quality management, and that can prevent these issues before they actually impact drug availability. Ultimately, by improving quality management maturity across the industry, we can address one of the root causes of drug shortages and better serve patients. So in January 2024, we published a Federal Register notice soliciting volunteers to participate in the first cohort of the QMM Prototype Assessment Protocol Evaluation Program. Out of the pool of volunteers, 9 establishments were selected. These included manufacturers of generic drugs, innovative drugs, biologics, contract manufacturing organizations, and a contract testing laboratory. These establishments range in size from medium-sized family-owned companies to large multinational corporations. So before I share any further details on the 2024 program, I would like to share just a quick snapshot here of the most recent milestones as far as the progression of the KMM program is concerned. As I've already mentioned in the previous slide, we announced the prototype assessment program for cohort number 1 in January 2024, followed by the execution of the program from September through December 2024. Another Federal Register notice was also issued out in April 2025 for cohort number 2, and that round of assessment was started in June 2025 and is currently still ongoing and expected to complete in the May of 2026. For the 2024 program, which is cohort number 1, the goal was to really gain experience with the prototype assessment tool that CEDA developed to determine its fitness for use. The tool included an assessment protocol with a series of questions and a rubric that described maturity across 5 levels. And this rubric was used to score an establishment's level of maturity in its practice area. Subsequently, lessons learned from the 2024 program informed refinements to the prototype assessment tool and the assessment process for the ensuing cohort. In the 2024 assessment, we tested different assessment formats. All assessments were scheduled for 5 business days, and 4 of the assessments were performed on site, 1 assessment was performed remotely, and then 4 other assessments were performed using a hybrid format. For the hybrid assessments, the first 2 days were conducted remotely, and the remaining 3 days were performed on site. So the prototype assessment protocol contained questions in each of the given practice areas, and each question had a rubric defining 5 levels of maturity, with level 1 being the lowest and level 5 being the highest. Here, I'm going to show an example of what the protocol question and the rubric look like. In this example, the question was, how does the establishment use risk management to inform decision-making? At level 1, establishments do not consider risk in decision-making. At level 3, establishments systematically consider risk when making decisions that impact commercial manufacturing. And then at level 5, risk is considered in decision-making across all stages of the product life cycle. And also the application of risk management to decision-making is continually being improved. So let's talk about the scoring process. One of the challenges with quantitatively scoring quality culture or behaviors is the influence of subjectivity or bias. And to manage that subjectivity and bias, a number of strategies were used in scoring an establishment's level of quality management maturity. First, each establishment was independently scored by 3 assessors. Also, the responses to the protocol questions needed to be substantiated to receive full credit. So what do I mean by that? As an example, an establishment may share how they perform a risk assessment, or they may share their standard operating procedure to explain how they perform a risk assessment. But this information could only be substantiated by evaluating risk assessments that have already been performed. Second, all assessors use a standard set of questions and a scoring rubric to evaluate and score the establishment's maturity. What this did was that it introduced a standardization to the assessment, which helps us to manage bias. Third, inter-rater variability in the scoring is also monitored and flagged, and it must be resolved before the establishment can be assigned a final score. The resolution process also involved the assessment team working together to understand why there is a discrepancy in the scoring by revisiting the assessment tool and going back again to review the information that was gathered during the assessment. So as far as the reporting goes, each participating establishment received a QMM assessment report containing the numerical scores for each of the 5 practice areas. The maturity scores were further broken down by major topic areas, which we call the elements, to provide more granularity on the areas of higher or lower maturity. The report also contained a narrative describing the strengths and opportunities to improve in each practice area. Establishments were also given time to review the report and then had an opportunity to meet with the assessment team to discuss the assessment findings and also pose any questions that they may have. So following each assessment, we maintain ongoing engagement with the participating establishments through a structured series of meetings that is designed to drive meaningful improvement. So once establishments receive their KMM report, which includes scores across the 5 practice areas, along with a detailed summary of their strengths and opportunities for improvement, we transition into what we call the post-assessment phase. During this phase, establishments are asked to select at least 1 improvement opportunity from those identified in their report and develop objective, measurable goals to address them. This information is also shared with the QMM team at least 1 week before the first post-assessment meeting to allow for productive discussion. In subsequent meetings, establishments provide progress updates on their implementation efforts and share where they are in terms of executing their selected improvement opportunity. Again, this creates an accountability pathway and allows the QMM team to offer guidance, share best practices, and also help troubleshoot any challenges that arise during implementation. This slide here summarizes the practice area scores for all the 9 establishments that were assessed in 2024. There's a lot of information here, but I just want to call your attention to just a few main points here. First of all, the maturity was scored on a scale of 1 to 5. Higher scores here are shown in green, and then lower scores are shown in yellow. Some establishments, as you can see, had higher scores. For example, establishments E and F. Some also had lower scores, as you can see here, A and G are typical examples. And then there's those that had a mix of higher and lower scores, such as establishments C and D. Again, you can see here that the prototype assessment was very useful in distinguishing between maturity between the establishments and also between practice areas within an establishment. Also, in this cohort of 9 establishments, the highest scoring practice areas were management commitment to quality and employee engagement and empowerment. In this figure, you can see 3 main elements in each practice area. Communication appears twice here because it is assessed from both the perspective of senior management and also from the perspective of the employee to determine if their perspectives align. By contrast, the lowest scoring practice areas were advanced pharmaceutical quality systems and technical excellence. 3 elements were of particular interest here because they generally had lower scores: quality risk management, data excellence, and knowledge management. So what lessons did we learn overall from our 2024 prototype assessment protocol evaluation program? First, the protocol and the rubric were able to clearly distinguish differences in maturity, not just across companies, as I've already mentioned, but even across practice areas within the same establishment. Second, the assessments provided actionable feedback. This means that establishments didn't just receive scores, but they also received insights that they could use to build improvement plans with measurable success. This notwithstanding, we also identified we deemed ready for refinement. For example, the use of complex rubrics naturally introduces subjectivity in the scoring. So we decided to do a further simplification of the rubric to reduce variability and among the assessors as they work to score the establishment. Similarly, some of the questions were insufficiently clear, we found, and required additional follow-up questions to adequately address the desired topic. So we worked to finesse that as well. And then finally, we realized some of the questions needed to be simplified or reworded to clarify their intent. So these are all some of the improvements we've been working to add to the protocol as we continue to advance towards more cohorts. So finally, what are the next steps? In February 2026, FDA published a Federal Register notice soliciting a 3rd cohort of volunteers to participate in the QMM prototype assessment protocol evaluation program to allow CEDA to continue to gain experience with the refined tool and assess its fitness for use. As we work to execute this 3rd round of assessment, it is our intention to continue to foster quality, mature quality management practices through the identification of strengths and opportunities. And it is also our intention to share the lessons learned and also publish the results for the 1st and 2nd cohort assessments. And ultimately, it is our hope that our continuing collaboration with industry will continue to foster the mindset that will drive reliable supply chains. And talking of supply chains, I think you've seen a version of this slide, but here's a final reflection of something that we can all ponder over. When supply chains are reliable, patients receive quality drugs exactly when and where they need them. Thank you for your attention.
Thank you very much, Dr. Toom. And now I welcome our speakers from this morning to join us on the panel here. And I would like to introduce our two additional panelists. First, we have Dr. Julia Lee, who is the Deputy Division Director for the Division of Filing Review in the Office of Regulatory Operations within the Office of Generic Drugs. And then we have Christina Marshall, who is a Project Manager within the Office of Regulatory Operations at OGD. Thank you all.
Out some improvement opportunities. So they're thinking how that information is going to be shared with the FDA enforcement bodies. They will come and do the GMP inspection on them, right? So what's the relationship between these two teams? Should the industry be very concerned? What's their finding in the QMM program?
That's actually a good question. So what I'll say is that currently there is no direct relationship between the compliance program and QMM, right? So the QMM team is basically focused on quality management practices and also how firms can improve their quality management, whilst compliance program focuses on meeting regulatory requirements. So you've mentioned that firms, there is a concern that firms, the information that we collect from QMM may be used to as part of a compliance program. I can tell you currently now there are no plans as far as I'm concerned. So the information that is collected during the QMM assessment is shared within the QMM working group, and the analysis is done within the QMM working group. And currently I don't have any official information related to how the QMM program will be related to a compliance program. But currently, yes, we share the information with only the participants. So for example, if we do an assessment at firm A, we do a report, we send it to the firm, and even when we have to publish information out there, and as you can see, we've been working to publish a number of papers, we anonymize firms and we don't provide any firm-specific data. Thank you.
That's good. Can I ask one more?
I don't know. Anyone else in the room? Hold on. If you might get an opportunity if no one else comes. Anyone else for a question from the room? All right. No, you can come.
Like very shy audience, right? So, Dr. Eric, sorry, I'm asking one more question. I think this is the third year of pilot program going on. do you have any plan to scale this up? When exactly do you think it may happen? And if it is happening, who is going to do it? I don't think FDA is going to do all the assessment themselves, right? What is the plan to make it really scalable?
Man, that's also a good question. I wish I had all the answers. But what I can tell you is that there's been discussions in the corridors of the FDA to scale this up in the future. But as to the actual definitive plan in terms of rolling this out on a large scale, I'm not privy to that conversation. But what I can tell you is that for the current year and the few years ahead, we will continue to use the protocol evaluation to kind of learn more about the protocol, because even before we scale up, we have to ensure that the protocol that we've developed is robust. So currently, yes, there's been discussions, but as to how that plan would actually dovetail into the specific tactical plans that we will implement to get the program running at a scale, I don't have information yet, but we can talk offline, and if I have additional information, I'll let you know. Thank you.
Thank you so much, everyone.
Okay, in the absence of room questions, let's go online and we'll go at a fast pace online.
All right, our first question is for Lieutenant Commander Fields. What is the best way to follow up or contact FDA if we don't receive controlled correspondence response within the time frame?
Thank you for the question. We do have a designated email address that they can refer to for follow-up information, especially for that specific case. So they didn't say particularly when they were expecting that information. So I don't know if that means the initial that the agency will review, give the substantive review. We try to do that within 7 calendar days. And the 60 days for the level 1 and level 2, they can email the inbox for a status update regarding that.
Excellent, thank you for that. Next question is for Dr. Julia Lee. Will DFR refuse to receive an ANDA if the Form FDA 356H is unsigned?
Thank you for the question. Yes, under 314-101D, DFR will refuse to receive an ANDA if the Form FDA 356H is unsigned.
You can go ahead, because unless you see someone steps up to mic, just continue.
Right, we'll just keep going.
Yeah.
Okay. Oh,
you gotta wait now online.
Hi, my name's Adam from Apotex. Question for Marcia. I know there's limits on control correspondences for only 3 levels or 3 formulations. Are there any limits when you're submitting a post-CRL clarification correspondence on the number of deficiencies you could request clarification on?
Thank you for that. If I'm understanding correctly, is there a number of deficiencies that can be addressed in one control correspondence in the complete response letter? And to my understanding, there is not. If you are identifying what was the deficiency in the correspondence letter. So in the control correspondence, if it's associated with that specific discipline. So they will be categorized on if it's bioequivalence deficiencies, so try to group them based upon the discipline. So if there are 5 deficiencies in the complete response letter for bioequivalence, you can submit inquiries related to that deficiency for that office, but separate the offices.
Got it.
Thank you.
Thank you.
Thank you. Online question, please.
all right. This next question is for Dr. Zhang. Regarding PK analysis, what is the appropriate level of detail that should be pre-specified in the study protocol?
Yeah, thanks for the question. Study protocol is a high-level framework to guide the study design. So usually it's not expected to include a lot of details, but it will benefit applicants or industry to have some level of detail, such as I mentioned, how to calculate PK parameters, such as AUC, and what's your statistical model, and there will be different results between GLM and mixed model. So it's better to pre-specify some level of details, but it's not expected to have a very detailed study protocol, but just a high level. Yeah. Thanks.
Thank you for that response. In that case, we'll move to the next online question. This is for Christina Marshall. If I have multiple questions and a single controlled correspondence, do all the questions have to be related to a specific discipline?
Good question. It's kind of similar to what the gentleman just said. Yes, it has to be specific to that particular question, because we send them out to to different disciplines. So if a question is regarding Q1, Q2, then that will go to filing, and vice versa versus, as far as any other discipline. So yeah, to answer that question, yes, it has to be specific to that particular discipline. And in that regard, the company would submit a separate controlled correspondence for each question within that discipline.
Thank you for that response. Our next question will be for Commander Fung. So can you provide a justification for deviating from the recommendations after ANDA submissions?
If I understand that correctly, is that after ANDA submission, as in when the ANDA is with filing? Or is that after filing? Do we know?
It specifically says after ANDA submission.
Okay, so my answer will be based on it's during the filing review. So justification that you may see would be, maybe it's a product specific guidance that recommends a certain approach to demonstrate bioequivalence. And maybe that study that you're using to demonstrate bioequivalence or another method, right, does not meet the recommendations outlined in the product-specific guidance. So in which case, there's actually other ways, but this is at the time of the ANDA submission, so it's already gone through pre-ANDA development and drug development. So to answer that question, is a justification, if it's related to the BE, the bioequivalence aspect, it can be provided in Module 2.7 or Module 5 to show to explain what method you're using to demonstrate bioequivalence for your drug product.
Excellent.
Just people in the room, attendees in the room, you made all this effort to come out here. I know you love the campus, but I'm sure you have some question on your mind. So please, let's get some participation in the room as well. Okay? Thank you. Online, please.
All right, and this will be another question for Commander Fung. Please elaborate on how the filing review assessment differs when submitting a new strength as a PAS versus original ANDA.
Thank you for that question. So it's not different in the new when an applicant chooses to submit a new strength prior approval supplement to amend their approved application, the filing review is the same. We look at everything as if it was originally as if it's an original submission.
Excellent, thank you for that. Our next question will be Dr. Twum. Regarding the completed QMM assessments, does FDA plan to publish results and lessons learned? If so, will you publish results with company name?
I think I already addressed this earlier, but no, we plan to first publish the results for the first and second cohort. It's in the pipeline, but as to whether we will disclose the actual companies that we assess, I think I've already made that clear. We will anonymize company names. We only provide the hard data and remove any company identifying information. Thank you.
Thank you for that response. My next question will be for Lieutenant Commander Fields. Can a DMF holder submit controlled correspondence to obtain FDA's feedback on a specific impurity control strategy if that DMF is referenced in multiple approved ANDAs? If yes, does a specific ANDA need to be referenced? Or can the DMF holder describe only the RLD since the question applies to multiple approved ANDAs? I can repeat if needed. Of course. Can a DMF holder submit controlled correspondence to obtain FDA's feedback on a specific impurity control strategy if that DMF is referenced in multiple approved ANDAs? If yes, does a specific ANDA need to be referenced, or can the DMF holder describe only the RLD, the reference listed drug, since the question applies to multiple approved ANDAs?
I'm going to respond to that for Marcia. If I understand the question correctly, we usually do not answer DMF questions. However, we do answer questions that may address multiple ANDAs. So, in that regard, they can submit a control correspondence that is asking about multiple ANDAs regarding the DMF in a general, as far as a general question, but if it's actually about a very specific ANDA, those questions we kind of refer out, and we really wouldn't accept those as a control correspondence. But if there's any question about it, they can totally submit that question to the to our generic drugs mailbox, and we can answer it more clearly and more general to what they're actually asking. And if we don't have the question, if we don't have the responses as to how they should proceed, we definitely can refer it out. But usually we don't handle questions regarding DMFs.
I see. Thank you for that response. My next question is for Dr. Lee. What are some resources that could help in filing an ANDA?
Thank you for the question. I think that the resource that helps most is the map that was cited in the presentation, the filing. It includes the filing checklist, but also the RTR guidances also identify the major refuse to receive reasons. So those are resources that are definitely helpful, and all the other documents that were cited in the presentation.
Excellent. Thank you for that response. My next question is for Dr. Zhang. When unexpected challenges arise during study conduct, what are the appropriate steps to take if the pre-established study protocol needs to be modified?
Yeah, thanks for the question. So I think the first step is to start an investigation on why this unexpected challenges arise. So, for example, whether it is due to the safety of this drug product or whether it's a study conduct issue. If you can rule out safety or study conduct issue and you decide to revise your protocols, then you should start revision as early as possible and before the statistical analysis. Thanks.
Thank you. Our next question is for Commander Fung. For an injectable product, can I propose a different fill volume than the RLD, even if the dose can still be administered per the package insert?
That's a great question. So strength is defined for injectable products as the concentration and the fill volume. So this is the total drug content. So if it is different from the brand, the reference listed drug, then you would need a suitability petition to get that approved before submitting an ANDA.
Thank you for that. I believe we have time for a few more questions, so I'll keep going on my end. So for Dr. Twom, what are some of the characteristics of a robust quality culture?
That's a good question. So in our assessment, one of the characteristics that we've seen that is indicative of a robust quality culture is, first of all, one underlying theme, which is management, commitment, and accountability. We see that characteristic very well in firms that have a higher maturity. And in those firms, you see that management doesn't just, define quality objectives and business objectives, but they actually walk the talk. They champion quality initiatives. They also show accountability. And you see management presence not just at management review meetings, but you also see management following up on even management review meetings, follow-up actions, follow through, making sure that the actions coming out of management review meetings are actually implemented. You also see management also showing presence on the floor. And it's not just presence on the floor to catch quality issues, but presence on the floor to engage with employees. And that leads me to my next second, sign that we see with mature firms, which is employee engagement and empowerment. In highly mature firms, you see that the employees from the top to the bottom, all of them are engaged and empowered, so that typically you don't, for example, if you have a packaging technician, and they notice an issue on the packaging line, for example, like, different coloration of the tablet, they don't need to really, think twice before they stop the line and then escalate the issue, right? And that in those organizations, you see that this kind of proactive mindset that is stopping the line and then notifying management or whoever is in charge is actually not seen as something that is punitive, like they don't punish, but rather it's seen as an improvement opportunity, and then they work on that, and out of that comes, maybe an implementation of a CAPA. In such firms, we also see that there is always laid out channels for reporting of just not quality issues, but also near misses, right, and also potential issues, so that it gives it provides some kind of like leeway for the firm to identify issues way before they crystallize into the actual issues. So these are some of the signs that we see with firms with robust quality culture. Thank you.
Thank you very much. That is our last. How long you been standing there? All right, you're our last question.
Sure. First and foremost, thank you very much for the excellent presentations. I know you covered pretty much everything that we always look for, starting from the control correspondence all the way going into the filings, making sure that the BE aspects are all well done, major roadblocks are all taken care of. So thank you so much for that. I'm Sharath, and I'm representing Laurus Labs Limited. My question is for Dr. Susan. I know she is not on the dais, but regarding the pre-check program, there were 9 finalists selected on April 1st. Would that information be made public for the strengthening of the domestic manufacturing, or is that something that you'll have to get back?
We'll have to get back to you because, as you said, Dr. Rosencrance isn't here right now to address that, unless someone from OPQ wants to address it. No? Okay. Thank you. Okay. Thank you to our expert panelists. Thank you so much. We will break now for 15 minutes. We will return at 11:25. If you've pre-ordered lunch, please pay at the kiosk to make it simple at lunchtime. You'll also have snacks if you didn't order anything, snacks and refreshments for purchase. And make yourself happy for the 15 minutes. Thank you. Okay, everyone, thank you very much. We are 5 minutes behind time because I wanted to give the folks out there at the kiosks some time to pay for their lunches, but we need to start. So we'll start our afternoon session here, and I'll introduce all our speakers as we did before, and then they will come up one by one. So first, we'll have Commander Diana Solana Sodene, talking about using FDA's inactive ingredient database, best practices and considerations. She will be followed by Dr. Melanie Muller. Standardized principles used to determine maximum daily dose will be her topic, master toxicologist. And bringing up the final presentation for this session is Dr. Likan Lian, supervisory pharmaceutical scientist with the Office of Product Quality Assessment in OPQ, and he will speak to us about complex drug products, quality considerations for generic oligonucleotides. So let's start off first with Commander Diana Solana Soledis.
Well, thank you very much for that kind introduction. And as Captain Brenda said, yes, I'm going to be given some guidance on how to use the FDA's inactive ingredients database and basically just cover some best practices and considerations. So here's just an outline for today's presentation. I'll be providing you an overview of the inactive ingredient database. We're going to talk about how to use it. We're going to talk about how to calculate the maximum daily exposure. And also we're going to talk about the IID program enhancements under GDF. We're also going to talk about a special case with the red dye number 3, and I'll provide you some helpful resources, our IID mailbox, and contact information. So what is the IID, the Inactive Ingredient Database? Here's basically a snapshot of how it looks. If you go on the FDA website and you just search for the Inactive Ingredient Database, you will come to this homepage where you can search for the excipients and ingredient name. So the IID provides information on excipients present in FDA-approved drug products. In 1987, FDA first made available an inactive ingredient guide, and this was available in hard copy paper format. And FDA began making the information available in an online database in 2003. So the IID provides information on excipients present, as I said earlier, in FDA-approved drug products. It includes excipients in ANDAs, which are the abbreviated new drug applications, and new drug applications, which are known as the NDAs. And of note, excipients in approved biologics license applications, our BLAs, and our over-the-counter monograph drug products, OTC products, are not contained in the IID. Now, if an excipient is used in an approved drug product for a particular route of administration, the excipient generally is not considered new and may warrant less extensive review the next time it is included in a new drug product. So again, just showing you the homepage of the IID. And what you'll do is you'll see here, if you're like, oh my gosh, how do I know how to use the inactive ingredient database? There's a guidance for industry that has been posted, and it provides you information about what the inactive and active ingredients are. It also provides recommendations for how to use the IID in the development of drug products and how it can be used in evaluating excipient safety, how it's structured, the data regarding excipients, and how nomenclature, maximum potency levels, and units of measure are presented. It also describes the database's benefits and limitations. The draft guidance uses the term excipient rather than inactive ingredient. And it's just because historically the term inactive ingredient was used in naming the database. So the excipient may be more accurate, right? So the term excipient may be more accurate because it recognizes that some ingredients may have some biological activity, even though they are not intended to exert therapeutic effects. So while the guidance uses the term excipient, I will be using the terms excipients and inactive ingredients interchangeably. And of course, let's not forget the FAQs. You are welcome to check them out. Here you'll find like frequently asked questions regarding, what the inactive active ingredient means. It explains some of the data you will see after doing a search and how it is updated and how to obtain a copy. Moving along, let's talk about how to use the IID. This is a snapshot of the IID database. So let's say you want to download the entire IID. You will simply click on the ingredients database download link, and that will give you the ability to download the entire IID to your computer. If you would like to initiate a search for a specific excipient, all you have to do is enter the first 3 characters of the excipient name, and then click submit, or you can actually select the alphabetical, the first letter of the excipient name and search for the specific excipient that you're looking for. So this is exactly a query result that will come in. Let's take the example of carbamide homopolymer. And as you can see, there are different forms, different grades, type B, type C, type A. And for each of this, for each of the excipients shown here and within the specific grade, you will see there's a specific route of administration. For each record. There's also a dosage form for each record. There's a Unicode. And then you will have either the maximum potency per unit dose that's listed or the maximum daily exposure that's listed. And if a record was updated, you will see a yes or a no. You would also see, as I said earlier, the CAS number and the Unicode. The CAS actually stands for the Chemical Abstract Service Registry number that is associated with the specific excipient, and along with the Unicode, which will be highlighted. However, it's not highlighted in this example, but the UNI stands for the Unique Ingredient Identifier that's assigned by FDA's Global Substance Registration System. Each record is showing a unique excipient or inactive ingredient associated with the route of administration. Here it's topical, and the dosage form combination. And here it's in cream. And then, of course, as I mentioned earlier, for each record, you're going to have either or. Either you're going to have the maximum potency per unit dose, or you're going to have the maximum daily exposure value that is associated with each excipient. And again, any changes made to the record will reflect a yes or a no. So interestingly, we do have some limitations when it comes to the IID. It does not provide context of use. What does that mean? So if there's a duration of exposure, you want to see if it was chronically used or acutely used, it doesn't show that. It doesn't indicate that in the IID. Also, it doesn't indicate the patient population and indications for use. What's interesting also for us is that the IID, this is something that we really believe in as FDA. We do not disclose proprietary excipient information. So you're not going to see the reference list and drug formulation associated with the specific excipient record. And there isn't a link between the maximum potency per unit dose and the maximum daily exposure for each record. So don't think that they're associated, okay? It's either or. And lastly, as I mentioned earlier, we do include excipients from that are listed in the NDAs and ANDAs, but not BLAs or over-the-counter OTC monograph products. So interestingly now, how do you calculate the maximum daily exposure, right, for products, which is also known as the maximum daily intake for oral products? You will simply just follow this formula. It is the maximum daily exposure is equal to the maximum number of dosage units recommended per day based on the product labeling multiplied by the dosage unit level of the inactive ingredient or excipient. So let's say you have an excipient level of 20 milligrams per dosage unit. You will multiply that by the number of dosage units that are recommended for that day. So if it's 2 dosage units recommended for that day, then you have a total of 40 milligrams maximum daily exposure or maximum daily intake for the specific product, okay? And since we're talking about oral medication here, we'll talk about the MDI. So the MDI will be 40 milligrams for this specific oral product. So now let's talk about the IID program enhancements. Congress first enacted the Generic Drug User Fee Act, or what we call the GDUFA, in 2012, following negotiations between FDA and industry with input from public stakeholders like you. I want to believe most of you are here today. And GDUFA was implemented to ensure that patients have access to safe, high quality, and affordable generic drugs. And it brings greater predictability and timeliness to the assessment of generic drug applications. IID enhancements were included in the commitment letter of GDUFA 2. Specifically, the commitment letter stated that by October 1st, 2020, FDA will complete enhancements to the inactive ingredient database so users can perform electronic queries to obtain accurate maximum daily intake and maximum daily exposure information for each route of administration for which data is available. FDA will update the inactive ingredient database on an ongoing basis, and this is what we do on a post. We'll post quarterly notice of updates made. This is what we've been doing. And also such notices will include each change made and for each change, the information replaced. Then GDUFA was reauthorized, and we continued with our commitments to update the IID on an ongoing basis and post quarterly notices of updates made. The addition of MDE, so our maximum daily exposure, to the IID was an important addition to the GDUFA 3 commitment letter that was based on stakeholder feedback to really benefit the end user. Now, since after submission, my office looks at it from an MDE perspective. We were looking at different bases or different database bases and other sources than the publicly facing IID that only contained per unit dose information. So I think it's definitely beneficial that we are all looking at the similar information now, mainly for those that have MDE populated. And next slide, I'm just showing you how we have continued to provide quarterly updates. We started July 2020 with 4% records that were filled with MDEs, and now we are at 44%, and we continue to increase steadily. As I continue with the enhancements, let's talk about the quarterly change log. So when you go to the inactive ingredient database and you search, right, for any product, but you can also get access by clicking on the most recent changes to the database link. You will be able to download the quarterly change log. And the quarterly change log shows 3 types of status changes. You either have C for corrected records, D for deleted records, or R for replacement records. In the interest of time, let me go back here. For C, so whenever there's a record that is corrected, right, it is important to note that new records are displayed on the primary IID webpage. Okay, you will not find it here in the quarterly change log. And the MDE records that are higher than the previous quarter are also displayed on the primary IID webpage. What you will find here in the quarterly change log are the changes, current changes that are made because you have a change to the ingredient name, unit of measure, or potency that were corrected in the current quarterly IID. Note that the potency corrections that resulted in a decreased potency value are displayed in the change log. So for instance, you have thromethamine topical gel. As you can see, there was a replacement, right? There was a change. It changed from 144 milligrams to 56 milligrams. And so these are the types of changes that will be made, and we will illustrate that in the quarterly change log. Next, you have D for deletion. We don't really like to delete records. They're not really deleted from the IID. However, there's always a tracking of these, and you will find them in the change log. And so let's say for safety reasons, an excipient that was contained in a drug product, the drug product was deemed unsafe and it was withdrawn, we will delete the record and place it in the change log. And then as far as the replacements goes, with the notation of R, it is when we have replaced a maximum potency per unit dose with a maximum daily exposure. Okay, and as you can see, subsequent records will show that in the quarter, you'll have either the replacement of potency for MDE value. So please continue to monitor the IID and please get access also to the changelog for additional information. And so, of course, you may be wondering, where are these changes, or what rises, what gives way to these changes? So we have the MDEs that come in from newly approved products, and we also have MDEs that come from previously approved products. One thing to mention is that we've been populating this IID since 2019. And so just imagine all the products that were approved prior to 2019. So we continue to form internal work groups. We have SMEs, subject matter experts, that are calculating the MDDs, which will allow us to calculate the MDEs, and then we'll use that to populate the IID. I just want to quickly just touch over, some situations where we have to remove an excipient from the IID, and this is a special consideration, is the red dye number 3, also known as erythrocin. So the FDA had published a Federal Register notice, and they posted this last year on January 15. And what happened was it was pretty much to address a ban. And so FDA revised 21 CFR color additive regulations to remove red number 3, and drug manufacturers now have until January 18, 2028 to remove this color additive. So what action did we take? We had to definitely remove red number 3, red dye number 3, and erythrocin, and any alternate name that was associated with the red dye number 3. And here are some resources if you're interested to learn more about what actions we had to take. I've also added here some more information regarding the other color additives that were of concern, but just visit the website for updated information on these. And so I just want to leave you with some helpful resources. Again, this is our search page for the inactive ingredient database. You have the guidance, the draft guidance, and also the FAQs as well. If you do have any questions, which we do get a lot of questions, we welcome those, because we keep trying to improve the IID. So we want to hear from you. So any question about changes in the IID listings or reports of error that you find, please feel free to email us. And then if you have requests for clarification of units or excipient names or anything that has to do with the identity of the excipient record, feel free to also email us. However, we will only take general inquiries if you send it to the mailbox. What are we not going to be able to answer? Questions that may disclose proprietary information. For instance, you have an excipient record. What was the associated NDA or ANDA to this specific record? Or what is the reference listed drug product formulation? We cannot disclose that. That is proprietary information, and that's so important to FDA. Also, we, as far as acceptability of proposed excipient levels, we will not be able to respond to that through the IID mailbox. However, for general inquiries, feel free to send your questions and concerns to the IID update at fda.hhs.gov mail inbox. And nomenclature corrections, questions about excipient names, and uni requests, you can send that to FDA-SRS@fdahhs.gov. And lastly, for any application-specific questions related to the use of excipients in generic products and under development, should be submitted through the control correspondence. And there is a 60-day turnaround for those, so it's pretty fast. So feel free to submit your questions to that route as it relates to the specifically to the application. And so, of course, I just want to thank the dedicated team that work hard behind the scenes. We are definitely doing a lot of work trying to enhance the IID, responding to all the questions that you may have, and communicating as well internally and externally with our stakeholders. So, of course, here's a list of everyone who is involved. It takes a whole village, right? And that's the end of my presentation. And thank you so much for your generous listening. But right now, I want to call over Melanie, who I like to call Dr. Mueller, please come over. Thank you. Who will be presenting on the standardized approaches, right, for MDDs. Thank you very much.
Okay. Let's see here. How's the audio? Good? Okay, great. Thank you. Good morning, everyone in the room, and good morning to folks online. I'm excited today to talk to you about standardized approaches we use when determining the maximum daily dose, short the MDD. Over the next 20 minutes or so, I'll be covering background information, which includes the definition of MDD, its impact, and the challenges we encounter when we're trying to determine MDD. Next, I'll provide information about CDER internal alignment efforts. Here I will focus on the importance of alignment and the key alignment elements. Then I'll provide some recommendations on how industry can obtain feedback on MDD determination. And to wrap up my presentation, I will summarize the key takeaways. So let's get started by defining MDD. What's the MDD? For regulatory purposes, the MDD is a numeric value, an amount per day, which is calculated based on the API, reasonably administered within a day to most patients as described by labeling. Let's take a closer look at the term reasonable. MDD generally cannot capture each dosing scenario per labeling. Instead, for regulatory purposes, the MDD is a suitable standard representative for most patients based on our best judgment. And as such, the MDD is a multidisciplinary standard that's used during technical review of new and generic drugs. Importantly, the MDD is not intended to impact dosing in clinical practice. Why is the MDD important for us? Well, the MDD is critical during safety and quality review of new and generic drugs. The MDD is used to determine impurity control in drug substances and drug products. The MDD is also used to set acceptable limits for nitrosamine impurities. And the MDD is critical to establish the AET for extractables and leachables, review and study design. And then, as Diana mentioned, the MDD is also established to determine the excipient maximum daily exposure that you can find on our inactive ingredient database. Overall, I'd like to point out that the MDD is critical to ensure regulatory consistency across similar drugs. Now that we defined MDD and understand the importance, let's take a look at the challenges. It's important to remember here that the MDD for regulatory purposes is an amount per day, for example, expressed as milligram per day. Unfortunately, labeling often does not list the MDD as amount per day, and instead we need to convert information available on labeling to this numeric value. To add insult to injury, often labeling contains variables that introduce calculation uncertainties. I have a few listed on this table right here. For example, labeling could say dosage administered in milligram per kilogram per day or milligram per meter square body surface area per day instead of just milligram per day. Or labeling could mention apply a p-size of a topical product to an affected area. So what does that mean? What's a p-size? How much does that weigh? How do I get from p-size to my milligram per day or gram per day? And then for ophthalmics, what we often find challenging is to determine the actual number of drops that being administered per day for a patient, considering if it's one eye or both eyes. So it takes a little bit of clinical judgment and clinical understanding how these products are used in practice. That's not where it ends. Labeling also often contains complex dosing scenarios that add further uncertainties to MDD. So for one, we could have multiple indications with different dosing regimens. Or we have different dosing regimens for specific patient subpopulations for the same indication. Or a drug is dosed to effect, in which case the MDD depends on the individual patient response, such as the pharmacological effect, the tolerability, or adverse events. And then to make it extra fun, you could encounter a combination of any variable and any complexities. However, we see these complexities and challenges as a great opportunity for standardization and alignment. But why standardize? So why is that actually important? Why does it matter that we all are consistent? Well, for one, standardization ensures regulatory consistency across similar situated drugs. For example, we regulate similar drugs similarly or the same way in terms of impurity control, such as nitrosamine impurities, if we have the same denominator. Standardization also ensures consistent and efficient review of new and generic drugs. Standardization allows for consistent and clear communication between industry and FDA, and therefore it will help drug development and application approval. Overall, standardization streamlines drug review by reducing the complexities of MDD determination. So let's take a look what we're doing internally across CDER. This slide shows the 4 key elements of CDER alignment. We have standardized approaches and principles. We're also working to implement a single MDD concept, and we're aiming to develop active knowledge management and quality control. In my next few slides, we're going to take a closer look at each of these elements. I want to start off with standardized approaches and principles. The goal here is to standardize and align on as many of these variables and complex dosing scenarios that I described earlier under challenges. We started off by standardizing clinical parameters. A few examples listed here are we consider 60 kilogram body weight and 1.62 meter square body surface area for adult patients if we need that variable to determine a numeric value for the MDD. Unless, of course, the patient falls significantly out of this scope, then we would apply something different. And again, that's based on clinical judgment and the indication. Drugs that are dosed multiple times throughout the day, for those we determine the MDD based on 24 hours. We generally do not account for waking hours. So if a drug is administered every 4 hours, we derive the MDD by considering 6 doses. We also standardized clinical principles for common scenarios. Examples listed here are, if labeling lists multiple indications, in general, the MDD is based on the highest dose across labeled indications. Here's an interesting one, where labeling lists adjust dosing for concomitant use of CYP enzyme inducers. We determine the MDD based on the dose that is representative for most patients per labeling. And that means generally we do not apply the CYP inducer dose as MDD. We would refer back to what the most patients for that indications are administered. If we have drugs that are over the counter and prescription, the MDD for the prescription product is generally based on the MDD for the OTC product is generally based on the prescription labeling if this drug is available in both forms. Thirdly, we also aligned on consistent calculation methods for some of the specific therapeutics. For ophthalmic drugs, the MDD is based on the API per dose unit, let's call it per drop, how much % API is contained in the drop. Then we take the number of drops applied in a day, and now we have a volume. But we need to get from this volume to the milligram per day. And here we use the application-specific drop size and density to convert the information to the numeric value for the MDD. Drugs that are injectables with specified infusion rates and adjustments. Here we determine the MDD based on the sum of the maximum amount of the bolus dose and then the maximum amount of the maintenance dose using the specified duration for the maintenance dose. And lastly, for inhaled and injectable anesthetics, we determine the MDD based on the maximum infusion rate listed on the labeling, and we consider a 10-hour surgery for duration. That's the variable that's usually not given in labeling. Moving on to my second key element, the single MDD concept. Let me explain what that is. The goal here is to determine the MDD at the time of original NDA approval, and then to use that MDD throughout the drug's life cycle. That means any reference standard and any referencing generic will have the same MDD. This ensures that ANDAs are evaluated using the same standards applied during ILD review. And to note, MDD updates for the ILD that might happen for an efficacy supplement or similar, they are then propagated to subsequent ANDAs. The third key element is knowledge management. Active knowledge management seeks to ensure that the same MDDs are applied consistently across therapeutic equivalents for therapeutic equivalence across disciplines and applications. We're aiming here to develop a process for reviewers to efficiently and reliably identify the MDD. And an added benefit here is that we can preserve our institutional knowledge. And then my last key element I want to touch on is quality control. Now think about it. We're expecting all disciplines to apply the same standardized approaches and common clinical scenarios, and we're expecting that the same MDD is applied throughout the life cycle. Therefore, we need quality control of MDDs that are documented and used subsequently during regulatory review. This is a small group of staff that also provides resources for review staff when questions arise, and resolutions of discrepancies in MDD verification. And then depending on bandwidth, this group also determines MDDs for NDAs that were approved prior to our alignment efforts. Since our alignment efforts are relatively recent, there is still a very large number of NDAs that does not have the single MDD concept yet. So on to my next talking point, recommendations how industry can obtain feedback. If you're a generic applicant and you have questions about the MDD you think should be applied to your product, you can submit a controlled correspondence for confirmation of this MDD for your regulatory assessment and drug development. These controls are particularly useful for drugs with complex dosing instructions, such as drugs that are dosed to effect, to a pharmaceutical effect, and also to drugs with complex routes. For example, topicals comes to mind, because very often labeling for topical does not provide sufficient information to consistently calculate the dosing instructions to this amount per day. And that brings me to the end of my presentation. And in summary, I would like to reiterate that standardization makes determination of seemingly challenging MDDs less complex and therefore streamlines review. I would like to reiterate that MDD is a reasonable standard applied during regulatory assessment. It is not intended to impact dosing in clinical practice. Internally, we're continuing to develop knowledge management in support of MDD alignment, and some of our future efforts seek to further align complex MDDs. These are generally the MDDs that warrant expert clinical judgment. And as a reminder for industry, if you require feedback for an MDD, you can do so through a controlled correspondence. And that's my time, and I look forward to answering your questions at the panel discussion. And next up is Dr. Liken Liang, and he will be talking about quality considerations for generic oligonucleotides. Thank you.
Good morning. Today, I will be discussing some quality considerations for generic oligonucleotides. And quite often, quality considerations are parallel to sameness considerations. So you'll be hearing me talking about both today. Okay. Approved oligonucleotide drugs are short strains of synthetic or semi-synthetic nucleic acids with therapeutic effects. They can be single-stranded or double-stranded, and most of the approved oligonucleotide drugs are heavily modified in the nucleic acid structures. Here are some examples of the modifications. Many of the approved oligonucleotide drugs have phosphorylthioate or the PS modifications at the internucleotide linkages. And if the PS modification is non-stereospecific, it would introduce a chiral center, a new chiral center with a mixture of RNS configurations at certain ratios. If there are multiple PS modifications, it can lead to 2 to the nth power of diastereomers. And in some kind of distribution. For generics, the 2' modification is also common. So as the base modifications and also the conjugation to some targeting ligands. For generic oligonucleotides, the diastereomer composition sameness is very important, and it's one of the most challenging aspects to be addressed. Here's an oligonucleotide analog phosphorodiamidate morpholino oligomers, or the PMOs. The ribose ring, you can see from here, can be replaced by a morpholino ring. And the internucleotide linkage here is a so-called phosphorodiamidate modification, or the PDA modification. If such a PDA modification is also non-stereospecific, it would lead to 2 to the nth power of diastereomers. The next 2 slides will summarize the 24 oligonucleotide drugs the CEDA has approved so far based on the drug product labeling information. From the last column on the right, you can see that the approved siRNA drugs have the relatively small number of possible diastereomers. For example, glycerin has 4 diastereomers on the sense strand and 16 diastereomers in the anti-sense strand. And there are 64 possible diastereomer combinations to form the duplex between the sense strand and the anti-sense strand. For most of the ASOs from this table, you can see the number of diastereomers is much higher, ranging from thousands to millions and to more than 1 billion diastereomers. From this table, you can see defibrotide actually is the only CEDA-approved drug, oligonucleotide drug, that does not have the nucleic acid structure modification. However, defibrotide has its own complexity. It is a polydispersed mixture of short strains of the DNA, ranging from 4 mer to 100 mer, predominantly single-stranded oligos. Listed on slide are some of the key quality and sameness considerations for generic oligonucleotides. Active ingredient sameness studies should confirm the sameness in sequence chemical structures, which would include but not limited to correctness in structure, location, and stereochemistry, etc., as well as the completeness of the modifications. Active ingredient sameness should also include composition sameness, such as the diastereomeric composition, a duplex composition or the profile, double-stranded content where applicable. If necessary, sameness in activity should be evaluated as well. And also the potential in forming higher order structures or aggregates in the drug product needs to be assessed and compared with that of the RD. All of the sameness considerations mentioned above are also part of the quality considerations. Manufacturing consistency should be demonstrated to ensure consistent performance and quality of the generic oligonucleotide. Impurity profile and controls are another challenging areas for generic oligonucleotides. Apart from typical considerations for impurities, the risk in immunogenicity, inflammation, and off-target effect should be considered for oligonucleotide impurities, especially for those that are new or at much higher levels than the RD. The preamble of the proposed rules implementing the Hatch-Waxman amendments states that active ingredient in this context means the active ingredient in the finished drug product prior to its administration. This is very important for drugs, for active ingredient that has the potential for change from the drug substance to the finished drug product prior to its administration. For example, the interaction between the sense strand and the antisense strand in the duplex of the siRNA are not through covalent bonding. There is a potential that the diastereomer pairing between the sense strand and the antisense strand in the drug substance in solid state could be disrupted in the process of the drug product manufacturing. In such a case, or if there's a dispute, the ultimate sameness consideration for duplex of the siRNA should be in the finished drug product prior to its administration. Therefore, it is important to obtain thorough understanding of the potential changes to ensure sameness and consistency of the generic product quality. Another example is the one that can form double-stranded oligonucleotides in the finished drug product. Same considerations. There have been questions about whether a sameness test should be conducted on drug substance or drug product or both. With the implementation rules in mind, the answer to the questions is apparent. It really depends on whether there's a potential to change in the active ingredient related to the test being in question. Firms may provide their justification for their choices. siRNA have relatively small number of diastereomers, as you can see from the previous table. Therefore, quantitative RLD comparison at the individual diastereomer level is quite possible. If one method cannot separate all the diastereomers, one may consider using multiple complementary or orthogonal methods. If strand separation from the duplex is involved before the single-strand stereomeric composition analysis, it is very important to ensure and demonstrate that the strand separation process does not alter the diastereomer composition itself. Also, influences from the co-eroding impurities should be assessed, minimized, or corrected. In addition, the profile of the duplexes in its native state should be compared with that of the RLDs. So diastereomer composition analysis of the oligos with high number of diastereomers is very challenging. This is due to limited resolution power of a single analytical method, compounded with possible co-erosion of the impurities and sensitivity of the method towards various composition changes. The graph here adopted with permission from ACS Analytical Chemistry Journal, published by Rosses and others. The graphs are used here to illustrate the need to demonstrate suitability and sensitivity of the methods. The authors changed the RNS ratio at some selected PS linkages of a 20-mer by using a different activator for those elongation cycles. The table on the left shows how many elongation cycles use the DCI activator or EET activator for each of the 6 experiments. By using DCI or EET, you change the ratio. From experiment 1 to experiment 2, you can see from the graph, only one linkage was changed in the RNS ratio, but then method C on the graph shows quite significant profile changes, whereas method D shows very little change in the profile. And from experiment 2 to experiment 3, RNS ratio of 4 more PS linkages were changed. However, method C only responded with the amount of profile change similar to the earlier one that with 1 RS change. Whereas method D profile changed more significantly than before. So what does that mean? This comparison indicates that large composition change may not always correspond to large profile change, and that a method may have different sensitivity towards different kind of diastereomeric composition changes. So to address the analytical method limitations and to reduce the risk of actual composition differences, one may consider using multiple analytical methods with orthogonal separation principles or complementary methods. The methods should be optimized to maximize the resolution and used to generate multiple detailed and broad distribution profile for ROD comparison. The profiles also serve as the baseline to support potential future manufacturing changes. To demonstrate suitability and sensitivity, one may consider preparing and using multiple diversified and suitability test standards. The test standards may have RNS ratio intentionally altered and verified at multiple sets of diverse but small number of linkages. Quantitative analysis should be performed for each of the orthogonal and complementary distribution profile. The analysis should include quantitative comparison of the overall profile and individual regions or segments of the profile. In addition, the RNS ratio at each of the chiral linkages may be measured after each respective elongation step. This can establish a baseline to facilitate the identification of factors that may impact the ultimate outcome of the diastereomeric composition and to ensure manufacturing consistency. There are some special considerations for oligonucleotide impurities, such as off-target effect, as mentioned before. For generic oligonucleotides, it may be desirable to minimize the potential to generate new impurities. This requires in-depth evaluation of the RD impurity profile, proper design of the synthetic route for the generic, and adequate control of the manufacturing process, etc. I included here a hypothetical extreme case for illustration purposes. Assume that company A designs a 23-mer with the hypothetical sequence shown on this slide. Company A makes the oligo by a linear synthetic approach, consequently adding one nucleotide at a time, as shown on the slide. Some of the common impurities are listed here. For example, the N-1, N-2, N+2, and N-C, etc., and many others. It seems that the target molecule and the typical impurities from this company A do not have the sequences of some of the known immunomodulating motifs. Now, company A conducts a series of non-clinical studies and clinical study to evaluate the safety profile and effectiveness of their proposed drug product, along with the impurity at the control limits. Company A might get the hypothetical approval. So now a generic company B wants to develop a generic version of this hypothetical product. The questions are, will the impurity profile be the same as that of the company A's product? And what about the synthetic routes? So let's say company B decides to use a convergent synthetic approach, since most of the fragments to construct such a molecule are readily available. Company B makes the target molecule by the scheme shown here and submits an ANDA. As you can see, due to the differences in synthesis, the impurity profiles are quite different, and there are new impurities in the proposed generics. One of the potential new impurities with fragment 3 missing actually has one of the known immunomodulating motifs. So of course, if company B performs thorough and comprehensive risk assessment for their ANDA programs, they might choose a different synthetic route. But still, the risk of new impurities, new sequences, and or higher than RD levels of impurities may exist. And the risk in immunogenicity, inflammation, and off-target effects, etc., should be thoroughly assessed. The analysis of impurities in generic oligonucleotides is very challenging due to limitation of the current method in resolving large number of impurities, and in some cases, differentiating some of the impurities. Significant co-erosion may exist. For comparative impurity profiling, and for quality control if necessary, it is highly recommended to use multiple methods with orthogonal separation principles or complementary separation, coupled with high resolution MS or other advanced techniques. Lastly, I'd like to point out that retention time region-based control strategy or similar grouping approach may not be adequate for genetic oligonucleotides without further controls or proper justification or qualification. For innovative products, safety profile of the impurities levels and control strategy would have been qualified and supported through multiple non-clinical and clinical studies, even if the RLD adopted a retention time region-based control strategy. For generics, the analytical method may not be the same as the RLDs, and therefore the retention time region-based criteria cannot be compared directly with that of the RLDs, even if the RLD adopts such a strategy, control strategy. In addition, there's potential for new impurities, higher than RLD levels of specific impurities, and challenges in co-elution and also distinction of some of the impurities. Therefore, leading to higher risk for generics in adopting such a control strategy. And with that, I'd like to thank all of the FDA colleagues working on addressing challenges in generic oligos, especially Melanie, our speaker earlier, and the people listed on this slide. And thank you for your attention. I'd be happy to answer your questions in the panel discussion session. Thank you.
Thank you very much, Dr. Liang. Okay, I invite our speakers to join us back at the panel table. And I invite any questions from the room. If you don't ask any questions, you don't get to go to lunch. All right, we're doing good on time. So any questions in the house? Okay, Corey, it looks like we have to turn to our online questions, please.
We've got a few. So the first ones are actually for Commander Solana Soriende, as well as Dr. Mueller. The first is, if excipient X is not available in the IID database, and it is used in an FDA-approved ANDAs for the same route of administration and dosage form, how can I seek advice about using that excipient in a future ANDA?
Sure, great question. In that case, I would recommend submitting a controlled correspondence for input.
Excellent. Short and sweet there. So we'll go ahead and give you another one, Dr. Mueller. For non-dose proportional strengths, should the MDE be calculated using a mathematical worst-case pill burden of low-strength units, or will the agency accept a calculation based on the highest strength until that reaches the MDD, especially if the mathematical maximum is clinically unrealistic, such as more than 20 tablets per day?
Is the question about calculation of the maximum daily exposure to excipients? That was like at the very beginning. Okay, great. It depends. So we don't actually have a cutoff of 20 pills per day or 20 dose units. So I think submitting a controlled correspondence for specific input is probably the best route to go. I cannot provide a standard default answer.
Excellent. Thank you for that response. My next question will be for Dr. Solana Soriende. Can I use the UNII code to connect the name of my excipient with what is listed in the IID database?
Yes. Short answer to that, yes. Yeah, that's it, because we actually search for our excipients internally like that. We use the Unicode. So if you do have an excipient and they have different grades or forms, you're definitely welcome to use the Unicode in there. Mm-hmm.
Excellent. I appreciate that.
Yeah.
Next question is for Dr. Liang. What are the exceptions in terms of setting diastereomic composition acceptance criteria for sameness evaluation or routine controls?
Thank you for the excellent question. It's actually a tough question. So first of all, acceptance criteria is a review issue, and the adequacy of those proposals would be determined during the full ANDA review. So in terms of the sameness for diastereomers, there are two kind of difficulties in here. So for siRNAs, I mean, as I presented during the presentation, the number of diastereomers is small. So you could separate each single diastereomers, it's quite possible. So in that case, it may be very simple because you can compare the individual diastereomers with that of the ROD and find out the ROD availability and propose some controls, acceptance criteria, and also demonstrate the sameness. Now, as of today, none of the approved oligonucleotide drugs in their labeling specify a particular diastereomers as the active ingredient, at least not so far. So any diastereomers, I mean the full-length target molecule diastereomers, would be, if you find it in the RD, would be considered part of the active ingredient. So you would have to match those levels. So because each individual diastereomers is considered part of the API, so you could probably apply those concepts for the assay for each individual diastereomers. For example, you can set a plus or minus 10% or 90 to 110% of the RD value, the average RD value. That's based on the assay concept. So you control each individual diastereomer that way. That may be easier to justify. Now, it is more complex for ASOs, which has a lot of diastereomers, and you cannot separate single diastereomers. And what you get is the profile. So it's very complex, as I shown earlier. Those analytical methods may not be sensitive to all different, same kind of level, sensitivity level to all different kind of composition changes. So there's a risk there. I mean, even there's no change in the profile, it doesn't mean that there's no change in the composition. So you would need to use multiple methods, and it gets a little bit more complex. So I would highly recommend that you engage the agency further, either in a pre-end product development meeting request or some kind of control correspondences. So, thank you.
Thank you for that response. My next question is also for you, Dr. Liang. Are there any plans for platform-level guidance for oligonucleotides like ASO, siRNA, apart from PSGs in the future?
Yeah, I would direct this question to OPQ or OGD, I mean, the policy group, probably. So, thank you.
Of course, thank you for that. My next two questions will be for Commander Solana Sodiende as well as Dr. Mueller. Are MDDs for drugs listed in any publicly available database?
That's a great question. There is no publicly available database that lists MDDs for drugs. Not at this moment. So if there are any questions for a specific drug, submit a controlled correspondence.
Excellent. Thank you for that response. And Dr. Mueller, your next question. For the purposes of calculating residual solvents in an oral solid dosage form, and the maximum daily dose is presented in the labeling as milligrams per kilograms per day, what should be used for the kilogram? If the PI indicates a higher than the standard range of weight, does the higher weight need to be used in the MDD?
So I'm gonna go out on a limb and make an assumption that it depends. If the patient population is not inherently obese, let's say it's not a drug product to control for glycemic control, for example, and the patient would be considered standard weight, then the 60 kilogram could be used. However, if it's a drug where the patient population would significantly fall out of the standard range, meaning either obese or very low weight, then you have to go by. I would recommend going by the clinical disease state. What's more typical for the clinical disease state? Thank you.
Thank you. The next question is for Commander Solana Sodiende. What are some limitations of the Inactive Ingredient Database, or IID?
Of course. Yeah, like I initially mentioned, it doesn't present the context of use or the duration of use. And that's just a limitation, and because it's associated with the products, so we have to be very careful what we do publish. The IID also doesn't have information, because we've received some questions sometimes about excipients in biologics or in over-the-counter medications, and we do not publish those excipients in there. So we'll just reserve the IID for new drug applications and approved abbreviated new drug applications, the generic drugs. Let's see, what else? Other limitations. One thing I can say is we do always get suggestions and, about enhancing our IID. So I do encourage people and industry to submit their questions because we do take that into considerations. We have a lot of discussions behind closed doors internally. And so, for instance, we've heard about populations considering like the pediatrics. So we've heard that. And we're just making some discussions about that. So that's what we welcome. That's all I have to say about the limitations. Yeah.
Thank you so much. We do have one brave soul who wants to have lunch today with a question in-house. Go ahead, sir.
Yes, question is for Diana. Regarding the IID, I saw you had a slide on the, I guess, evolution of the percent of MDE that's been updated. It went from 4 to 44%. So good job there, I guess. Is there a target that you're looking to achieve in the percentage? Is there a goal and a time frame to achieve that? Just curious about that.
It's a very good question. Thank you so much for that. Yes, it'll be nice to say, in an absolute format, 100%, right? But we want to shoot for at least 70% plus. So that's why we have a lot of enhancements going on to our systems. We're trying to modernize them as well and use different IT systems. So slowly but surely, but we are optimistic. We are working behind the scenes really hard so that we can get as many MDEs completed for industry to use when, manufacturing their products. Yes. Thank you so much.
Is there any time frame that you're shooting for? I know it's probably behind.
It depends. I can't really give a set timeline. Trust me, we're working hard and we are ambitious. However, there are lots of factors to consider, but we have you in mind. Yeah. So thank you. That's as much as I can provide. Yep.
Thank you.
Stay tuned. Yep.
Second question from the House. Thank you.
Hello.
Hello, ma'am.
Rebecca Dandecker, Wiley Ryan Law Firm. I noticed that on the slides, you give information about reaching out to the IID team, mostly through controlled correspondence, but you also provided the direct email.
Yes,
Can
ma'am.
you clarify when we should use that direct email? Because it seems like controlled correspondence is the default.
Yes, actually for us, the default is the IID update mailbox. So it's for general. We get a lot of general inquiries, just everyone asking, I noticed this specific excipient level. I just want to, ask, is it correct? Or is it possible is that? Or I noticed something was on there and it was replaced. Can you provide a rationale? The ones, as it relates to the IID, right, and the excipients, anything that is related to an application specific. So we've had, a few maybe requesters that will ask us questions, and it's a general question, starts out as that, but they want a specific answer, more tied or customized for their own application because maybe the manufacturer is manufacturing an excipient for a specific drug product application. And in those situations, we say, yes, go through the control correspond route, because then we really route those questions to the specific subject matter experts that can do more digging and more research for the specific application and then respond to the requester that way.
Thank you.
You're welcome, ma'am.
Thank you. Okay, we'll give our house people a chance as long as they want to come up to the mic.
Because everyone loves IID, I figured I might have said
Okay.
Candace Edwards from Intentus Strategic Regulatory Consulting.
Yes, ma'am.
So if a client has done a meeting, let's say a meeting, and in that meeting, as part of the development meeting, is asked an IID question, will it get the same level of review and as opposed to if you do a direct control correspondence? So will you get the answer, or do you need to have that control correspondence?
No, you don't necessarily have to have that.
Okay, so it can be addressed.
It can be addressed.
in a
As
meeting?
long as it's not tied to a specific application, then we have to go through the control correspondence route. But if it's just a general question,
no, it would be specific. So it would
it would be specific?
Yeah, development meeting for a product.
Yes,
that might include a question.
Yes.
You would still get the same level of review as if you just okay.
Yes, you would. In fact, you might even get a more intense review because it's a control and it has a clock on it, 60 days. We have clocks also for it. We have timelines for the general inquiries, it's 30 days, but with the control, it's more targeted. So you should be able to get a response within 60 days.
Thank you.
You're welcome, ma'am.
Thank you. Okay, Corey, back to online. We have 5 more minutes.
Of course, then our next question will be for you, Dr. Liang. What should be the reporting threshold for any unspecified impurity in oligonucleotides, given that the FDA has not provided specific guidance? FDA has published guidance for peptides. Can the same limit of NMT 0.10% for unspecified impurities be considered for oligonucleotides?
So the reporting threshold should be lower than, typically should be lower than the identification threshold. So there's a relationship there. However, for the identification threshold, it's a case-by-case basis at this point. And the FDA has not published a guidance on setting those thresholds yet. So I think once you determine the identification threshold, of course, with a lot of risk assessments and justification, and then you can set your reporting threshold lower than that. And of course, the reporting threshold has something to do with your method sensitivity. For example, the LOQ and all those kinds of things. So you can propose something and justify it, and then we'll review it case by case. So.
Excellent, thank you. And our next question is also going to be for you, Dr. Liang. Is it acceptable to use one method for routine testing of diastereomeric composition?
Good question. So it really depends on whether that method is capable of accurately determining and sensitively determining the composition and also sensitivity composition changes. If that is not the case, then multiple methods may be required, and whether that's the same as testing or routine testing. Now, there's a possibility that after you get the approval and after you get enough information or data to support that method is sensitive to these changes, and you have demonstrated consistency of your manufacturing, and sounds like your quality is stable. Then post-approval, you may propose something to change. Maybe reduce or eliminate one of the many tests. So it is possible, but that should happen post-approval.
Excellent, thank you. And I think we have time for at least one more question. So this one will be for Dr. Mueller. How should drugs that are not applied on a daily basis, but rather weekly, monthly, or at longer defined intervals, be addressed from the MDD perspective?
That's a great question. Thank you for that. Generally, we apply the amount administered within 1 day, even if the drug itself is administered in intervals. So what is administered that day? For example, transdermal systems, you apply a transdermal assistant on Monday, that is considered your maximum daily dose, even if the system stays on for 5 days. And the reason is that we evaluate not just, so we're actually not looking so much towards the API, but we're looking at residual solvents, extractable leachables, excipients, impurities. So, and we do not have time release information available for either of those attributes generally. So therefore, worst case scenario, we apply all goes in at once, which is the day the drug is administered. Great question. Thank you.
Okay, thank you very much to speakers and panelists. Thank you so much for coming out. Thanks, Lori. So I'm going to be good. I'm going to let you guys go for lunch, okay? Some people did step up to the mic. 12:45 to 1:45, lunch break. Please be back here at 1:45. We will start on time. Reminders, the survey link where you can do the survey, download the certificate of attendance is going to be sent in today's email. So look out for that email and do the survey, please. You have only 2 weeks to do it. Get your lunch, free to roam about on the patio area immediately adjacent to the building, and please be back for the afternoon session. Thank you.
Let's see, it'll be interesting.
Yeah.
Good afternoon, everyone, in-house and online. We have a terrific surprise visitor, our very own FDA Commissioner, Dr. Marty Makary. Please. He was in the neighborhood. I told you what you get when you get to FDA campus, right? And he has we are so, fortunate to have his presence here. So thank you, sir.
Thank you, Dr. Stoddard. Well, thank you, Dr. Stoddard, and thank you, everybody who's interested in this topic of generic drugs. I understand this is the first of 2 days where you'll be talking about generic drugs. It's not enough. We probably should have a 2-week conference with a hackathon at the end because it's really an important topic for so many Americans. As you know, access to drugs is an issue in the United States, and that's because of the lack of affordability of drugs that has been a problem for decades. And now that we have expensive drugs like biologics, it's important that our biosimilars, which I know is a separate discussion, are also more available to Americans. If you have a drug that is curative, if we theoretically, as a medical profession, discovered a cure for breast cancer, but that drug was so expensive that half the population could not afford it, or half the population wouldn't take it because they don't trust us as physicians, then on a population level, that drug is only 50% effective. And you can keep going down that scale. So affordability is an access issue, and it's one of the massive priorities that we have outlined. It's a massive priority in this administration. We have done everything we can to try to see what we can do short term and what we can look at long term on this issue. As you may know, on the biosimilars side, we eliminated the confirmatory efficacy trial that saves about $300 million or so, and a couple years from the approval of the generic versions, if you will, of the expensive biologics. With traditional generic drugs, we have seen more approvals by the scientists here at the FDA. In the last year, approvals are up about 20% to 91 generic drug approvals last year. And that means more competition. More competition can mean more price transparency by which people are able to see lower prices. For example, one of our pushes is to increase the number of drugs that are over the counter, because when a drug is over the counter, something magical happens on the shelf of a store. You see a price, and that price enables shoppers to compare the cost in real time as they look at the attributes of any product. It is different from when you pick up a medication behind the counter of a pharmacy with a prescription, where an employer may be playing through a PBM, a money game by which there's a shell game of several known mechanisms by which the price may be inflated, and it may also, you're paying for it indirectly through a paycheck deduction and not as much through directly out of your wallet. So what happens is we end up with spread pricing. We end up with the improper categorization of some drugs as specialty drugs, which require a specialty pharmacy with limited options. There are about 6 or 7 major games that are played by PBMs. So we have included generic drugs on the new website known as TrumpRx, where people can buy direct if they'd like. We want to do anything we can to try to encourage more competition and more price transparency. On the GDUFA legislation that is being discussed right now, we're very open and honest about the fact that we would like to see an America First strategy to bring more generic manufacturing to the United States, more APIs to the United States, and more key starting ingredients to the United States. Now, we have to work within the system that we have and do what's feasible and work in an incremental way. So we have asked in all of our user fee negotiations for an America First approach. So if you're going to make a generic drug in the United States, we want to reward that in the user fee program. So all of the teams at the FDA that are negotiating user fees know that we would like to see a financial incentive to companies that make their products in the United States. We want to see more drugs made in the United States, more domestic manufacturing. We want to see American companies thrive, and we want to see companies that do business in the United States thrive. I know you're discussing many different issues in this two-day conference, so I just wanted to come in and say thank you. You don't have to be here. You don't have to be listening in virtually and participating in this, but this is an important discussion that directly relates to everyday Americans who pick up their medications or have them mailed to their home. It is one of the central issues for people who are taking medications, is the affordability of those medications. So thank you for being here. Thank you, Dr. Stoddard, for letting me crash in here. But we were able to carve some time out, and I did want to come by and just say thank you for your work. So appreciate the opportunity. Thank you.
Thank you. All right. So you see what you get when you come in person. Or if you sign on back on time. This was indeed a pleasure and a total surprise. So how much time do we have now? We're continuing. All right, so let's start our afternoon session. And after this, I hope we're going to get questions from the room, right? All right, so what we're going to do, we have a couple presentations that have shared presenters. So we'll start with suitability petitions, and that will be shared by Karen Rothschild and also Dr. Andrew Fine. Then we'll move on to prioritization request, and that will be shared by Dr. Bailey Laston and Dr. Andrew Kim. At 2:25, we'll do the addressing patent and exclusivity requirements, regulatory reminders, and that will be done by Dr. Heather Strandberg. And I'm going quickly because we have time to catch up on. Then Orange Book, how drug product listings are added, updated, and maintained. That will be done by Dr. Trung Quash. And then we'll segue into the questions and panel discussion. We'll keep to the times allotted, even though we're a little bit later. So let's bring aboard our first group of presenters, Dr. Karen Rothschild and Dr. Andrew Fine. Thank you. Andrew, you're here.
Hello, as you heard, I'm Karen Rothschild. I'm a regulatory counsel in the Office of Generic Drugs Policy. Welcome back. I hope you all had a nice break, and I hope you enjoyed that special visit from the commissioner. Let's see, I'm going to be talking to you about suitability petitions, basically what they are, how they work, and what you need to do. Ooh, I need to... If you are developing a generic drug product that differs in any of the ways you see listed on this slide from the RLD that you're referencing, you will need to submit a suitability petition before submitting your ANDA. A suitability petition is a way for your company to ask FDA whether they agree that an ANDA pathway is the correct pathway for submitting your application. If it isn't, it can always come in as an NDA, a new drug application. But here we are specifically talking about ANDAs, the abbreviated new drug application. You need to have an approved suitability petition before you can submit your ANDA. A suitability petition is a type of a citizen petition. So of course that means that there are going to be technical requirements as far as how to submit it. Fortunately for you, it's all outlined in 21 CFR, that's the Code of Federal Regulations, at 10.30. You need to upload your suitability petition to a docket, the one that's identified, and then you will be given your own docket number that's going to be specific to your product and this suitability petition. When you correspond with the agency, please always remember to reference that suitability petition docket number, because it's not only part of the law, but it's how we track what we're doing with you, and you don't want to cause delays, and we don't either. There are 5 parts to a citizen petition, and fortunately, really only 3 of those apply to suitability petitions, and that would be sections A, B, and E. The A section, action requested, is where you want to identify that this is a suitability petition and that you are seeking agency's opinion on whether or not the ANDA pathway is appropriate for the changes that you are proposing. The statement of grounds is exactly what it sounds like. It's where you provide all the factual and legal evidence that you can to aid the FDA in evaluating whether or not this is an appropriate product for the ANDA pathway. The last section that's really important on the traditional citizen petition is the certification, which is just certifying that you have provided all information, legal or scientific, that supports or doesn't support your ANDA pathway, so that a really good decision could be made at this stage of the game. Now, in addition to what's in regular citizen petitions, there's a few things that are specific to suitability petitions. You will need to identify the reference listed drug, the RLD. You need to provide a copy of the current RLD label. And if your product is no longer being marketed, you need to find the most recent RLD label that you can and present that. You also need to provide a copy of the label that you're proposing for your drug formulation. An additional thing that's helpful but not required is a screenshot from the Orange Book, which will let you know which of the RLD. And that will also help you determine that you have correctly identified an RLD and that the RLD isn't withdrawn from the Orange Book. If it has been discontinued, if it's in the discontinued section of the Orange Book, there's often a little piece of information on the side that says that it's been determined that it was not withdrawn for reasons of safety or efficacy. That needs to be there. It needs to have been published in the Federal Register. If it is not there, you can request that the agency do one by submitting another citizen petition. Some suitability petitions also trigger the need for a waiver of the requirements. You need to request a waiver of the requirements of the Pediatric Research and Equity Act, which is known as PREA. PREA gives FDA the authority to require pediatric studies in certain drug products and certain biologics for the purpose of obtaining appropriate pediatric labeling. If the change does trigger a PREA review, you will need to include the reasons why it should be waived in your situation. If FDA is not able to waive the PREA requirement, you cannot have the suitability petition will be denied, so it will not be appropriate for an ANDA pathway. One thing to point out is that the guidance document that's referenced on this slide is a replacement of a previous guidance document. So another sort of just general recommendation and reminder to make sure you're looking at the most current guidance documents that we have available. So you might have noticed that one change in the suitability petition that was not on the previous slide was for strength. If you are only looking for a change of strength in your suitability petition, you are waived automatically from the PREA requirements. You do not need to submit a waiver. let's see. The guidance that I mentioned before, the one that was updated in May of 2023, that provides all the information you need on the criteria to request a waiver of the PRIA requirements. And the information on this slide is taken directly from that guidance. You can see it even gives you citations, so you know exactly what legal language you need to include. The most common reasons for a suitability petition to being denied are finding that investigations are needed to establish safety or efficacy due to the changes we've proposed. Another one is that there's already an approved NDA for the change that you are seeking. You will get your suitability petition denied because that's not an appropriate we can't approve a suitability petition if there is an ANDA an NDA for the change that you are requesting. That just means you can just go ahead and send an ANDA with that as your basis of submission. The other common reason for a denial of a suitability petition is if there's enough change to your proposed drug product that it's going to require significant labeling changes. It can't differ too much or what we call significantly from the RLD labeling. So remember, if you're denied, your suitability petition is denied, it does not mean that you cannot come in and get approved as a drug by FDA. It just means that the ANDA pathway is not appropriate. Likewise, if you are approved and get an approval for your suitability petition, that is not a guarantee that you're going to be approved as an ANDA. It just means that you are able to be evaluated as an ANDA. So with respect to best practices, before you submit a suitability petition, please check the Orange Book. Remember, this is where you can make sure that if the RLD has been withdrawn, it has a safety and efficacy determination made. If it hasn't, then you know you need to ask for one. It also is where you can check and make sure that there is no NDA approved for the change that you're seeking. And the other thing is just make sure that your labeling doesn't have to differ so much from the RLD labeling that it's going to go against the same labeling requirement, which is a little bit of a misnomer. It doesn't have to be the same labeling. It needs to be pretty darn close. So now I'm just going to quickly take you through some GDUFA 3 updates and tell you how we're doing. Our commitment under GDUFA is with respect to completing a certain percentage of reviews of suitability petitions within 6 months, with the percentage of those reviewed going up each year until we hit a certain percentage, up to 90%. And the numbers go up as well. Well, as you can see from the slide, we're really pleased to say that we actually have surpassed our ultimate goal for 2027. We have last year, we reviewed more than 90. It was 94, and we got them all done on time. So if you are, unless something goes sideways, you can pretty much count on us getting a final response to your suitability petition within 6 months. So the petitions, you can see how many we've received. Over the last couple of years, we received approximately 100, give or take. Last year, we didn't miss any goal dates, as I mentioned, and of course, that's always our goal. The majority of proposed changes in suitability petitions is for the change in strength, followed by dosage form or a combination of dosage form and change of strength. From this slide, you can see that in fiscal year 2024, approval versus denial rates were approximately half and half. Last year, we had more approvals, so we'll see what this year brings. You'll also see the most common reasons for denial, which I just touched on. So now I'm going to pass the podium to my colleague, Commander Andrew Fine, who is the Associate Director for Regulatory Affairs in Office Generic Drugs, Office of Safety and Clinical Evaluation. He'll take you through a little deeper dive on how suitability petitions are assessed. Thank you.
Thank you, Karen, and good afternoon, everyone. Pleasure to be here and really to share the work we've been doing under GDF 3 as we've really surpassed all those expectations under the GDF 3 commitments for these petitions. There's really three main pillars when we assess petitions in OGD. The first one is we look at the petition itself. What is the proposed change and the rationale from the petitioner? We took a really close look at RLD labeling, focusing on the sections listed here, and I'll talk about these in the next subsequent slides. And lastly, we do look at the proposed labeling. Proposed labeling is a requirement for that suitability petition. So we do get a glimpse at how this proposed change would translate into potential changes in the proposed labeling compared to the RLD. But fundamentally, when we look at petitions, we're really answering this question: does it fail any of the criteria established in 31493? There are clear reasons for denial for petitions, primarily if there are safety and effectiveness questions that would require investigations or significant labeling changes that would not be permissible under an ANDA. That is really what we're looking for when we look at these petitions. So I'm going to walk through our assessment approach for the two main buckets of petitions, which are the strain changes, which is about 75% of all petitions, and then the dosage form changes. And I'll take a close look at orally disintegrating tablets, or ODTs. These encompass more than 50% of the dosage form changes we've seen over the past few years, and really shed some light into how we assess these petitions. For strain changes, it begins with that RLD labeling assessment. We look at RLD labeling and really closely at the RLD dosing. Is that RLD dosing weight-based? Is there a dosing range? Is there a fixed dosing titration? Is there a fixed dose titration? How flexible is that RLD labeled dosing to allow for the proposed change in this petition? For the specific strength change that's referenced in the petition, we look if that strength itself is actually mentioned already in RLD labeling. This could be in the dosage administration section or special populations, for example. And if you take these first two bullets, this final bullet really encompasses what we're trying to piece together. Does the change need new dosage administration instructions? Traditionally, I mean, if it does, that likely could be grounds for denial. But really, the labeling should be limited primarily to the house supplied section or dosage forms. We're not trying to create new dosing under a petition or a petitioned ANDA. To show how these come together, this slide really delineates what RLD labeling will say for approvals of a strain change versus a denial. So for approvals, if that strength is explicitly mentioned in the dosing instructions, those would be appropriate for an approval. For intermediate strengths, is that strength between the approved strains and supported by a dosing range listed in labeling? Is that additionally with that dosing range, is there weight-based dosing that allows the flexibility of an additional strength that's mentioned in this petition? That strength could be implied by titrating or individualized dosing that's clearly part of that dosing information in the RLD labeling. And lastly, if the special population allows for maybe a reduced dose for hepatic impairment, and that proposed strength corresponds to a lower dose that's mentioned in the RLD labeling, that type of change would be approvable. In contrast, for denials, primarily this includes lower strengths that are below the approved dosing range, an intermediate strength that does not have specific dosing information from the RLD labeling, if it's a higher strength that's outside that dosing range, and if that RLD labeling is rigid and does not permit individualized dosing that would be necessary for the proposed strength change. To illustrate these principles, I'll share a case study of a recent petition for a strength change. This petition 2025 P2077 was proposing a strength change of fluoxetine capsules, proposing additional strengths of 15 milligrams and 30 milligrams. The RLD in this case is fluoxetine capsules that currently is available as 10 milligrams, 20 milligrams, and 40 milligrams. We approved the 30 milligram strength because there's a clear dosing range and flexibility within the RLD labeling for several indications. It states a dosing range of 20 to 60 milligrams is recommended, and with doses up to 80 milligrams being tolerated, that 30 milligram fits into that 20 to 60 dosing range. In contrast, we denied the 15 milligrams because for the specific indication for lower strengths, the instructions are clear that you initiate treatment with a 10 milligrams per day, and after 1 week, would increase the dose to 20 milligrams per day. 15 milligram is not mentioned. It was not studied in clinical trials. In dosing, and the effectiveness and safety of a 15 milligram strength is unknown. Therefore, we issued a partial grant, approval of the 30 milligrams, and denial of the 15. Switching gears to the ODT changes, I'm going to walk through some general principles that we've applied to ODT changes. Again, this is the most common dosage form change requested under a GDF 3 suitability petition submissions. There is precedent for ODT petition approvals. Notably, carbidopa-levodopa, the RLD is Cinemet here. We approved a petition for an ODT change in August of 2002, and subsequently approved 4 ANDAs referencing the RLD as well as that approved petition, and those are approved and listed in the Orange Book. Again, a lot of these, as Karen mentioned, these changes could be submitted as a 505(b)(2) since they're different than this RLD product. And we have approved ODTs under a 505(b)(2) pathway with limited clinical investigations restricted to BE testing, which then would be translatable and allowable under an ANDA if a petition ANDA would only require BE testing for the proposed ODT change. One caveat with ODTs that they are not intended to be split. So if the RLD labeling has dosing for split tablets or instructions of how to split the tablet, that could be concern of a petition for an ODT change because those instructions would not align with the RLD and would not be feasible with an ODT formulation or dosage form. So to illustrate an ODT change, here's one case study of FDA 2023-4293 for glycopyrrolate ODT, 1 milligram, 2 milligram. The RLD here is glycopyrrolate tablets in the same strengths of 1 milligram and 2 milligrams. There was a detailed assessment consideration which noted that there is an approved NDA product that was approved as a 505(b)(2) for glycopyrrolate ODT, but it had different strengths of 0.85 and 1.7 milligrams. An ANDA applicant could reference this approved ODT product as a 505(b)(2), but they chose a petition route because they wanted to match the strengths of the RLD product of 1 milligram and 2 milligram. And during the 505(b)(2) development and ultimate approval, it was determined that the strength of that API was adjusted from 2 milligrams to 1.7 after initial BE pilot study. While there are concerns about potential bioavailability differences between an ODT and an oral tablet at that same 1 milligram and 2 milligram strengths, that bioavailability question would be addressed in a future ANDA. And those differences in bioavailability will be part of that scientific review and not part of the petition assessment. And therefore, there are no clear grounds for denial at the petition stage. Our final recommendation was approve. Just briefly, I'll touch on proposed labeling and how we look at proposed labeling. We primarily look at how the proposed change affect the current RLD labeling, which sections are going to be impacted. Is it only the house supplied section or dosage form changes, or does that proposed labeling for a future ANDA with this change require administration and preparation instructions, new dosing information, or new warnings? We must consider if the changes will be needed and if such changes would be allowable in an ANDA. At the petition stage, while proposed labeling is submitted in that petition package, the labeling assessment is not as thorough as it would be in a future ANDA. Labeling changes in general for petitions, for suitability petitions, changes in the house applied or dosage form strengths are generally permissible, as well as additional administration instructions to describe how to use those proposed product. These are viewed in general as permissible labeling differences. However, in contrast, new dose instructions and indications, new warnings to safely use the product, those most likely would be grounds for denial, as there's an unanswered clinical safety and effectiveness problem with the proposed change. As I stated in the onset, our rationale for assessment is really determining if there are any unanswered safety and effectiveness problems. And in this situation, if labeling would be needed to address such a safety or effectiveness problem, those are grounds for denial. I'll close with just some reminders about future ANDAs. So that if future ANDA citing a reference listed drug as well as an approved petition, we consider those petition ANDAs. And those will require adequate BE testing or additional information to show that it has the same therapeutic effect as the reference listed drug. Note, because these products are not pharmaceutically equivalent to the RLD, if approved, a petition ANDA will not be therapeutically equivalent to the RLD, like most 505J ANDAs. Reminders about dosage form change is that the ANDA labeling should describe administration instructions if they're applicable, and that appropriate BE testing is still required and should correspond to specific administration instructions for that dosage form change. And lastly, just to emphasize that an approved petition does not guarantee future ANDA approval. It really is granting permission to pursue a future ANDA as a petition ANDA under a J pathway. That's my last slide, and I'm going to pass it over to Bailey Larson, who's going to talk about prioritizing original and supplemental ANDAs. Thank you.
Hi, good afternoon. My name is Bailey Larson. I'm on the patent and exclusivity team in the Office of Generic Drug Policy. I'll be presenting today with my colleague, Commander Andrew Kim, from the Office of Regulatory Operations about prioritizing your original and supplemental ANDAs. I'll be using my time with you to review criteria that OGD uses to assess a valid prioritization request, recognize the most commonly used prioritization factors from the map, discuss key considerations when selecting a prioritization factor, and I'll provide an overview of the new onshoring prioritization pilot. I'm starting with this slide because it is the most important takeaway from this presentation. When submitting a request for priority review, all of the following criteria must be met for your ANDA or supplemental ANDA to be considered. Out of order. Number one, the cover letter to the submission must clearly state priority review requested and reference the ANDA number. Number two, the basis for the request must be consistent with one of the factors in the map. The map is titled Prioritization of the Review of Original ANDAs, Amendments, and Supplements. Three, the applicant must clearly and briefly state the basis for the request, including the factor. And number four, the applicant must include sufficient supporting documentation of the request. It's important to note that submissions related to drug shortage, public health emergency, or meeting the requirements of Section 501J11A of the FD&C Act may be eligible for priority review in the absence of an explicit request. So today I'm going to focus our short time on the two common factors that we see. The first is submissions for which there are not more than 3 approved drug products. And the second factor we'll be discussing is applications containing P4 or paragraph 4 certification. So if you are requesting priority review under the not more than 3 approved drug products factor, it's important to note that there must be no blocking patents or exclusivities in the Orange Book. This includes a patent that an ANDA is required to address with a paragraph 3 or a paragraph 4 certification. So often we see proposed justification that litigation has been dismissed with certifications that have a P4. And it's important to note that that still counts as a blocking patent, no matter the status of the litigation. If your ANDA has a paragraph 4 or P4 certification, it may be eligible for priority review under this factor. We encourage you to view the status of the drug product and 180-day exclusivity on the paragraph 4 website. And documentation is really key here to show that your ANDA will be eligible for final approval before the goal date. There's 3 circumstances where this factor applies that's important to understand and use in your documentation for this request, depending on the status that's listed on the P4 website of 180-day. The first circumstance is if you are a first applicant and 180-day exclusivity has not been extinguished. Second circumstance is if you're a subsequent applicant and 180-day exclusivity is triggered but not expired. And the third circumstance is for other ANDAs with P4 certifications if there are fewer than 4 approved drug products in the Orange Book, including the RLD. In other words, if 180-day exclusivity is extinguished, but there's still fewer than 4 approved drug products. These 3 circumstances you will see bulleted in the map under this prioritization factor and should be noted in your request. So some helpful tips for your requests. Make sure you are selecting the right prioritization factor with clear justification. If the wrong factor is selected, or any of the criteria from slide 3 that I mentioned earlier are not met, your ANDA may not qualify for priority review. There's no additional benefit to meeting multiple prioritization factors, and there generally must be an explicit request at the time of submission that includes the prioritization factor or factors under which the applicant believes the submission qualifies. If you are submitting an ANDA supplement or amendment to a supplement, only these factors apply. Drug shortage, public health emergency, submissions related to certain government purchasing programs, submissions subject to statutory mandates and other legal requirements, or requests pursuant to 21 CFR 31470B4, otherwise known as extraordinary hardship. Now I just want to bring to your attention and awareness the new ANDA prioritization pilot to support U.S. generic drug and manufacturing and testing. This was announced last fall. The criteria include the finished dosage form manufacturer or manufacturers must be located in the U.S. The active pharmaceutical ingredient or OPI suppliers must also be located in the U.S. And either the pivotal BE testing was conducted in the U.S. or the ANDA qualifies for a waiver of BE testing. Under the current pilot, all 3 must be met to qualify. And here's my challenge question. So a request for priority review must have which of the following? A cover letter that clearly states priority review requested. B, a clear statement of the basis for the request, including the factor or noting the onshoring prioritization pilot. C, supporting documentation for the request. D, a basis for the request that is consistent with the map, or once again, the onshoring prioritization pilot, or E, all of the above. And the answer is E, all of the above. Commander. Thank you, everyone.
All right, good afternoon, everyone. So this is part 2 of the prioritization request presentation that I'll cover. Bailey did a great job presenting on some of the most common prioritization factors and some helpful tips in the new prioritization pilot. So over the next 10 minutes, I'm going to go over some of the learning objectives today, which are what is the priority review and what are some of the benefits? Describe the process for prioritizing original ANDAs, amendments, and supplements. what are the roles and responsibilities? So who are the decision makers responsible for applying the prioritization outline in the map to the review of specific submissions? And then finally, I'll cover some of the common mistakes to avoid when submitting a prioritization request. So we'll start off with a poll question. Which of the following is not true? A, original ANDA or amendment must meet one of the prioritization factors in map 5240.3 to qualify for priority review. B, an ANDA supplement that addresses a public health emergency qualifies for priority review. C, FDA will evaluate whether priority review will be granted without an explicit request from applicant at the time of submission. Or D, submissions that are eligible for priority review may receive a shorter goal date or expedited review. All right, so I heard C, that's correct. So this is a little bit of a trick question if you were paying attention to the last presentation from Bailey. So FDA will not evaluate a priority review without an explicit request. But there are certain circumstances or exceptions to that rule. So meaning if there's a drug shortage, a public health emergency, in those cases, FDA will still evaluate that and determine whether that could qualify for priority review. But my recommendation to you would be, if it meets the map, always explicitly request it. It's recommended that you do that, and it'll be quickly processed, and you'll get that priority review acknowledged sooner. So some of the key points that I want to talk about is, what is priority review? So priority review is governed by MAP 5240.3. The current version is revision 6, which was issued in October of 2022. This map establishes what we call prioritization factors, so specific public health priorities that qualify your submission for faster review. So there's two main benefits to priority review that I'll cover in the next few slides. And then I'll go over how to request that priority review. So once you determine that your application may qualify for that prioritization review, what are the steps taken internally in the agency before it gets granted and acknowledged as a priority review? So the two main benefits, the first option, which is the best option, would be the shorter goal date. So according to GDF 3, for an original ANDA, it would qualify for an 8-month goal date versus a 10-month, which is the standard review process. For major amendments, if you receive that priority review and receive that shorter goal date, it would be either a 6-month, which is ones with that don't require inspection, or 8-month if you submit PFC 2 months prior. And then for prior approval supplements, it would be eligible for a 4-month goal date, which is ones that don't require an inspection, or an 8-month goal date for those that submit a PFC 2 months prior as well. I just wanted to also note here that we're not really talking about, in this point, about any minor amendments, because for those, they don't qualify for that shorter goal date, but they would be eligible for that second benefit, which is an expedited review. So that would mean that there's no change to the actual goal date, but FDA will strive to act as soon as possible, and they may complete the review prior to the goal date. So I'm going to talk briefly and kind of show you a visual picture of kind of what happens once you submit that priority review application. So step 1 would be the submission is received. So applicants will request that priority review in a cover letter, either for original ANDA, for an ANDA amendment, or a PAS multi-discipline supplement, or OPQ, or labeling only supplement. Or the other case, as I mentioned, would be that FDA may still identify that submission as qualifying for priority review if it's a drug shortage or public health emergency. The second step would be the evaluation stage, which is at the time of submission, we would review that prioritization factor and assign it to, it would first go to an identifier, and then we would assign it to a validator. Ideally, you as the applicant will be explicitly requesting that in the cover letter, include that prioritization factor or factors which you believe qualify for quality review, for the priority review, and state the basis for the request, including sufficient documentation. The third stage would be coordination. So depending on if it's the OGD RPM, or it could be filing review, or the labeling or OPQ RBPM, they would coordinate with OGDP or other validators, and they'll consult with OGD review disciplines and determine which is the next step, which is the prior review will be granted or denied after consultation with the validator. If it qualifies for that shorter goal date, we'll update that goal date in our system, and you will receive an acknowledgement letter specifying that shorter goal date. Or if it's an expedited review, it will be labeled in that project, and it will allow all review disciplines to know that this qualifies for that expedited review. And then finally, the review proceeds. So prioritized submission will be reviewed according to the assigned timeline, or will be prioritized internally in queue. So some of the roles and responsibilities, as I mentioned, the OGD regulatory project manager, or RPMs, they have the overall responsibility for applying the prioritization policy for original amendments, multidiscipline supplements, and they coordinate to ensure consistent prioritization across offices. The OPQ RBPMs are responsible for OPQ managed supplements, and the OGD labeling PMs would be responsible for labeling supplements only. OGD DFR, they identify those prioritization requests during filing review. So at the time of the original submission, they'll communicate with relevant OGD staff as well as the RPMs. OGDP will work with RPMs, DFR staff to identify and validate prioritization for original ANDA amendments and supplements. So some of the common mistakes that I want to go over. So the number one mistake that we see is a timing issue. So you have to make your priority review request at the time of submission. So not a week later, but when you actually submit your application. FDA will evaluate each submission for prioritization when we receive it. So if you submit your application on Monday and without requesting priority review, and then on Friday you realize, you should have requested it, it's probably too late. We would probably have already processed your submission and assigned the goal date. This is why I'm emphasizing that if we really want you to succeed, and we don't want you to miss an opportunity because of a timing issue when you could have qualified for that shorter goal date. So second common mistake that we see is requesting priority review with each cycle. So what I mean by that is, sometimes applicants, they receive that priority review designation with their original submission, and they think, oh, this is great. Now I have priority for the entire life cycle of that application. So, you may have gotten that priority review, it goes through a review, and you may receive a complete response letter. When you respond back to that complete response letter, we've seen, certain applicants not requesting that priority review again at the time of, the next subsequent cycle. Please note that that prioritization may have been granted in the first cycle, but that doesn't necessarily carry over to the next cycle. For example, for drug shortages, we have a drug shortage list that's fluid. So you may have been, on that, you may have qualified for drug shortage in the first cycle, but in the subsequent cycles, it may have not been on that drug shortage list any longer. So it may not qualify. So, please, I just want to remind all of you guys again, with each submission, with each cycle, you need to explicitly request that priority review. Facilities. So if your facility has an official action indicated status or withhold status, generally you won't be eligible for priority review. Now, there are some exceptions to that. So for public health emergencies, those are evaluated on a case-by-case basis. So if you have outstanding compliance issues at your facilities, you need to resolve those before you can benefit from receiving a priority review designation. And then finally, cover letters. Just some things that you should watch out for. So your cover letter should be crystal clear. State that priority review request. Reference, that prioritization factor that you believe that you qualify for, whether it's, drug shortage, whether it's that new domestic onshoring pilot program. And then, be clear in terms of citing your ANDA number and including all your supporting documentation. Make it easy for us to identify and process your request. So in summary, the primary benefit of being granted priority review for your application is that you would obtain that shorter goal date or expedited review. So this translates directly to faster time to market. And if you can get your generic product approved and on the market months earlier, than your competitors, that's a significant competitive advantage. So some of the limitations that I just want to summarize are, just because you are granted priority review for multiple prioritization factors doesn't mean you get an additional benefit, meaning, you get the same priority review or same shorter goal date and as if you qualified under just one factor. Now, if there's also, as I mentioned, facility issues, you generally won't be eligible for priority review. And note that for minor amendments, those do not get a shorter goal date, but will qualify for an expedited review. So I just want to mention here about review classification changes. So if you fail to request priority review at the time of submission, you still get an extra chance to request it during that review cycle. And we can change that classification from a standard to a priority review, but the only benefit would be that expedited review benefit and not the shorter goal date, which is better. So we're not going to retroactively shorten the goal date if we later grant priority review, but we would change the classification from a standard review to a priority review. And then finally, managing expectations. So priority status does not guarantee approval, meaning review timelines will still depend on application quality. My advice is to remember that submitting a quality application is more important than trying to obtain priority review. Here are some resources, and for those in the room, I'll be around to answer some questions after this session. Thank you, and I'll have Heather come in for the next presentation.
Okay, good afternoon. I'm Heather Strandberg. I'm with the Patent and Exclusivity team in the Office of Generic Drug Policy. And I'm here to discuss some how applicants should address certain patent and exclusivity requirements and provide some helpful reminders. So today's presentation will review regulatory requirements related to the most common patent and exclusivity deficiencies, including addressing later listed patents, patent information, and new exclusivities, adequate documentation of notice of paragraph 4 certification, notification of the filing of legal action, court orders, and licensing agreements, and pediatric exclusivities. I'll discuss timely notification of commercial marketing and review the agency's P4 certifications list and competitive generic therapy approvals list. So one of the most common issues that we encounter is failure to address later listed patents and patent information, which includes use codes. An applicant must address all new timely filed later listed patents and patent information with a patent certification or a Section 8 statement. Patent and patent information, including a use code, is considered later listed if it is submitted for listing in the Orange Book after the ANDA has been received, but before it has been approved. For later listed patent or use code to be considered timely filed, it must be submitted for listing within 30 days of patent issuance or within 30 days of approval of a corresponding change to the NDA's product labeling. If that timing requirement is met, the patent or use code is considered timely and an ANDA applicant must address it. If you believe a newly listed patent or use code is untimely, meaning it was submitted for listing outside of that 30-day timeframe, please submit a justification setting forth the basis for why it should be considered untimely. So all of that is to say that all new and updated timely filed patent listings require new certifications and statements. So that also means that a prior P4 certification to a patent does not sufficiently address a newly listed use code that is listed for the same patent. Sometimes we see applicants that try to rely on try to address a later listed use code with by relying on the P4 certification provided to the patent before the use code was listed, instead of providing a new P4 certification to the use code. But a previous P4 certification to a patent could not have covered the new use code because the use code was listed after that P4 certification was provided. And it's important to note that litigation with respect to P4 certification to later listed patents and patent information does not incur a stay of approval. If an applicant elects to submit a P4 certification to a timely filed later listed patent or use code, the applicant must also comply with the notice requirements under 21 CFR 31495. This includes sending notice at the same time as submission of the P4 certification and providing adequate documentation of notice. Prior notice sent for P4 certification to a patent is not sufficient notice for P4 certification to a new use code for the same patent because that use code was not listed when the original notice was sent. This is true regardless of litigation status or outcome with respect to that patent. Applicants must also update their exclusivity statement when a new exclusivity is recognized for the relied upon listed drug. The statement should clearly state the applicant's intent and indicate whether the applicant is not seeking approval of its ANDA prior to exclusivity expiration and is retaining the exclusivity protected information in its proposed labeling, or the applicant is seeking approval of its ANDA prior to expiration of the exclusivity and is omitting or carving out the exclusivity protected information from its label. Documentation of timely sending and receipt of notice is required for P4 certification to all patents and patent information listed at the time of and after submission of an ANDA. Adequate documentation of notice includes documentation of the date that the notice was sent and received. FDA will accept as adequate documentation of the date the notice was sent a copy of the registered mail receipt, certified mail receipt, or a receipt from a designated delivery service. And we will accept as adequate documentation of the date of receipt of notice, a return receipt, signature proof of delivery by a designated delivery service, or a letter acknowledging receipt by the person provided the notice. Applicants can only rely on another form of documentation if FDA has agreed to such documentation in advance. So these are a couple of examples of return receipts issued by various designated delivery services, which serve as proof of delivery. So whatever service is used, the return receipt must contain the necessary details to verify delivery. The delivery receipt should include the delivery date and the location of the delivery and signature proof of delivery. Signature proof of delivery is required to ensure that the package was delivered to its intended recipient. The delivery receipt must clearly document the name or identity of the individual that received and signed for the package. An inadequate signature proof of delivery is a common deficiency. So this is an example of a delivery receipt from a designated delivery service that does not comply with 31495E. So first, the signature or the signed for by field states signature release on file, rather than providing the identity or name of a person that signed for the delivery. And then this receipt also states that a signature was not required at the very bottom, which you may or may not be able to see. And signature proof of delivery is always required as stated in the regulations. So first challenge question. An applicant provided a P4 certification to the 123 method of use patent and the associated U456 use code in its original submission. A new timely filed use code, U789, is subsequently listed for the patent. True or false, the applicant must address the later listed U789 use code with a patent certification or a Section 8 statement. If you thought true, that's correct. An applicant must address all new, timely filed, later listed patents and patent information, including use codes, with a patent certification or a Section 8 statement. And if the applicant elects to address the use code with a P4 certification, they must comply with the notice requirements under 31495, which includes providing documentation that notice was sent and received. So moving on to notification of filing of legal action. Applicants must notify FDA within 14 days of the filing of any legal action that is filed within 45 days of receipt of notice of P4 certification to patents and patent information that were listed at the time of ANDA submission. So that means that the requirement does not apply to later listed patents and patent information, and that's because litigation does not incur a 30-month stay. Applicants should ensure that notification includes a statement that an action for patent infringement identified by a court, the case number, and the patent numbers of the patents at issue has been filed in an appropriate court on a specified date. A common issue that we see is the submission of a dummy case number. So instead of the actual civil action number from the complaint, so please always provide the correct civil action number from the original complaint. And submission of a complete copy of the first civil action complaint with civil action number is an easy way to meet all of the requirements. And even if you later provide a dismissal or another order that contains the civil action number, we still need the original complaint's civil action number to match and properly track the case. And if no legal action for patent infringement has been filed after the noted 45-day period, applicants should submit an amendment to notify FDA after that 45-day period has ended. So applicants must also notify FDA within 14 days of the entry of all final court orders or judgments. The regulation also states that applicants that have been granted a licensing agreement must submit a statement that they have been granted a license to market the ANDA product prior to expiration of the patents in suit. And this is particularly important for applicants that have an order that suggests or indicates patent infringement. Applicants will often tell us that they've entered into a settlement agreement, but then they fail to comply with the regulation by also stating that they have a licensing agreement for those patents. So please include this information because those two terms are not interchangeable. So that brings me to pediatric exclusivity. Pediatric exclusivity is a 6-month extension of the period during which the approval of an ANDA may not be made effective. The period is added to the end of certain listed patents and exclusivities, but there's an important difference between the two. Period is considered an extension of the exclusivity, so applicants that have chosen to carve out information protected by the exclusivity may do so for the duration of the exclusivity and the 6-month pediatric exclusivity period. Conversely, the pediatric exclusivity period is not an extension of the patent. So therefore, it is not permissible to submit a Section 8 statement to carve out the 6-month pediatric exclusivity. So when an applicant has provided a Section 8 statement to a patent that also has a pediatric exclusivity period attached, upon expiration of the patent, the Section 8 statement is deemed to be a Paragraph 2 certification. The Section 8 statement does not extend to the pediatric exclusivity period. So once the patent has expired, there is no longer a legal basis to omit information protected by the patent from the ANDA's proposed labeling, and the information must be added back. And lastly, when a pediatric exclusivity is added to a patent after submission of your ANDA, you do not need to submit a new certification or statement to address that exclusivity. Challenge question 2. An applicant provided a Section 8 statement to the 123 method of use patent and associated U456 use code and submitted labeling to omit the protected information. The patent expired on April 1st, 2026 and has an attaching pediatric exclusivity period that expires on October 1st, 2026. Which of the following is not true following expiration of the patent on April 1st? A, the Section 8 statement is deemed to be a P2 certification. B, the applicant can maintain its labeling carve-out of the information covered by the expired patent and use code. Or C, the applicant must revise its labeling to add back the information covered by the expired patent and use code. If you thought B, that is correct. Upon expiration of the 123 patent, the Section 8 statement is deemed to be a Paragraph 2 certification. The Section 8 statement does not extend to the pediatric exclusivity period, so once the patent has expired, there's no legal basis to omit information protected by the patent from the ANDA's labeling, and the applicant must submit the labeling to add back the information that was covered by the expired patent and use code. So now I just want to provide a few reminders about the timing of notification of commercial marketing. 180-day exclusivity may be awarded under 2 statutory frameworks: patent challenge or PC exclusivity and competitive generic therapy or CGT exclusivity. The requirements and recommendations for commercial marketing are different for each of them. With respect to 180-day PC exclusivity, a first applicant, as defined by the statute, must provide FDA with notice within 30 days of commercial marketing, which includes marketing of an authorized generic. And for notification received outside of 30 days, the date of first commercial marketing will be deemed to be the date of the drug product's approval. Oops. With respect to CGT exclusivity, the first approved applicant, as defined by the statute, for a drug designated as a competitive generic therapy is eligible for 180-day CGT exclusivity. The first approved applicant's commercial marketing determines when and whether and when the 180-day CGT exclusivity period begins, and whether that first approved applicant has forfeited its eligibility for exclusivity. Marketing must commence within 70 days of the approval date, and if not, the exclusivity is forfeited. And until we get this notification, we will continue to approve ANDAs because CGT exclusivity does not block approval of other ANDAs until marketing has commenced. So because the orderly operation of CGT exclusivity requires that FDA know when a first approved applicant commences commercial marketing, FDA recommends that first approved applicants let FDA know on the same day that they commence commercial marketing by sending an email to the PET email box and by also sending a correspondence to the ANDA file. And finally, I just want to remind you of a couple of FDA's public resources. First is the paragraph 4 certifications list, where FDA regularly publishes information relevant to 180-day patent challenge exclusivity. And generally, this list describes drug products for which one or more substantially complete ANDAs containing a P4 certification have been submitted to FDA. The list is updated bi-weekly and as needed. And then the next one is the CGT approvals list. This is a public list of all approved ANDAs for drug products that received a competitive generic therapy designation, and also notes which ANDAs cover drug products that were also eligible for CGT exclusivity, whether CGT exclusivity was forfeited, and the date of commercial marketing of ANDAs with CGT exclusivity. And this list is also updated bi-weekly. So that brings me to the end of this presentation. It's very important for applicants to adequately address patent and exclusivity requirements to avoid delays during the review of their ANDA. And that includes addressing all new timely filed later listed patents and use codes, and complying with the notice requirements for P4 certifications, updating exclusivity statements to address new marketing exclusivities, providing timely notification of filing of legal action and outcome of litigation, submitting a statement in compliance with the regulations if granted a licensing agreement, and awareness that pediatric exclusivity is not an extension of a patent and cannot be carved out. You're reminded to notify FDA of commercial marketing of exclusivity eligible ANDAs and to take advantage of the information that FDA publishes on the P4 certifications list and the CGT approvals list. And I will hand it off now to Trung.
Thank you. So my name is Trung. I'm a lead pharmacist in the division of Orange Book. And today I'll be talking to you about the how the Orange Book listings are added, updated, and maintained, basically like our workflow. Objective is, by the end of the session, is to understand workflow behind the Orange Book entries, describe common updated triggers like supplements, patents, exclusivities, therapeutic equivalents, ownerships, marketing status, and etc. Understand the Orange Book publication cadence, where we have daily, bi-monthly, monthly, and our annual to view data changes and know how and where to request these updates. So just a bit background about the Orange Book. The Orange Book was distributed as a proposal in January 1979. It included only currently marketed prescription drugs products approved by the agency through the new drug applications and the abbreviated drug applications under the provisions of sections 505 of the FD&C and DAPA's regulation at that time. In 1980, FDA published its final rule, therapeutic equivalent drugs, availability of lists and amendments under 21 CFR 20.117, and simultaneously published the first annual edition of the approved prescription drug products with therapeutic equivalence evaluation, otherwise known as the Orange Book. And then in 1984, we have first time written into statute under FD&C Act amended to require the FDA to publish and regulate updates in the Orange Book. 1985, the sixth edition of the Orange Book, we provided the discontinued drug products designation by characterizing by a special symbol. And then in 1987, in the 7th edition of the Orange Book, we added the discontinuation section. In addition to that, we have the Orange Book list of OTC drug products that has been approved in NDAs and ANDAs. And finally, we also included the first patent and exclusivity information addendum. And currently in 2026, we published our 46th edition of the Orange Book in mid-January. So on this screen, this is our snapshot of the Orange Book, and I'm using ibuprofen as an example. Why this matters to industry: product listing provides important information to prescribers, pharmacists, and other parties. It basically shows you the marketing status. For instance, for this one is our prescription drugs, OTCs, and discontinued, approved product, proprietary name, drug formulations, route, and strength, therapeutic code, whether the drug is designated as a reference listed drug or reference standard designation, current applicant holder, which can change over time, example, because of transfers or mergers. And new line listings are generally added to the active section under the approved prescription or OTC unless stopper is notified before publication that a product will not be marketed within 180 days of approval as consistent with the 506I(b). If a product will not be marketed at any time, a timely notification from the applicant holder can keep it out of the active section update cycle. Note that drugs that are not in the Orange Books are tentative approved drugs, OTC monograph drugs that are not approved under NDAs or ANDAs, drug marketed under biologics, BLAs, or approved drugs that are discontinued market prior to 1987. So where does the Orange Book data come from? It comes from new drug product line listings, like from the original approvals, 505B, NDAs or 505Js, ANDAs, certain types of supplements, therapeutic equivalent evaluations, reference listed drugs, and reference standard designation. It also comes from patent submission and exclusivities. And also, we also have updates to the safeties and effectiveness determinations for discontinued drug products. And finally, revisions to the listing information may come from marketing status notification 506Is, and ownership changes and name changes, which you can reference in 21 CFR 314.72. Patents of exclusivity, what gets listed and how it's listed. NDA holder submits the patent information through form FDA 3542. And then DAPRA reviews it for technical completeness as a ministerial role. We recommend you use the most current form that is found on the FDA form website, because that form gets updated. Orange Book records will record the patent fields under patent number, expiry dates, drug substance, drug product, patent use code, otherwise known as MOU. We also have patent submission dates and delisting request flags where applicable. As for marketing exclusivity, the Orange Book is the official reference for information about marketing exclusivity periods associated with the listed drug. We would like to note that recently we published the new clinical investigation exclusivity, otherwise known as the 3-year exclusivity for drug products, questions and answered, and that's dated in March 2026. How this information is disseminated: patent listings are published in the Orange Book daily. Available changes are also made in the monthly CS files and annual editions as of December 31st, and you can find that in the patent text and exclusivity text download. So marketing status changes, how products gets moved to the discontinued section. This gets moved by the applicant 506I notifications to the agency withdrawal, acknowledgement letter, correspondence like control correspondence. Marketing status notifications are under section 506I of the FTNC Act. DAPRA may also move products to the discontinued section based on DAPRA's marketing awareness for example, annual reports or sponsors' email correspondence. For non-marketing drug products that are in the discontinued section, DARPA requests the sponsor to provide a request as soon as possible to ensure a timely publication. Discontinued products do not carry reference standard, designation, or therapeutic codes in the Orange Book. As a reminder, for 506I, application holders must notify DARPA about withdrawing from sale, generally 180 days prior or by withdrawal date if practicable, and about products not available within 180 days of approval. So how often does DARPA update the Orange Book, and what are the cadence in each of these time? So daily website updates focus on newly approved generics and patent listing posting. Since February 2005, DARPA has provided daily electronic Orange Book product information for new generic approvals to keep substitution relevant information current. Updates are posted in the afternoon on a regular business days, Eastern Standard Time. In addition to that, we have our semi-monthly website update, which is the broader maintenance changes. Typically on a Monday, DARPA updates additional Orange Book website information, including things like drug approvals, ownership changes, marketing status updates, exclusivities, and various types of changes to drug listing information, as I mentioned prior. In addition to that, we have our monthly cumulative supplements, which is a monthly data file refresh. That includes monthly data file updates, monthly website updates, and monthly print updates, otherwise known as the cumulative supplements, generally occurring at the end of the second week of the month and reflects all changes that occur in the prior month. DARPA also notes the Orange Book downloadable data files are updated monthly. In addition to the monthly, we have our addition and deletion pages as change log. DARPA publishes a monthly list of the new additions and deletions to the prescription and OTC drug product lists as a data file, but not as the full cumulative listing, and has instructed users to use this alongside the most current cumulative supplements and Orange Book. The page explains symbols like the addition or deletion or the @ or exclamation symbols for certain non-marketing status. And then we have our annual edition, which the Orange Book prints annually to incorporate new approvals and other appropriate data changes as part of DARPA's continuing update cycle. It includes Orange Book preface, drug product lists, addendums, and appendices. Sorry, missed over that. So here is our landing page for the Orange Book. And if you click on the Search Orange Book database, you'll come to this. This is where you can search the information for the drug product, the applicant holder, dosage form, and so forth. But I would like to point to you right in between the search for route and administration and patent. There's a little excerpt in there that says to download the Orange Book product information into a spreadsheet that can be searched by additional field. You would click on that instruction link and it'll show you how you can download that information for your own use. And if we go back to our landing page, if you look down to the bottom, it says additional Orange Book resource. And if you click on that, this is where you would find the Orange Book data files that are in compressed zip files. And once you download that, you'll get our product text listing, which includes the ingredients, dosage form, route, strength, trade name, applicant, NDA, ANDA, product number, T code, approval dates, reference listed drug, reference standard in downloads and search output. And for products that have applicable unexpired patents or exclusivity, in the patent text, you get our patent number, expiry dates, drug substance, drug product flags, use codes, submission dates, delist request flags. And for exclusivity, we'll get the exclusivity codes, exclusivity expiration dates, links to NDAs and ANDAs plus product number. And to help you understand sort of our workflow and maybe guide you in answering some of your questions, we post. You can ask yourself, is it a timing issue? Meaning you might want to check the daily posting versus the monthly cumulative supplement data files and the CS monthly label to find if your products are updated or not. And then is it a field that requires an application action prior to being updated? Example, the proprietary name change generally require like an approval supplement. So if your supplement that for proprietary name change has not been approved, of course, we won't update that until we get that information. Is it a marketing status change where you would have to confirm if you would have to submit the 506I notification to us in order for us to take action? And then is it a potential discrepancy? So for example, do you see a different proprietary name or an applicant's holder name not updated? You can report that to our Orange Book email with the specific information like the application number, the product number, and expected versus the observed discrepancy that you see. So on to my first challenge question, which scenarios most directly results in the product being moved to the discontinued drug product list? A, FDA approves a label supplement for the product. B, the applicant notifies DOPR that the product is not marketed. C, a new patent is submitted on form FDA 3542. And D, the product receives a TE code. So if you answered B, that is correct. The Orange Book preface explains the products are generally added to the discontinued list when the applicant notifies DOPR of not marketing status and may also be moved via annual report or other actions. My second question is, which information has been provided as a daily electronic Orange Book update since February 2005? The A, is it the annual edition appendices A, B, C? New generic drug approvals, all line listing changes, including ownership and therapeutic codes, monthly cumulative supplements. So if you answer B, new generic drug, that is correct, as I mentioned in my prior slides. And then my last challenge question is, DOPR generally acts on requests to change a proprietary brand name in the Orange Book only after A, citizen petition is submitted, B, the applicant updates his website and label, the agency approves a supplement for a proprietary name change, and the next annual edition is published. So if you answer C, as I mentioned before, the Orange Book product information for new generic approval has been provided. We only make the change once the supplement is approved. And some key takeaways is that DOPR relies on different sources to update the information provided in the Orange Book. Examples like the agency's approval action letters or applicant notifications. Depending on the type of data or publication is updated. Our publication is updated daily, bi-monthly, monthly, and annually. And we advise folks to be proactive by keeping DOPR informed of marketing status and ownership changes. And here is some links, as I mentioned in my prior slides, that are useful for you guys. And just like to acknowledge our Orange Book staff and management on the incredible work that we do in providing, keeping up with the integrity and accuracy of the Orange Book information. Thank you. And I think we're going to move along to our question.
Thank you very much, Dr. Quash. I invite our speakers, everyone, to come up back to the table, please, and we'll start our Q&A session. Ina, go ahead. All right. Okay, let's see. In-house. Oh, we have a race. Come on, come on, come on.
Kind of had a question for Bailey, but maybe Heather could answer this one too. Regarding submissions where there are 3 or fewer approved, you said when there's a blocking patent, you can't have a P3 or P4 regardless of litigation. If there was no litigation, let's say it was settled before it's even filed, is that still going to be considered a blocking patent? Okay, easy enough.
Oh, yes, it's still considered a blocking patent because it's listed in the Orange Book as unexpired. So it's still, even his question was if there's no suit, it's still blocking. So that might qualify under the P4.
Right.
That would probably qualify under, it depends on the status of 180 day, but that does happen a lot. We see them just put the wrong factors, but your example would probably qualify under the P4 application with the P4 certification.
And then if my understanding of the MAPP is right, you can file a method of use certification though to a blocking patent, and that, I think there's a little footnote in the MAPP that says that.
Then it's not a blocking patent,
yeah.
Right? Yeah, if it's been carved out, right? We might have to get back to you on that.
No problem. Thank you.
Right, because that's not a paragraph 4. So that does count. If you have a section 8, that qualifies. Yep.
Having some excitement in the afternoon. Thank you. Second question, please, sir.
Good afternoon. I have a general question regarding the Orange Book. Lately, it was noticed some RLD additional strengths have been added that were not previously there. What is driving this? It seems some of these RLD new strengths have the original RLD approval date.
So your question is asking why are we adding new RLDs?
No, more specifically additional potencies. For example, I have an application that has 2 potencies, and all of a sudden, last year, it now has 3 in the RLD. And it's even not in the PSG.
Yeah, I guess just in general, the Orange Book is constantly looking at the RLD itself, and some of these, I guess, RLDs may have different volume size or so forth. So we're looking at that, and that's why you're probably seeing the expansion of like strength and so forth. So, I mean, that's one scenario that I can see. But if you have a specific example, like, question for a specific RLD, you can email the Orange Book. And
All right, thank you. I appreciate it.
Anyone else? Anyone else in the room? Okay, let's move on to online, please, Corey.
Of course, our first one is going to be for Commander Fine. The question is a suitability petition required when an ANDA applicant proposes to use a container closure system that differs from the reference listed drug, or RLD? For example, if the RLD is supplied in a vial, can a generic product be submitted in a pre-filled syringe without filing a suitability petition?
Thank you. That's an excellent question. So the short answer is it depends, and I'll explain why it depends. So a container closure change by itself is not a petitionable change in a suitability petition, but what is a petitionable change that could impact this question is a strain change. So strain change, especially for injectables, is defined as concentration and total drug content. So if you are proposing a different container closure system that has a different total drug count, for example, or a different concentration as the RLD, that would require a suitability petition prior to submitting an ANDA. But if your product has a different container closure system, but has the same strength and the same same strength and same concentration, that would not require a suitability petition, and that type of change could be submitted as an ANDA.
Thank you, Andrew. I forgot my manners. I have to introduce our newest addition to the panel, and that is Dr. Rosie Pagaduan, and she is the Supervisory General Health Scientist at the Office of Regulatory Operations within the Office of Generic Drugs. Welcome, Rosie, to the panel. Okay, we do have a question in-house, please.
Great.
presentations today. I was just wondering, with regards to the general discussions of the maximum daily dose and some of the strength and dosage changes, whether or not there've been additional studies required from the investigators. For example, in a lot of cancer drugs, when they're testing the initial dosages required for potency, there are effective concentration EC50 and IC50 determinations, whereas in those cases, your IC50 might be higher than your EC50 as compared to other medications, for example, like ACE inhibitors and ARBs, whereas if you set the same MDD, the toxicity is going to be reached for ACEs before it's reached for ARBs. Additionally, in some other medications, such as diuretics, there's new research stating that you have epigenetic level changes, which are going to be significantly at lower doses likely than even at anywhere near the toxic dose or the MDD, whereas things like eplerenone, for example, can EZH2 can reduce EZH2 expression. So I think when considering the full spectrum of efficacies, inhibitory concentrations, maximum daily dose determinations, and also like lethal dose considerations, what level of evidence have you all considered that's required for a lot of these new pharmaceuticals, as well as some of the old generic drugs, to validate the level of toxicities and the range of effects that can have within the human body, as well as some of the animal studies before that?
Thank you. No, it's a great question. Unfortunately, the technical components of applying appropriate MDD for the scenarios you're describing, we don't have the expertise on this panel to answer that question. If you have a specific MDD question and how it's going to be applied and some of the details you provided, that there's avenues to submit and get some feedback about the appropriate MDD to use and how you're applying that MDD. So I would just advise that you could submit a controlled correspondence with some of the details if it's application specific that will allow you to get the best advice possible.
Thank you. This was just in general in terms of making sure that there's essentially dosing and strength equity for a lot of the patient populations, because that's required. But thank you so much.
Thank you. Okay, Corey.
All right, our next question will be for Rosie. Can a product with multiple strengths approved under 2 different suitability petitions be approved in one ANDA?
Thank you. That's a great question. So the short answer is yes. However, you do want to ensure that both of those suitability petitions reference the same RLD. And then you would cite both of those petitions in your ANDA as the basis of submission along with the RLD.
All right, and then my next question will also be for you, Rosie, as well. Are suitability petitions subject to GDUFA user fees, or are they fee exempt?
Suitability petitions themselves are exempt from user fees, but of course, once an ANDA is submitted, that ANDA is subject to GDUFA fees, user fees.
One simple question. For an injectable product, is a suitability petition always required for change in fill volume, or are there situations where it can be managed through a change control?
So for injectables, Andrew somewhat touched on this, but yes, a change in fill volume would require a suitability petition if the RLD does not have that fill volume. So parenteral products are a little weird in a sense where it could be a change in total drug content, which would be the change in fill volume, or a change in the concentration itself. So that would require a suitability petition.
Okay, anyone else in the room? Okay.
Is there any plan to update the list of suitability petitions on FDA's website, or it's always through regulations.gov, which is very difficult to kind of navigate?
I agree with you there. It is a little challenging to navigate regulations.gov. One tip is to, you want to type in that petition number exactly in order for it to pull up if you're looking for a particular docket. As you mentioned, some folks may not be aware that there was a database or a log of suitability petitions at one point in time. It hasn't been updated in quite a while, and we've gotten this request from industry. It's something that we are looking into because it would be beneficial for industry to see all the approved suitability petitions. It could promote, further ANDA submissions. So that is something we'll take back.
Thank you, Rosie. Corey, please, online.
Of course, our next question is going to be for Heather. So if I select multiple prioritization factors because we believe they all apply, will FDA review all of them?
So we will review all of them. I'm sort of answering for Bailey, I guess. We will review all of them, although only one is needed to get priority review, and having it being meeting criteria for multiple isn't going to get you any extra benefit, I suppose.
I see. Thank you for that. And then one more question for you, Heather. Why do I need to send notice of PIV certification to later listed use codes for a patent that we were already granted a license to?
Short answer is that there's not an exception in the CFR for not having to do so. So anytime you submit a P4 certification to a new use code, even if that patent has been litigated in the litigation, you have a settlement agreement and you have a license to that, you still have to send your notice and comply with the reg, just simply because there's not an exception not to.
All right. This next question will be for Trong here. Does discontinued status marked for any RLD in Orange Book always mean withdrawn status, or sometimes discontinued status means that they are not having market availability of that product anymore for a reason other than the safety aspect of that product? When does the RS assigned to some generic products for those types of cases? Could you give an example?
When RLDs are discontinued, or even if an ANDA is discontinued in the discontinued section, it's more of a likely because of a marketing status change. And once an NDA gets discontinued, of course, as I mentioned, the reference standard and therapeutic codes gets. Oh, sorry. It gets dropped, and then we would reassign that reference standard to an therapeutic equivalent ANDA if there's one available.
Excellent. Thank you for that. This next question is, what is the typical response time for Orange Book email inquiries? Also for you, Trong.
We hope to do that within a reasonable time. Usually we say 7 to 10 days. If you email it, if it's more of a complex, then it might take longer, and we'll let you know if we need a follow-up.
Excellent. Then my next question will be for Commander Fine. Why would firms go for a suitability petition pathway over a 505(b)(2) route? Thank you.
I can't answer what a firm would do, but I will say that just as a point of clarification and just make sure we're on the same page. So a change that, the allowable changes under suitability petition, each of those changes are also suitable to come in as a 505(b)(2) because they differ from the RLD and they're not pharmaceutically equivalent. So a firm has that choice. Well, if I want to if we took the example from my presentation that fluoxetine 30 milligram capsule, we approved that as a petition, a suitability petition, and ANDA can now come in under an applicant come in under an ANDA citing the RLD as well as the approved petition. Alternatively, that applicant could also submit that same change 30 milligram capsule as a 505(b)(2). The answer to all your questions is often money. So traditionally, the ANDA pathway is more affordable and allows access to an easier access to some of these changes versus some of the maybe uphill challenges, whether it's financial, others that were required for a 505(b)(2). So it's really applicant specific on what pathway is preferred for you. But again, both those changes could come in as either a petition ANDA referencing a petition of an approved petition or as a 505(b)(2).
Okay, any questions in-house? We still have some time. Okay.
Under suitability petitions, you had some information about when PREA is waived and when it's not. Can you comment on which ones apply to which kinds of changes?
So for the changes that are allowable under a suitability petition, all changes except strength changes would trigger PREA and require a waiver request within that suitability petition. Any strength change in a suitability petition does not require a PREA waiver as it's not triggered. So that hopefully that clarifies your question. So if you have a strength change, if you're proposing a strength change in a suitability petition, you do not require a PREA waiver because PREA would not be triggered.
Required when you sometimes waive, but
7%
of the time
yes.
you don't.
Yes. So thank you for clarifying. So, but the waiver requests are handled internally at the FDA. There's a committee that individually evaluates each request. That committee is comprised of pediatricians, regulatory experts, just to ensure that if that proposed change is going to be likely used in a pediatric population, that's one kind of trigger they use, whether something would require additional pediatric studies. If the product is not anticipated to be used or kind of unlikely to be used in a pediatric population, those are indicators where the waiver could be approved. Mm-hmm.
Thank you, Dr. Fine. Corey, back to you. We have just about 3 minutes.
Of course. These next questions, this one will be for Heather. So is there a penalty if we do not send notice on the same day that we provide PIV certification to a later listed patent?
So no, well, the regulation does not describe a penalty if the applicant does not send it on the same day.
Excellent. Thank you for that. This next question is for Trong, and it is, how do you contact the Division of Orange Book Publication and Regulatory Assessment?
You can contact via email, which is the Orange Book at fda.hhs.gov.
Okay. I know, we're squeezing in so many now. Then we'll come back to you, Trong. The current available form, FDA 3542, is expired. What form should we currently use?
Actually, we just published the unexpired form. But in general, please always use the current form that is on the form FDA form website, even though if it's expired or not, just use that current form.
One more? Okay. So this one will go to Rosie. What constitutes a complete petition for the purposes of starting the 6-month clock? And what are the most common reasons petitions are found incomplete, requiring an information request?
So a complete petition will include all the sections as outlined in the section for citizen petitions, because suitability petitions are a type of citizen petition. And so those sections were mentioned in the presentation, and it's also found in 21 CFR 1030. But in addition to those requirements, there's a couple other things that are required, and those are listed in 21 CFR 314.93. These are specifically for suitability petitions. So you'll need to identify the reference listed drug, provide a copy of the RLD labeling, a copy of the proposed labeling for the proposed drug product, and then if PREA is triggered, a waiver request to waive the requirements under PREA. And usually if those are the items that are missing, the ones that are related to suitability petitions. If we find that something is missing, the Division of Filing Review will issue an information request. And then once that response is received, we'll complete the completeness assessment and issue a correspondence indicating your GDF goal date for the petition.
Thank you very much, Rosie. A big hand of applause to our panelists, please. So after the excitement of the after lunch period, we are running a bit late, but we caught up some time. So it's 3:30. Let's plan on coming back here at 3:45. The kiosks should be open for this afternoon break, so get yourself a pick-me-up. And please come back. Don't run away. We have 4 more sessions, very interesting, and a final panel for the day. So see you in a bit. Thank you.
Yes.
Okay, everyone, thank you very much for returning. And we are on the home stretch of day one, and we have 4 very interesting presentations coming up. And we're going to start off with the overview of the FDA product-specific guidance PhD program, and that will be handled by Dr. Joseph Kotzibar. Then we will go down to decoding PhDs. It's all about PhDs for the first 3 presentations. Decoding PhDs for complex non-oral generics, and that will be given by Dr. Yan Wang, followed by Dr. MJ Kim, who will speak to us about PhDs in action, key lessons and common questions in oral generic drug development. And bringing up the last presentation for this session will be Dr. Sheila Rajesh, speaking to us about drug device combination products, a quality perspective. And as usual, we'll follow up with a Q&A session. So let's start, and will my first speaker come to the podium, please? Dr. Kotzibar? I always say it wrong, but I think I got it this time. Thank you, Joe.
All right, thank you, Brenda. And thank you everyone for joining today, both in person and online. Hopefully today has been great, full of a bunch of talks on generic drugs. So one thing I noticed with the earlier presentations is that a couple presentations referenced the product-specific guidance program. So many of you may be asking, what are these PhDs that are referenced in these presentations? So now you have a presentation here that's going to go specifically over all of the PhD program. So what we'll start with today is we'll go over and understand the FDA's PhD program, or product-specific guidance. I'll be using the acronym PSG quite frequently through this presentation, so just a fair warning, I would get used to the abbreviation PSG, and its role in supporting abbreviated new drug applications. We'll learn about the PSG process, the GDF 3 commitments, and the reasons for PSG revisions. We'll identify available PSG online resources, withdrawn PSGs, and the upcoming forecasted PSGs. And then we'll understand how to utilize PSG public requests process, the public comments, and GDF 3 teleconferences and PSG meetings to engage with the FDA. So we'll start with the basics first. What is a product-specific guidance, otherwise known as PSG? So these are going to be individual guidances that reflect FDA's current thinking and expectations on how to develop a generic drug product that is therapeutically equivalent to its reference listed drug, or RLD. They contain product-specific recommendations, obviously, aka PSG, as opposed to general guidances that will typically give guidance on broad topics or processes. These are going to give very specific guidance and methodology to help develop generic drugs and generating the evidence that will be needed to support the ANDA's approval. They include both key science and research outputs. And one of my favorite part about the PhDs, it's an extremely unique aspect to the generic drug program. But when you think about PhDs, it's not just a one size fits all for your bioequivalence approach. A lot of times within the PhDs, they will reference the regulations in 21 CFR 320, as well as other general guidances, again, covering those broader topics. However, the PhDs remain an integral part of the ANDA program. So I know we had a great talk earlier today on controlled correspondences. And additionally, I feel like this audience is familiar with the pre-ANDA and ANDA meetings. PhDs is another tool in that toolbox that can be that can help applicants in their quest for developing generics that are therapeutic equivalent to the RLD. It really should be the first resource you turn to when you try to decide to say, okay, what do I need to put into this ANDA package to make sure my product is going to get approved? PhDs are a great place to start. They're also based out of and funded partially through GDF research. So some of this research that is happening in-house, we're able to do these research projects and put these recommendations directly into these PhDs to help industry. They've helped both internal reviewers as well as external applicants guide their ANDA programs to figure out which studies are needed for bioequivalence. So some background on PhDs to begin with. PSG started in 2007. Since then, the FDA has been publishing PSGs that provide clear and distinct recommendations to ANDA applicants. More than 2,400 PSGs are posted as of this month. And you can see over time, PSGs have somewhat stayed readily consistent in how many PSGs were published per year, about 100 to 200 a year of new and revised PSGs. There is a glaring outlier on this graph in 2024. So you may be saying, what happened in 2024 with the 814 M13A batch of PSGs? So that was a special batch posted in 2024 that revised 800 of our guidances to align with the ICH M13A guidance. So I won't go into depth today on that topic, but last year at the Generic Drug Forum, I believe there were a couple presentations as well as this one that covered some of the M13A updates. Last year, we did 162 revised PSGs and 130 new PSGs. And overall, of these 2,400 PSGs that are published, about 1/3 are for complex products. So when you think about developing, when you think about the PSGs and when to expect new PSGs to be issued, you really need to look at the GDUFA 3 commitment. This would be the most important driving factor for when to expect new PSGs to be published. So for non-complex NCE NDAs approved after October 1, 2022, which was the start of GDUFA 3, the GDUFA commitment says a PSG will be issued for 90% of those NDAs within 2 years. This is probably familiar because it has not changed since GDUFA 2. What was new in GDUFA 3, though, were for complex products. So the first change here is it's complex products approved in NDAs, not just NCEs. So all complex NDAs approved, again, after October 1, 2022, the start of GDUFA 3, the commitment says a PSG will be issued for 50% of those products within 2 years, and then an additional 25% to get us to that 75% mark within 3 years. And then, of course, the FDA will continue to develop PSGs for complex products that were approved prior to that GDUFA 3 start date. So now that we've had a couple years, to start the complex product goal dates, this was a hurdle that the agency needed to internally figure out, how are we going to make sure to hit these consistent goal dates for all of these complex NDAs? So one of these efforts, the SME Triage Team program for complex products PSGs, was initiated. So the STT program, again, the SME Triage Team, is a cross-office program that was established in 2021 to support the PSG development for newly approved complex products. It started as a pilot in 2021 before GDUFA 3, but by the time GDUFA 3 rolled around, it was fully operational. What this program does is it successfully connects research and review assessment with PSG development, achieving early and proactive identification of knowledge gaps and timely addressing technical needs. It enables cross-disciplinary collaboration for effective acknowledgement to maximize efficient use of the center resources. It contains several SME teams, typically by complex area, such as topical products, inhalations, peptides, etc., with membership comprising of experts from research, review, and policy. So it is truly interdisciplinary. It helps better plan for PSG development timeline for complex products to facilitate the generic complex product development and meet the GDUFA 3 goal dates. So how are we doing with these? So what is the impact thus far into GDUFA 3? So now we have a couple of years of GDUFA 3 data. We can kind of look back and see how we're doing thus far. So since GDUFA 3 started, and from October 22, again, to February of this year, 100% of the newly approved complex products have been triaged through this process, leading to 9 distinct research projects. For complex NDAs, thinking back to the GDUFA goal dates, 90% of products have been published within 2 years, and all of the PSGs for complex NDAs were published within 3 years. So we are definitely meeting and exceeding the GDUFA 3 commitment. Now that we have this data, we can kind of go back and reflect and say, okay, well, how did we improve compared to GDUFA 2? So we saw through some internal analysis that the percentage of complex products for PSG publications within 2 years essentially doubled from about 40% to 80%. In addition, the median duration of complex product NDA approval to planned PSG publication decreased from 4.7 to 1.5 years. After doing this analysis, we were really surprised and somewhat, elated at how good this data is and how well this program has been working so far. But there are other ways we prioritize and develop PSGs. It's not just the GDF commitments. So some other initiating events that may lead to a PSG development could be recently approved NDAs and supplemental NDAs, FDA analysis of products without PSGs. Believe it or not, with 2,400 guidances, we still have products without a PSG. And then working with industry through pre-ANDA meetings, public requests, comments submitted to the PSG docket, which I will cover on future slides, controlled correspondences, and citizens petitions. The data to support PSG development, pharmacokinetic and pharmacodynamic modeling, previous BE studies, NDA review and labeling, pharmacovigilance, and GDUFA-funded research outcomes. If you think back to my initial slide, that GDUFA research translates directly to recommendations in the PSGs. Another common question we receive is, how are these prioritized other than the GDUFA goal dates? So obviously the GDUFA commitment is going to be the #1 priority that the agency makes sure it's hitting and exceeding. But other than that, we also prioritize PSG development by stakeholder interest in ANDA submission, drug availability and access, public requests from the generic drug industry and other stakeholders, which I will cover on a future slide, and public health priorities. The PSG process is very cyclical. It happens multiple times a year, typically 4, where we post batches or standalone batches. To do this, the PSGs are gathered into a kind of a working log and prioritized based on those prioritization criteria, and then they begin to be developed. After they're developed and cleared, they'll get posted and published either through a quarterly batch or a standalone posting, and then seek public comment. After the comment period, the FDA can finalize the PSGs at their discretion, and then the process repeats, again, typically 4 times a year. When should you expect PSGs to be published? New and revised PSGs, again, are generally published quarterly. Typically, February, May, August, and November are the months that you can pencil in to expect the PSGs. As an example, 2 months ago now in February was our last batch of PSGs that was posted. Here again is what I've previously said, that the PSGs are published as a PSG may be published as a standalone guidance or a standalone batch. These are going to be outside that quarterly expected timeline. Many times it's to coordinate with citizen petition responses, to meet the GDUFA goal date, efficiency in developing products in the same class together, and then to coordinate with general guidance, publication, or revision. That last example, that standalone M13A batch that we talked about a few slides ago, is a really good example of the agency posting one of these standalone batches outside that quarterly expected schedule. However, anytime a standalone batch or standalone guidance or quarterly PSG batch is posted, the FDA issues notices in the Federal Register for every batch. So the past few slides are really focused on developing products for new PSGs. However, PSGs do sometimes need to be revised. As you saw in the initial graphic, for the past about 10 years of PSG data, we were typically posting about the same new and revised PSGs each year. So what are some of these reasons why PSGs need to be revised? #1, changes to the RLD, such as labeling update, supplements, new strengths, newly identified safety concerns, consistency with revision to general guidances, again, thinking back to that ICH M13A guidance that kind of reshaped how we think of in vivo recommendations for solid oral products. Comments received to the PSG docket, which we'll touch on in a couple slides. Citizens petitions, new BE approach from research, and new knowledge from ANDA assessments, pre-ANDA meetings, and controlled correspondences. Revisions are typically grouped into a few different categories: critical, in vivo major, in vivo minor, in vitro major, and in vitro minor. In general, critical and major revisions are the ones that you should take the note of. Critical, for instance, are PSG revisions that will include additional bioequivalence studies or evidence recommended to support FDA approval that reflects the change in safety or efficacy of the drug product. This would be a critical revision that would impact all ANDAs, even already approved applications. In vivo major and in vitro major are going to be revisions to the PSG that will add additional in vivo bioequivalence or in vitro bioequivalence that will be necessary for support of an ANDA approval. Minor is essentially everything other than those revisions. So again, there was a great talk earlier today on controlled correspondence. How does that relate to PSGs and PSG development? So again, controlled correspondences can be submitted through the CDER Direct's NextGen collaboration portal. These CCs can be triaged as what we internally call public requests. If the submitter is submitting or proposing a general bioequivalence recommendations for an RLD with no PSG. If the drug product is complex, however, submitters may send a pre-ANDA development meeting request. So what to expect if you submit a CC and then you get a response back essentially saying it was a public request. So what you'll see in that response back is the request for information related to the product-specific recommendation for generic drug development was sent to ORS for further processing. ORS will take this request into consideration and prioritize based on other prioritized metrics such as GDF goal dates when we think about how we publish PSGs. The other way to interact with the agency is leaving comments on PSGs. So again, I mentioned all PSGs are posted to a Federal Register notice number in the docket. FDA 2007 D0369. The notice will identify a comment period for the draft recommendations. Comments can be submitted to the docket by mail or electronically, and there's its own specific help desk within the regulations.gov online system. FDA does consider these comments when revising the PSGs. So now we're going to turn the page to some online resources to help guide industry and the general public using everything that we have on the PSG website. So the first slide I want to stop on is this slide. It seems jumbled. I will fair warning that the PSG page, the landing page, if you just Google FDA PSGs, will typically take you to this website. This slide here has some screenshots of all of the important information you will find on that website. I do want to draw your attention to the very bottom of that website. Here you'll see those resource links. This is where you could find other information such as withdrawn PSGs, the dissolution database, past presentations on PSGs, as well as the upcoming PSG or forecast list. This is a very important resource that, again, was touched on prior presentations that are going to be really resourceful for your generic drug ANDA programs. What this forecast list is a list of PSGs that describe the FDA's plan for all upcoming new and revised PSGs for generic drug products in the next 12 months. It enhances transparency in PSG development and will give the revision plan for products undergoing a revision. It updates include the PSG publication dates or a descriptive timeline as either beyond or within 12 months. Overall, the forecast list ensures consistency in FDA recommendations, decisions, and following previous iterations of the PSG and established principles for the PSG revisions to reflect accurate, sensitive, and reproducible approaches. The forecast list, like PSG batches, are updated quarterly at the same time. So when we post a batch of guidances, you can expect that forecast list also to be updated with those PSGs that were just posted will come off the forecast list. Products will be added on that are additionally forecasted a year out. So here's a quick example of what the forecast list looks like. A couple things to note is that we do have two separate forecast tables, one for new and one for revised. So this would be the new example. On the forecast list, again, you have the planned publication date of when you expect to see those PSGs live and any updates from previous iterations of this forecast list. We try to stay consistent when we forecast products that they will go out when they were expected to. However, there are times where we unfortunately have to delay PSG development. That can be transparently seen here. Last but not least, there's a search option to make it very quickly to find active ingredient or specific RLD or RS numbers. In contrast, here is the forecast list for the revised PSGs. One thing that's new is you have a planned revision category with description. So thinking back to my revision slide, this is where you'll see minor, major, critical, or editorial revisions. The one hidden thing on this website that I like to show are these plus signs on the left side of your API column. So this is kind of tucked away and sometimes not very obvious. But clicking on that plus sign also pulls in some information that you can see on the new list, such as the planned publication date of when the revised PSG will post, as well as any updates from that previous iteration of the forecast list. PSG website, on the other hand, is again a link you can either get to by using the drug database overall link from the FDA website, or specifically that FDA page I was showing a couple slides ago. The FDA PSG database is incredibly user-friendly and easy to use. You can search alphabetically or type by API or RLD. One additional thing that has made new PSGs readily available are these, at the bottom of the page, we always post the most recent batch of guidances. So once we post the batch, you can scroll down to the bottom and you will quickly see the new and revised batch here. So you can see in February 26, this would be the area to go to see those PSGs that were posted in February. Clicking on that, the drop-down link, you will see again then all of the PSGs listed. By clicking on the API naming, it will open the actual PSG itself. So here's an example of a PSG that posted last batch in February. I do want to note sometimes there is some other information tucked away in these PSGs that's very important. I know there were many questions and conversations on suitability petitions today. PSGs have started to proactively recommend suitability petition recommendations in the PSGs. Won't touch on that more, but I do think a future speaker will have much more in-depth discussion on the suitability approach in PSGs. Last but not least, the GDF 3 meetings and PSG TCONs. So ANDA applicants can request a PSG TCON anytime during their product development when FDA publishes a new or revised guidance if you meet two criteria. One, the BE recommendation is for an in vivo BE study, and two, the ANDA applicant has already commenced an in vivo BE study, and then that may be different from the PSG's new recommendation after the revised publication date. A prospective ANDA applicant should submit a request through the pre-submission PSG TCON or pre-submission PSG meeting electronically through the CDER Direct NextGen collaboration portal. And ANDA applicants can also submit a request for a post-submission PSG TCON or post-submission PSG meeting electronically through the Electric Submissions Gateway. Last year in calendar year 2025, there were 2 PSG TCONs requested, 2 PSGs granted, two PSG TCONs granted. PSG meetings. So how is this different from the PSG TCONs? So a PSG meeting can be requested following a PSG TCON if additional discussion is still needed. So this allows a forum to discuss the scientific rationale for an approach rather than the recommended approach in the PSG. Potential applicants may submit additional questions following the PSG TCON via CCs for any remaining issues. Applicants may submit a pre-ANDA or ANDA scientific meeting as an alternative to a PSG meeting. Applicants may ask additional questions that are not in the PSG meeting scope, if applicable, if they request that pre-ANDA or ANDA scientific meeting. However, we do recommend that applicants do not submit both a CC and request for a meeting at the same time with the similar questions. Last year in calendar year 2025, no PSG meetings were requested. So hopefully this presentation was helpful, giving you that high-level overview of the PSG program. In summary, some key points we touched on that I want to leave you with is the PSGs do reflect the FDA's current thinking and expectations on how to develop a generic drug product that is therapeutically equivalent to the specific RLD. The SME triage team, or the STT program, helps to identify and address any potential outstanding knowledge gaps and research needs proactively for PSG development for complex products, and has made quite an impact on our timely PSG publications, specifically for those complex products. PSGs are generally published quarterly or as a standalone batch, as we saw in that big ICH batch in 2024. However, all PSGs can be found on this forecast list if they're about to be developed or revised. FDA does keep resources online, such as PSG forecasting and past informative presentations, also on that PSG website I showed earlier in the slides. And last but not least, potential ANDA applicants are encouraged to submit public requests, request PSG TCONs and PSG meetings, and or leave public comments on those published PSGs when applicable. There's a lot of resources on PSGs available, so please make note of this slide. SBIA has done a lot of webinars and other discussions on PSGs, so there's many, many resources on PSGs out there. But thank you. I'll turn it over to Yan now for the next presentation.
Good afternoon, everyone. Thank you, Joe, really for the excellent presentation, which provides a very clear description on the scope of the PSG and the general process. So in my presentation, I'll dive further into the PSGs for complex non-oral generics with the goal to, at the end of the talk, hopefully you have a better understanding on the scientific and regulatory logic underlying the PSG development, and have a better strategy to systematically interpret PSG recommendations for complex non-oral products. And I have a few slides listing the most commonly asked questions and the responses on PSGs for your awareness. And then at the end, I hope you'll be able to translate PSG into a more actionable generic development strategies. So Joe already covered process-wise how PSG is developed. So here I'm going to share some more technical aspect as how PSGs are developed. How we view PSG, we view that as a optionized science-based pathway for product complexity to translate strategy into product development. So generally, we would ask 3 key questions when we first start a new PSG. The first question is, what makes a product complex? Then the second is, what are the critical attributes that dominate or control the product performance? And then the third is, what are the studies really best evaluates the identified product attributes to establish bioequivalence? So the first question, what makes this product complex? So we identify the source of complexity, and it could come from such as the drug substance is a complex active ingredient, or the formulation is a complex dosage forms. Then the route of administration could be considered as complex, such as inhalation ophthalmic, topical, intrauterine. Those are more as locally acting. And then lastly, but not least, the product could be a drug device combination, and the device is actually a very complex device. Nowadays, it's very often a product actually has multiple rather than one of those sources of complexity. Once we identify those, then we move on to the second question to really understand what are the critical quality or formulation product attributes that control the clinical performance. Because those are key things for us to really understand how is a product is designed and used to achieve the clinical performance, so there are proxies. So just a few quick examples, like for suspension products, right? Regardless is for injectable ophthalmic, it's well accepted. Particle size is one of the most critical attributes because it directly relates to drug dissolution and absorption. Then for liposomal product, again, the size is critical because that affects biodistribution, cell uptake, in vivo stability, then translate into exposure. Then for inhalation product, the aerodynamic particle size distribution is critical because that directly impacted the position of the particles. So move on to question 3, right? Once we go through the thought process, have a general idea as what are the critical quality attributes and how those are correlated with the product performance, then we would say what are the study type to really evaluate the attributes. And then PSG, most commonly, conventionally, the bioequivalence studies are more clinical based, such as B study with pharmacokinetic endpoint, comparative clinical endpoint, or sometimes pharmacodynamic endpoints. But with the increasement in complexity of the products, we start to, you start to probably notice more combination approach. And sometimes those complexities really put the conventional approach at a very challenging position that you could theoretically conduct an in vivo B study, but some of the practical challenges make the study design practically infeasible. So those are the cases we need to really dive into the critical, what are the critical physical chemical characterization in vitro performance. So we often nowadays would require a totality of evidence approach mindset to first define what are the pivotal BE evidence, either in vivo or in vitro or in combination. Then whether in addition to pivotal BE, do we need to have additional supportive characterization studies in many cases. And then we also are prepared to anticipate what could be some additional complications with the recommended studies. And if those occur, what would be the justification of bridging strategies? Those nuanced background may not always be reflected in the PSG, but we're trying to capture those as much as possible in our internal review. So move on to talk about how they view or hope a PSG can do for a generic development. So we felt like, first of all, with the STT implementation, our goal is trying to work on the PSG development as soon as the NDA is approved, or sometimes when we know it's near to be approved, to be ahead of the time. So company can use the PSG to really identify, first of all, if there is any regulatory expectations that the formulation needs to be quantitatively and qualitatively the same, or is there a need to have a no significant different formulation, be able to utilize certain novel BE approach, and then whether it's a drug device combination, is there a need to do a user interface analysis? Then also, if the PSG mentions some of the in vitro characterization, even they are not pivotal BE studies, the list can help company quickly identify the critical quality attributes as starting point to further investigate the development plan. And then the third, if in vivo BE studies are recommended, what are the endpoints, and are there any practical challenges? That needs to be further discussed with the agency to really make the study executable. And last is, because now we recommend in vitro studies, some of them are BE studies too. So it could help you to understand what are the expectations for retaining your samples. So the key thing is really the PSG is not just the recommendation. We view that as a development blueprint. And move on to next slide is the most commonly raised misinterpretation for PSG is PSG defines requirements. But in reality, from our perspective, PSG provides recommendations based on current scientific understanding, and alternative approaches or deviations from our recommendation may be acceptable with adequate justification. So really here the key is, if you have anything being different, as long as you have justification to support your deviation, then that will not necessarily lead to refuse to file or a failed proposal. But the key is really the justification. So in the next slides, I have two examples just to walk you through the PSG to share some underlying thought process. So better to hopefully help you better read the PSG. So the purpose is to really identify, the PSG to identify what are the BE options, what are the possible pathway to establish BE, what are the pivotal studies versus the supportive studies, and then whether there's any concern on feasibility. So the first example I'm using is buprenorphine. It's an injectable solution. Extended release is really a polymer-based in situ depot. So this product, Joe already showed the overall PSG page, so I'm taking section by section. So this section really, the purpose is to give an overview of the product, and then the recommended study session is to provide a quick summary of all the recommended approaches. So if you don't read the whole PSG, you still have a general idea as what is recommended here. And when approaches are recommended as options, then that means they are selective. So you can pick either one of the options. So moving on to the in vitro BE option. So here, for an in vitro B option, if we feel there is certain precondition on the formulation composition, usually is the case to be able to utilize this option. We would have the first session to clearly articulate what's the eligibility to follow this option. If the product itself by regulation has to be Q1, Q2 the same, then that usually is our criteria, meaning the Q1, Q2 applies to all inactive ingredient. But if the product doesn't have the Q1, Q2 requirement per regulation, then the language is softer, saying no significant difference, but Q1, Q2 is one example which can fulfill the requirement. That being said, although we put this as a general recommendation, if you have a non-Q1, Q2 formulation that is fileable and you believe in vitro approach recommended here is still applicable, then don't feel like automatically you are already not eligible. Then you should discuss with us. So then we have footnote indicating the manufacturing scale. For some products, if we foresee the later scale up could be a problem, then we generally clearly indicate in our PSG, we recommend a commercial scale for the batches for BE study. But most of the time, we indicate the commercial scale should be reflect the final, but we don't necessarily recommend commercial scale. Then the type of study and design really are trying to clearly indicate the role of the recommended study. So if it's a pivotal, then we will clearly call it in vitro BE study instead of characterization. So that means it's a pivotal study and the sample used there would require retention sample. So moving on to the next portion, which is the supportive characterization. I'm not going to read through all the studies in detail, but I will talk about some general expectations. So generally, we do recommend 3 lots of both the reference standard and the test products to be tested, because this really gives the data to allow establishment of a range for comparability purpose, because we generally do not recommend specific statistical requirements. So the comparability is really by comparing the range established by the ILD to the range of the test. So it's really to the applicant's benefit, right? If you test the 3 lots of the RS or more. But that being said, we recognize sometimes availability is an issue, and that's one of the frequent asked questions. If that's the case, then less than 3 lots of the R may still be acceptable as long as you provide evidence to support availability is a practical challenge. And then we always encourage the applicant to continue the effort on getting a new lot. But if you can complete your development on less than 3 lots, it may still be acceptable as long as you provide the evidence. And then if there's any differences observed on the data, then our expectation is you should provide scientific discussion to explain why you think maybe lead to the differences and why it doesn't matter to the overall product performance. And you could leverage from your in vivo study available or in vitro BE study. So the bottom line is you should try to make a cohesive discussion to show you really understand your product. And then as Joe mentioned, we're trying to act on research gaps early through GDF, and we publish papers like describe certain analytical methods, and when available, we include those in footnote for reference, but by no means you have to follow it. So move on to in vivo option. The strength really gives an indication as what and the dose what should be used to conduct the BE study. And we also provide additional comments to either articulate a specific PK design data analysis or clinical safety for subjects. And then if anything beyond what's described in the general guidance for BE, for PK analysis, then we would have a session recommend the additional PK metrics. In this case, it's a long-acting, so we recommend partial UC. Then moving on to the second case example, which is a drug device combination for glucagon auto-injector. So the complexity for this product is that the active ingredient is a peptide, and we generally consider that as an active, complex API. Then the peptide products would require rigorous impurity assessment and then also lead to potential immunogenicity risk assessment. This product does contain a complex device and is also indicated for emergency use. So when the API is complex, then in the PSG, the first session, we would provide specific recommendations on the comparative characterization studies to support API sameness. And then the footnote, we also include additional recommendations, either on sample preparation, literature resources, and other things, which I'll talk in the next slide. So in this case, for instance, we recommend biological activities, but we do have a footnote indicating if you find, you have additional information you could justify to skip this test, then you could provide that, again, to really show our recommendations are recommendations. You are allowed to have deviations. And then PSG is really probably the most time-efficient venue for us to reflect our updated scientific thinking. So in this case, the paragraph in the middle or at the bottom in the box really provide recommendation to reflect the updated thinking on the origin of the peptide source. Like historically, we have said generic peptide shouldn't be recombinant, but now we are open to that as long as the applicant could provide appropriate, adequate strategies. Then the footnote 3 further elaborate what are the potential strategies company could leverage to develop their immunogenicity development plan. Then moving on to the next part, because like I mentioned, this is a device, this is an emergency use product. So we do recommend additional in vitro BE study on the device performance. So here we have 2 in vitro BE studies on the device and 2 comparative characterization on the device. So the 2 BE study would require retention of the product, while the 2 characterization they don't. Then for every single drug device combination product in the additional information session, we would have specific detailed recommendation on how to evaluate the device component from a user interface standpoint. So that session is a clear indication to tell you if this product is a drug device combination. But that being said, we do recognize there are some PSGs for drug combo products that do not contain because they could be an older PSG. We just haven't had time to get to it. But for the new PSGs, if it's a combo product, we always have that session there. And this part really provides recommendations on evaluation of the device, and the information includes the description of the ILD presentation and its device constituent part, and recommendations on the studies to examine and characterize the design differences between the test device and the ILD, and guidance on conducting comparative analysis with reference to the relevant FDA general guidance. So given the time, I will skip through the next 4 slides, which lists frequently asked questions. But I included 16 questions here for the audience reference. It covers a really broad range of products, from the number of lots, from if it's multi-strengths but differ in different field volume, whether the study has to be in the US, a lot of different, and if for inhalation products, topical. So I encourage you to really read it in detail, but I wouldn't go over because of the time constraint. Just to summarize my talk, the PSG is really reflects a scientific-based decision framework. So we start with analyzing the product complexity to identify what are the performance drivers to develop PSG strategy accordingly. And the recommendations are tailored to each product based on their specific risk and uncertainty. So in terms of initial analysis, really understand the critical quality attributes are central because there are the mechanistic links between the product formulation and clinical performance. And B, approaches are really selected based on what is maybe the best approach to capture potential differences. There is increasement in terms of adopting a B strategy relying on totality of evidence. It could be a combination of in vitro pivotal and comparative characterization or a combination of in vitro in vivo studies. There is an increased emphasis on justified in vitro or other alternatives such as modeling approach, really to enable those development and discussion. So PSG provides a development blueprint, so it's not a constraint. If you have alternative with robust scientific justification, we always are encourage those kind of discussion with the agency, which lead to the last bullet point, early and proactive engagement is highly encouraged because success really depends on aligned science rationale between the generic and industry. So with that, I'll pass the floor to MJ.
Good afternoon. It's really late in the afternoon. It's really good to see you guys being focused, and I hope you can observe all these discussion points because as much as I've been at the agency for so long, I'm learning a lot from today's presentation. So I hope you're doing the same as well. In terms of this workshop, you got some surprises, which we weren't expecting, which was the FDA commissioner came to our workshop, and it was really pleasant to see him. So I hope you understand the nature and the importance of our work that we do at the agency, and it's really good to see you here. I've been with the agency for almost 25 years, it's about 15 years in the new drug domain, and about I'm approaching 10 years in the generic drugs. So what I'm here to present is from my regulatory experiences and how you may be able to benefit from what we do in terms of the PSG development. I kind of put some learning objectives, but I'm not gonna kind of go into very specifics because I think I cover some of the fundamental items about PSGs and what we usually recommend in my presentation about several years ago with one of the panel members who's sitting here, Dave, from the OGDP, our policy group. So I thought maybe I should kind of cover little different topics which may be relevant, and I hope you find them useful. What I try to capture is in terms of the PSGs and our recommendations, I've been struggling in terms of how do we kind of convey our message clearly to the applicants, and how can you benefit from our documents. So I try to shift from different perspectives. So each word that we put in our PSGs, I question my division folks in terms of what are we specifically recommending and what's the reason behind that. So with that in mind, I'm trying to kind of convey to you that we are trying to be as transparent as possible and try to convey our recommendation in a proactive manner instead of being reactive. So with that in mind, I put some learning objectives. So in terms of the FDA's key messages, once again, I'm trying to be as proactive as possible when we develop PSGs for oral drug products. And my division has been working on this PSG development for quite some time. So with that, you heard from Joe's presentation about M13A recommendations, how it's almost approaching 2 years, October of 2024, we actually updated PSGs of the IR products based on the M13A and some topics on the BCS-based bio waiver. We get that questions a lot. And the suitability petitions, you heard very great presentations this afternoon regarding the suitability petitions. So we are trying to be proactive as well on that topic. And cross-referencing, which I'm going to cover shortly, and then the narrow therapeutic drug index products. And once again, my goal is to be as transparent as possible and try to convey why we are recommending whatever that recommendations are in the PSGs. So with that briefly, this is the work that we are very proud of. The M13 implementations of the oral products, immediate release oral products, based on the M13A guidance that was published in October of 2024. We actually updated over 800 PSGs and going from 2 studies, fasting and fat BE studies, to 1 study, whether it's a fasting or one fat study, which was significant contributions, I feel like. And the so as of now, we do have a little more than 1,000. So we are approaching 1,500 PSGs on the IR products, where of that, about 77% recommend 1 BE study. So this is a drastic change from what FDA has done in the past, recommending 2 studies. So we are reducing the burden on the applicant in terms of submitting the ANDA for the IR products, where you can only submit the 1 BE study. In terms of the BCS-based bio waiver and PSGs, then this is really, really critical component. What we have in our FDA database is that we have a list of products where we have identified a drug product to be a BCS class 1 or 3. And based on that information, what we have done is we have revised our PSGs to reflect this information. So I hope you find that to be quite useful. However, what we are trying to proactively do is for those products that may be BCS3, but we don't have the data yet in-house, and we get these questions in CSIS a lot, because based on the BCS-based waiver, you can perhaps get the request for waiver for in vivo BE studies, or maybe you don't have to do studies in patients because of some of the restrictions behind it. So I put some link at the bottom of my presentation slides with some of the examples of the PSGs where we have a recommendations for the BCS-based waiver for those products. Or if you look at the second bullet point, which is the BCS-based bio waiver as an alternative approach. So what we are encouraging you to do is consider submitting the appropriate information, such as permeability data, so that we can review during the ANDA period, and then we can use that for the BCS3-based waiver. And the other proactive approach that we are recommending in our PSGs, you heard in the afternoon's presentation about the suitability. So in terms of the suitability-wise in general, as you know, we are getting lots of suitability petitions requests, and they are mostly related to the different dosage forms or with the strengths. When it comes to dosage forms, you heard from Andrew's presentation that they're mostly the ODT related. And in terms of the dosage forms and strengths, what we have done in the past is that our PSGs were not actually updated with that type of information, but we waited until first ANDA is approved with the suitability petition strength or dosage form, and we would put that type of information in our PSGs. However, what we are trying to do more proactively is that even before petition ANDA is approved for those suitability-related the dosage form or strengths, what we have done is that we already revised our PSGs to include that information, so we can give you the option. And I'm going to kind of show some examples. Once again, ODT. So the primidone is the ODT products, and I put two options here. So the first option is that you gotta do 2 BE studies for the using the designated RS for the tablet. So of course there's a tablet going from tablet to ODT. So in that case, that's when the petitioned strength, the dosage form is not approved. So you gotta do 2 studies, which is the typical recommendations in our PSGs. However, if you look at the second bullet point, it talks about how there's a 1B study recommendations using the approved ODT as a dosage form. So you have that option once the petition ANDA for ODT is approved. And this is one other example of trying to kind of be a little more transparent and try to be useful as possible for our PSG recommendations. We've been receiving some CCs about drug study design related questions. So this is PPI related. And when it comes to acid reducing agent, what is the typical product to use? What's the study durations? Things like that. So when we receive those types of CCs, we kind of contemplate because sometimes our general PKBE guidance may have some relevant information, but in some other situations, maybe our specific PSG can kind of give a little more concrete or specific recommendations. So if you look at the that's the PSG, we kind of put the study instruction when it comes to how to do a PPI study. So I gave that example here. But once again, we're trying to kind of balance how much of information to include when certain information may be already available in general BE guidance. But in some situations, when there is a specific study recommended, then we try to be a little more proactive and give us a little more information for your usage. The other topic that I want to briefly touch upon, and I do have perhaps 2 slides on this cross-referencing approach. The one really good example is bupropion, hydrochloride ER tablets. So there are 2 products available with the different strengths as listed in my slide. So one may use, if you already have a approved application for one product with whatever the strengths, line of strengths, you should be able to cross-reference those BE study findings for your other subsequent submissions for that product. So I kind of put that example in here. And the other example is the oral contraceptives and I'm not going to be going specifics, but there are several products out there with all those different strengths and combination of estrogen and progestins. So how if you can find out exactly what type of information you have in your package, then you should be able to cross-reference those BE study findings when you try to submit applications for FDA's review. So you might be finding this type of information quite helpful. And underneath the slide deck, I put more references for your information, and we are trying to utilize this approach for your usage. The other information, in addition to the PPI type of study that I briefly touched upon, when we put our recommendations in PSGs, we try to be a little more explanatory in terms of the study design and why we may be recommending some specific PK parameters. So I put that example here as well. So for example, if a drug is primarily intended to be released in certain part of the stomach, then in order to adequately characterize the PK, so we say certain number of blood samples should be obtained in order to adequately characterize the full PK profile, or making sure that we can capture the drug to be released in certain portion of the period. So we put that type of information for your usage, and I hope you find that information useful. The one other example regarding the transparency and all is that we have a good number of PSGs with the narrow therapeutic index drugs, and I put some examples on the right side of my slide here, and then of course we do have a very specific recommendations when it comes to the anti drug products and how to do a study. And underneath that slide, there's one reference, publication reference by one of our staff in ORS about our experiences when it comes to anti drugs and how we utilize the PSGs for these types of drugs. So with that said, what I am trying to convey to you is that when I'm looking at the PSGs from your perspective, when you read our PSGs, you may be wondering how come FDA is recommending this and that. So I try to kind of put my situation into your head and try to be a little more proactively transparent. And so I hope you find this presentation to be useful. But when it comes to the transparency, once again, so we have updated our PSGs with the latest information. But once again, you have heard from several speakers already that guidance, our PSGs are guidance, and it's not a requirement oftentimes. So I remind our staff members that guidances are guidances. And if you have alternative BE approach or different approaches, then you can always submit the justification along with your applications. So once again, just remember that FDA is very open to the alternative BE approaches or different recommendations. And we are hoping to be a little more transparent with our recommendations, and we do pay attention to your submitted CCs, and we try to reflect some of the CC inquiries to revise our PSGs, so that of course we realize that when we receive CCs from you with some specific questions, then we understand that there may be some needs for revision of our PSGs. So with that presentation, I'm gonna kind of stop here and welcome the next speaker, Dr. Rajesh.
Thank you, MJ, for the great presentation. Hi, everyone. I'm excited to share with you a quality perspective on drug-device combination products. I have a disclaimer to show you. this is what I'm going to cover. To begin with, I will give a brief introduction of drug-device combination products, then assessment team structure and process for generic drug-device combination products, drug product critical quality tests for parenteral products, recommended performance tests for drug-device combination products, common deficiencies we see in generic products, drug-device combination products, and in the end, we can look together some case studies. What is a combination product? Per 21 CFR 3.2e, a combination product is a product comprised of two or more regulated components like drug and device, biologic and device, drug and biologic, or drug, device, and biologic. Here, a component is also called a constituent part. 21 CFR Part 4, Subpart 4 establishes which current good manufacturing practice requirements are applied to combination products. For example, here for drug CGMP, requirements for drug CGMP requirements refers to the regulations in 21 CFR 210 and 211. For device CGMP requirements are set forth in 21 CFR 820 Quality Management System Regulations. Primary mode of action is the single mode of action of a combination product that provides the most important therapeutic action of the combination product. When drugs provides the primary mode of action, CDER is the lead center. Today, I will cover only about CDER-led drug-device combination products. As you know, therapeutic equivalence is equal to pharmaceutical equivalence plus bioequivalence. A generic combination product classified as therapeutically equivalent to the RLD can be expected to produce the same clinical effect and safety profile as the RLD under the conditions specified in the label. This does not, however, mean that the proposed generic product and the RLD need to be identical in all respects. However, before approval, we need to confirm that the differences in device and labeling for the proposed generic combination product are acceptable, and the proposed combination product can be substituted for RLD. Please note that this part of the assessment is done by Division of Clinical Review team, so I will not cover anything about comparative analysis and human factor studies today. There are three types of combination products. They are single entity combination product, co-packaged or kit, cross-labeled, and cross-labeled combination product. In the next few slides, I will give specific examples for each one of these. So single entity combination products. Here, components are physically or chemically combined or mixed and produced as a single entity. Pre-filled drug delivery systems and devices such as pre-filled syringe, auto-injector, pen injector, insulin pump, or IV bags pre-filled with the drugs all belong to this category. Co-packaged kit or kit. Here, two or more separate products packaged together in a single pack or as a unit. These are some of the examples. For example, drug in a vial packaged with a syringe, or drug filled in a cartridge packed with an on-body infuser, drug in vial plus syringe pre-filled with diluents. Another example is a first aid kit or surgical kit. The third category is cross-labeled. Here, the constituent parts are packaged separately but labeled for and required to be used together so to achieve the intended therapeutic effect. A classic example is this photosensitizing drug and a activating light or laser source. You are not alone here. As you already heard from the previous speakers, PSGs are available for complex products. In the case of complex drug-device combination products, we we recommend that the applicants examine the size and shape, external critical design attributes, and external operating principles of the RLD device when you design the device for your test product, because these are general device constituent features that can affect the user interface. This is an example of a complex drug-device combination products PSG I downloaded recently. So as you can see on the right side, under additional information section, details about recommendations for device and user interface assessment is given. Regarding review of assessment teams for ANDAs, once your application ANDA submission is received and accepted for review, OGD assigns Division of Clinical Review team for comparative analysis and related assessment. And labeling is also part of OGD. I am from OPQ IQA, Integrated Quality Assessment Team. Our team focuses on drug product quality. So the drug product assessors reach out to CDRH via inter-center consult request for device engineering and performance. In addition, Office of Pharmaceutical Manufacturing Assessment Team also consult with CDRH for device quality management system regulations and facility readiness. This demonstrates a collaborative and coordinated risk-based assessment of drug-device combination products. As stated in ICH Q8 guidance, the design and conduct of pharmaceutical development studies should be in accordance with the intended scientific purpose. The level of knowledge gained form the basis for science-based submissions and regulatory evaluation. Pharmaceutical development studies should contain at a minimum these following elements: quality target product profile, critical quality attributes, critical material attributes, and risk mitigation strategy. In addition, comparative physicochemical data and device performance data of test product with RLD is needed for drug-device combination products. In general, ANDA submissions must include description of the product, description of the manufacturing process and controls, and the test data to show that the drug product meet the specifications, including release and stability data. Each type of drug-device combination product should have its own characteristic critical quality attributes and administration-dependent design device performance characteristic. These are the tests recommended for an injectable solution, drug product quality test. As you can see, these are mainly USP Chapter 1 test, universal test, and specific test. These tests ensure that the safety, efficacy, and purity of this product physical stability evaluation of parenteral suspension products should ensure test to confirm that the particle size of the suspended particle does not change over time, and the particles, solid particles in the preparation can be readily resuspended to yield a uniform suspension. So these are some of the additional tests recommended for parenteral suspension product in addition to the test showed earlier. Together with this, you also need to pay attention to the general container closure system test given in the USP chapters 660, 661, 381, 1663, and 1664. In addition to these quality tests, applicant need to demonstrate that the device consistently achieves drug delivery functions to its specification. Performance of a container closure system refers its ability to function in a manner for which it was designed. Performance test and acceptance criteria should be appropriate to the dosage form, route of administration, and design features. The applicants should propose a control strategy for the characteristic of the device that relate to drug delivery. Certain characteristics can be controlled through upstream control, some through release, if not impacted by aging. In addition, test samples should also undergo preconditioning to evaluate in-use stability, stress, shipping, and aging. This is especially important for drugs used in emergency situations. For example, here in the case of pre-filled syringe, these are the recommended performance tests. Here, break loose force and glide force ensures the performance of the device. These are some of the tests. These are the tests recommended for pen injector, auto injector, and on-body injector. Here, some of the tests are common. For example, delivered dose accuracy and delivered volume. If the injector is automated, then activation force rather than injection force is the essential drug delivery device performance characteristics. In the case of auto-injectors, you need to make sure the delivery of the intended dose per the label at the site within a certain time frame. On-body injectors are used to deliver large volume of the drug during a fixed period of time. Hence, additional tests are recommended here. As you can see, injection time or dose delivered time is also important for on-body injectors, in addition to break loose force, glide force, and then adhesion peel force. Regarding stability study recommendations, at the time of ANDA submission, 3 batches of the product in the proposed container closure system for marketing. For injectors, 1 batch filled, fully assembled, and fully packaged in the container closure system, including secondary package and labeling. For other 2 batches, filled, however, not necessary to, the entire batch is not necessary to fill into the assembled and packaged. Only a representative number of assembled batch packets into the device. This is for stability studies, related studies, and for sample retaining. And stability orientation, primary batch in inverted position and an upright or vertical position. But you can choose the worst case orientation with appropriate data and justification and use it for routine stability studies. These are some of the common deficiencies topics I collected. Many times we notice that stability studies do not adequately address or consider the drug-device combination product as a whole. That means some of the device-specific tests might be missing. Here, delivered dose or dose accuracy and air bubble headspace assessment in PFS is important. There are many questions usually we see. Break loose force and glide force testing, inadequate peel force specification, or failure to evaluate temperature excursion effect, inadequate in-use stability, extractable and leachable assessment. Let's check the case study right now. Incompatibility issues between syringe and certain lure activated valves is a long-standing issue. FDA tried to address by labeling update and field alerts over the years. However, FDA continued to receive additional reports of adverse events related to syringe malfunction and clogging. In 2013, FDA issued a draft guidance to address these incompatibility issues with the syringe and LAV connectors. It is possible that a syringe that conforms to ISO 11040-4 standard may not fit properly to a connector that conforms to ISO 594-2 for connecting devices. So in these situations, we recommend the applicants work closely with their manufacturers and suppliers to address the issue. Just like in any other, any medical product, the overall application holder is responsible for any product-related issues. So we also recommend early interaction with the agency, requesting a joint meeting with the CEDAR and CDRH, and outline your proposed step to ensure the connectivity of the glass syringe when connected to a connecting device. I have two examples to show you. This one example here, the drug product is an injectable emulsion packed in a 1 ml pre-filled syringe with lure lock. This application we reviewed and approved last year. Same product to different applicants. The material used for the syringe barrel tubing is glass in both cases. In the first case, product A, lure lock adapter is made of glass, and for B, it was polycarbonate. In the case of product A, CDRH was consulted and Division of Clinical Review team was also involved. There were major deficiencies cited at the beginning, highlighting this field alert report and potential incompatibility issues. During the review cycle, the applicant provided data with the justifications for 3 connectors, including one with an internal pin design. And in addition, they included a warning statement to say that the syringe may not be compatible with the lure-activated valve connectors that have internal pin designs such as LAV with a clave design. And in the case of product B, before the application was submitted, the applicant submitted a control correspondence. OGD evaluated the comparative threshold analysis and found it is of low risk from a user interface perspective. So there was no need for any warning in the label. Last week I checked both the applications. So far there is no adverse events reported, so it's doing great. This is the last example. This one, OPQ addressed last year a consult regarding incompatibility issue between a LAV connector with a clave design and a glass syringe. This picture I took from Medication Safety Alert Report in 2024. Here, a nurse tried to connect the syringe to a needleless connector for IV administration. As the nurse was trying to inject the medicine, 2 scenarios happened in the case of issue A, causing the medication leak. In both cases, as you can see, the syringe barrel tubing was made of glass and the lure lock was plastic. So after the incident, for issue A, the applicant revised the package insert to include caution statement saying that incompatibility with the LAV connectors such as clave design. They also listed acceptable lure activated connectors. In the second case, actually the syringe wouldn't attach and fell to the floor. In fact, there was a warning statement saying syringes may not require needle or blunt. However, the practitioner may overlook this because of the small size, and it is also on the side of the box. After this reports, applicant submitted a CBE 30 and proposed to replace the existing glass barrel syringe with a new PFS, and supporting data was provided, and it was found that the internal bore diameter is within the range proposed by the manufacturer of clave connector. So there was no warning statement needed for the package again. So with that, I want to emphasize know your product and the RLD well to avoid delay in approval, understand critical quality considerations and device performance recommendations for drug-device combination products, communicate early with the agency. With that, I want to acknowledge my colleague Jinsu, my supervisor Brock Roughton, my Division Director Pahala Simamura, and SBIA organizers, and thank you all for listening. And I included 2 resources here, and I think we are ready for Q&A panel. So let me give to Brenda. Thank you, Brenda.
Thank you very much, Dr. Rajesh. Thank you, and let's invite our speakers to come and join the final panel for the day, please. And joining our speakers will be our guest of honor here to the panel, Dave Coppersmith, Regulatory Counsel in the Division of Policy Development with the Office of Generic Drug Policy at OGD. Thank you. So it's the last panel of the day. I thank you all very, very much for staying with us. I especially thank the people online, as I know they also have that factor of have been in different time zones. So thank you so much for everyone being here. And when you complete the survey, which you will get an email today with a link, please, put in your comments there. We're always looking to grow. We're always looking for feedback as how we could be better. Tell us what you liked about the location, tell us why you didn't ask any questions, things like that. So yeah. Okay, so let's start. Any questions in-house? Corey? Thank you.
This first question is for Dr. Kotsabar. Under what circumstances would FDA consider developing and publishing PSGs for products with discontinued RLD and no generics in the market?
Yeah, thank you for the question. It's a really good question. So the online question was, what circumstances would the FDA consider developing a PSG for a discontinued RLD with no RS available? So that does pose a challenge for generic industries if you can't go get the reference product, obviously, and there's no RS to fall back on. In those cases, there are PSGs that recommend alternative bioequivalence approaches with bridging studies on sometimes the same API but different dosage forms, often based on PB modeling approaches. I think right now there's only a few of those PSGs out there, but this will be something that you can definitely look for in the future for more of these PSGs, trying to provide a bridging option so generic industry isn't stuck when you just can't go get that physical reference product to conduct your bioequivalent studies on. Thank you.
If I may, I do want to add a few more comments to what Joe said. So first of all, we're going to have a workshop in May 3rd and 4th, if I recall correctly. And this specific topic of no available RLD or RS will be covered at the May workshop. So look for that. And second of all, we are very mindful about this challenge. So we are trying to address that proactively once again in our PSGs. So if you can kind of look at the, for example, the potassium chloride PSG, you can use that as one of the examples where we are trying to be quite innovative and creative, trying to link the tablet versus the suspension, that type of approach. And we do use alternative BE approach, utilizing the modeling, that type of approach. So the agency is very aware of this challenge, and we are going to be discussing at the May workshop. So you should come to that workshop. Thank you.
Thank you, MJ. Thanks for the invitation. Okay, so question in-house. Thank you. That would be good. You can detach it.
Yeah, kind of follow up, I guess, to that question. When a PSG for such a product has been pending for years and years and years, is there ever a decision made to just cancel issuing that as a PSG? Like, do PSGs ever get put on the chopping block? Is that a thing?
Yeah, it's a great question. Honestly, right now, no. We still have all of the PSGs that are available in the PSG database. There are the current recommendations that can be used. There may be some changes coming up, though, to where we start looking at those PSGs, the PSGs that are essentially inactive with no reference product available. I mean, they're essentially not useful to industry at that point until an RS comes back to market, or I mean, the RLD comes back to market. So I would say be on the lookout for more updates like that. But as of now, no, those PSGs will still remain in the database.
Thank you.
Thank you. So I think we have some tech issues with both mics. So.
Hello, my question is regarding USP 382. As that USP was implemented December 1st, 2025, does the agency expect ANDA holders to also do assessments for ANDAs approved prior to December 1st, 2025?
Like us to repeat the question?
That's related to the device, right?
Yes,
Yeah.
it's related to the elastomeric suitability testing of parental device delivery systems.
I know that there was the 382 USB 382. I read about it, but that part of it we usually consult with the CDRH, so they are the ones taking care of that.
Okay, all right, thank you.
Thank you.
Okay, thank you. I'm getting my exercise now in the afternoon.
Thank you very much. What a great presentation from all the speakers. I'm a former reviewer here at the FDA. I did a lot of post-approval changes, and some of the cohorts, the first cohort, second cohort for GDF. So I left the agency and moved to the industry. And I just have one question, and also in the next maybe 3-4 months, I'm conducting some scholarly research in generic drugs where we still have some disconnect with the FDA in terms of failure to acquire first-time right ANDAs. There's still numerous review cycles and stuff like that. I just want to have just a quick question from all of you. What do you see in terms of ANDAs not getting first time right? And when I say first time right is, you have PSGs, industry follows that, bioequivalence studies and results, everything is good. But then when it comes to the review process, there's still a lag. And then, some of them go even beyond the GDUFA time frame. What do you think is the really disconnect? And do you think the industry is not getting it right, or the FDA is overreaching in terms of regulatory requirements, or do you think there is no communication all along through the review cycles of an ANDA? Thank you.
Thank you for that great question. I think it's $1 million question. I hear what you say. I think there's really no one straight answer to that question. But based on what we have observed, hopefully industry could agree our product-specific guidance start to be more and more specific and informative with the goal to really ensure we can articulate our expectations in detail, right? So one thing we observed is sometimes certain deficiencies company get not because the studies cannot be done, simply because they were not aware of the expectations. So those things can be more easily mitigated by effective communication, either through the PSG or through other venue. That's why we keep emphasizing on the importance for developing certain complex products is really beneficial for the company to proactively engage with the agency. And there are so many different pathways, right? If you have specific, especially for certain discipline, then time-wise is most efficient to use CC. Or if you have a more complex multidisciplinary question that could involve, like, for instance, just the device itself, right? Involve the clinicians in like the device expertise and human factor potentially from the OGD demand path, then all the way to quality, OPQ, and CDRH, right? So the meeting is really an effective way to bring all the relevant parties to the table to have an engaged discussion to really make sure the company is on the right track. So one thing, like in terms of first cycle approval, there's also then for analytical method development validation, right? So there's also sometimes the ambiguity, like the company really has to, it's a learning curve, right? Depending on how experienced you are, either side, how you really present your data. And then one thing we see, even just in the PDAF space, company propose, here is my alternative approach, A, B, C, and D. They think it's very clear. But from our perspective, if you want to convince us of something being different, you can't just tell me what you want to do. You have to tell me why you want to do it and how you're going to do it, right? So similarly, in submission, many times the deficiencies could be simply because the details are missing, or sometimes companies have done the work already, but it's not presented in the correct way. So the initial assessment may not get exactly what was submitted, right? So then depending on the mechanism used, if IR was did or DRL can be quickly addressed, right, it may not lead to CR. But if those things are not in place, then many times, right, those could lead to a CR. And then council. So many things can be, so that's where we are trying to do the exercises internally to really identify what are the bottlenecks, right? If we see an application get stuck after 2-3 cycles, then now we proactively have certain program, we're trying to do the analysis to see what can be done to really moving things forward.
I do want to briefly touch upon your question so the others can also chime in. I want to be positive about where we are going. I feel like we are going towards the right direction. I feel like our agency, we've been very transparent and we are open-minded and we are having lots of opportunities for the interactions, including the ways that you can communicate with us, whether through the meetings or the PSGs or just, or even workshop approaches. Okay. So I feel like we are going towards the right direction. Maybe in the past, there was a relatively lower rate of first cycle approval, but I feel like we are getting there. So I want to kind of put that positive spin to it. If others want to contribute on this topic.
Yes, we are all positive. Everybody? Okay. Thank you much, Mijay. Thank you.
So because I do have a survey and part of the engagement is that I do get your feedback. Will it be possible if I send you so you can take part in my feedback, in my survey? It's not really proprietary in nature. It's a general scope of how the industry expect the FDA sometimes to function.
I think that's out of the scope of our conference here for the survey, but thank you very much. Okay, did you have a question, sir?
Thank you, everybody, for very informative presentations. Just on the topic of PSGs for complex products, I know GDUFA 3 doesn't require publication for PSGs on complex products that are subject to new chemical exclusivity, to NCE. But my question is whether or to what extent FDA prioritizes complex products, PSGs on complex products that are subject to NCE in order to allow ANDA filers, sufficient runway to develop their products, do their bioequivalence studies, gather their stability data, and get a complete ANDA in, within the 4-year time frame after approval of the RLD.
Yeah, no, thanks for the question. So to rephrase the question again, so you're right, the GDF commitment for the complex products, it's specific to complex NDAs only, not specific to NCEs. NCEs are included, though, into that kind of overall umbrella of all complex NDAs. So the NCEs will be under the same metrics all NDA complex NDAs are, the 55 or the 50% in 2 years, then the additional 25% in 3 years. NCEs will be prioritized. We want that PhD out within 2 years. The only thing that would really prevent that 2-year PhD publication date would be if there is ongoing research needed. And that's really why we have that 2-year, 3-year cutoff. It's when we have to have those kind of OPQ-led research projects before we can get a PhD in place. So I wouldn't say they're not prioritized like the non-complex NCEs have their own GDF goal dates. They are definitely still prioritized and included into that complex NDA umbrella. It's just not specific to complex NCEs only.
Thanks. Very, very helpful.
Thank you. Anyone else in the room? I know we had a preponderance of online earlier, so I'm trying to give the room opportunity. Go ahead.
Good afternoon. I have a general question regarding PSGs. So let's just say we received a complete response letter to do a failed BE study for a Fed condition, and the PSG was recently revised to only do fasting. Would we be held to that standard at and the filing, or the new PSG if we were to respond?
Okay, I'm sorry. If you can rephrase your question one more time.
So let's just say we have an ANDA that received a complete response for one failed BE study, which was under a Fed condition. And the PSG was revised to only have a fasting condition. Would we still be held to the standard at the time of filing, or the new PSG when we respond?
Is that more like a policy, Dave?
Oh, sure. Yeah, thank you for the question. So it could depend, obviously, if you conducted both the fasting and the Fed study, originally with the recommendations in the PSG, and the one was, adequate to demonstrate bioequivalence, there are some considerations that would allow that to be, sufficient to demonstrate bioequivalence with the, revised PSG. So it could be very fact specific though. So I can't give a blanket answer, but there are, it is possible, yes.
All right. One follow-up question. All right. Would it be more appropriate to, I guess, address that in a controlled correspondence, I guess, to get a finite answer?
So again, it's going to depend on the status. So if you received a CRL letter, certainly you could submit a control correspondence, depending on what types of additional studies you're looking at conducting. I mean, there's also the post-CRL scientific meeting pathway, which is available. But again, there's specific criteria for that. So it can really just depend on the best way. But yeah, the control correspondence is definitely one possibility.
Right. Thank you.
Thank you very much, Dave. Okay, we probably have time for 2 online.
All right. My next question, we'll go ahead and direct this one to Dr. Kotzabar. So why are historical PSGs removed after the old version is replaced by the new one? Is it possible to access the archived version somewhere?
Yeah, another good question. So the question is correct. So when we post revised PSGs, it's a new version of the guidance, and it's always going to be updated to show the current agency's thinking on how to show a generic is therapeutic equivalent to the RLD. So the previous iteration of the PSG would essentially no longer be valid because there is a new recommendation in place. So that is really the short answer why we don't keep the past PSG shown. That probably could lead to confusion from applicants being able to see multiple versions of this. And then to address the second part of that question, no, there's no archived of past PSG versions online available.
All right, thank you for that response. The last question I think will go to Dr. Rajesh. If the RLD was approved before a combination product regulation was implemented and a proposed generic is now considered a drug-device combo and there is no PSG, would a pre-DEV, pre-sub, or controlled correspondence be the best way to get a faster response to the sponsor's question regarding the HF studies?
Yeah, thank you for the question. That's good. I would say control correspondence. That would be faster.
Excellent. Thank you for that response.
Okay. Do you have one that's pretty straightforward? Well, we think might be straightforward. You can never tell.
Let's see. Let's see for Dr. Kim if this one will be pretty straightforward for you. If a PSG of an IR product recommends just one BE study after a fasting condition, is it still acceptable to conduct a study under a fed condition instead?
It really depends, and I don't like to give that type of response, but it really depends on how the drug is supposed to be taken per labeling, whether there is a safety considerations for conducting such a study under fasting or fed. So typically, what I post or what I recommend applicant is, like, if you know your drug formulation, the property, and if you look at the RLD labeling, you would basically know what type of study you'll be conducting from the applicant's perspective. So, like, once again, if you know your drug, you should know how to kind of go about, but it really depends.
Thank you, Dr. Kim. Okay, that brings us to a wrap for today. Thank you to our panel and to everyone else who presented and is still in the audience. So thank you very much. Again, thank you. Tomorrow we will begin at 9 a.m. Nice and fresh. The kiosk again will be open at 8 o'clock, I believe. My apologies if it was not open this afternoon. I am looking into why that was so. Again, if you pre-ordered, same thing, try to pay early so you can facilitate the line at lunchtime. And I know it was a long day, so thank you for sticking with us. I hope you get to relax tonight, and we shall see everyone tomorrow. Thank you.